Okay, so I think now I'm audible and visible to everyone. Yes or no? If yes, then just start commenting in the section so that I come to know that whether am I properly audible and visible to all my students. Beta, I hope, first of all, my question is, how was the paper? How was your paper, everyone? I just hope you all must have done great and the paper was extremely easy. Especially I'm talking about botany. This time in botany, 100%, not even one question was out of NCERT. Matlab, this is a record in the history of NEET examination. This is truly amazing. This is truly a record itself in the history of NEET examination that none of the question, at least in botany, was out of NCERT. And yes, and then one more thing to add over here. If you all have studied the last marathon which I took in the class, I think that was day before or 2 days back, 5 to 6 questions were directly from that marathon. So if students are attending the session, then I'm sure you must be knowing that whatever questions ma'am discussed actually were asked in your NEET paper. So see how that marathon was useful. Very good, Sunny. If you're scoring 180 on 180, that's amazing. I just hope that many of the students are really going to score 180 on 180. Absolutely damn easy paper. No doubts, no questions, no queries. Everything straight away from your NCERT textbook as well as from your notes. Of course, that has to be added. And today I am here to discuss the question paper. Code is G3. Very good, Aarti. So if you're scoring 170 plus, it's very good. I hope you get selected in your NEET examination and go to some good medical colleges, right? Chalo, so the paper that we are going to discuss today is G3. Very good, everyone. Okay. Reena, 135 is also a good score. Absolutely amazing. Chalo, so let's start with the discussion of botany. The first question on your screen, very easy question. You just have to tell what is the role of RNA polymerase 3. You know, beta, in case of eukaryote, there are 3 different types of RNA polymerase that helps in the transcription process. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. So here the activity of RNA polymerase 3 is asked, and you all know ma'am has already taught in her excellence batch that RNA polymerase 3 is used to transcribe snRNA, tRNA and 5S RNA. So the answer of question number 1 will become C over here, right? Yes or no? Now keep checking. Very good. Koyal, very good. Reena, very good, everyone. So whatever, keep answering the question and let's see how much you score at the end. Second question. Movement and accumulation of ions across a membrane against beta. Just focus on the term against the concentration gradient. Against means active transport. ATP will be utilized. All these 3 beta, they take place along the concentration gradient. They take place along the concentration gradient but not against. So answer will become 1. Very good, beta. Very good, Shravan. So easy question. Very good, Koyal. Next question. Upon exposure to UV radiation, DNA structure with ethidium bromide will show. वैसे तो this question belongs to zoology, but here I will tell you the answer. The color of the bands when they're exposed to ethidium bromide, they are appearing orange in color, right? So over here, answer will become A. Very good, everyone. Next question. The thickness, again the direct question from the chapter environmental issues. This question I took in my marathon session. I explained you that the thickness of ozone is measured in Dobson's unit. Yes or no? Isn't that very easy question? Direct question. I don't think anyone must be having any doubt in understanding this question, right? So answer over here becomes B. Very good Koyal, very good Sunny, very good Somik, this Baba, Car, whatever. You are scoring 190 plus. Amazing. Hats off to you. May God send you to AIMS College. Okay, very good. B is the answer. Next question. Among eukaryote, replication of DNA takes place where? Very easy again, beta. You know that when cell undergoes cell cycle, then there are two distinct phases, interphase and M phase. Interphase are again divided into three substages as G1, S and G2. And during S phase, the amount of DNA gets doubled, right? During G1, there is major organelle duplication along with protein synthesis. During G2, again, there is major protein synthesis. So, but where DNA replicates? Yes, answer is 3. That is S phase. I hope you all have understood this question. No, beta, Chandrakant, the answer is going to be C, not B. Somek, answer is B. Yes, very good everyone. Answer is C. Next, spraying of which hormone is going to fasten? Hasten, beta, means speeding up the process. Hastening means, beta, speeding up the process. So tell me, the speeding of the process for germination of seed is done with the help of which hormone? Gibberellin hormone. You know, gibberellin is known to break dormancy. It is used to promote germination in the seeds. So here answer will become C, gibberellic acid. It is responsible for seed germination. This is the direct line from NCERT. If you study the roles of hormones, if you study the roles of gibberellic acid, the direct line from NCERT. Students, very easy paper. Again, I am saying, अगर आपने थोड़ा सा भी ध्यान से पूरे साल पढ़ा होगा, then I'm sure you must all be scoring 170, 160, 180. Yes, of course. Now, moving towards the next question. Now here, beta, there are two statements. You have to tell which one is right and which one is not. Okay, very good. Tell me, beta. Statement 1 says the forces generated by transpiration can lift a xylem-sized column water over 130 meter of height. Yes, transpiration results in the development of negative pressure, which is called as transpiration pull. It is a very strong negative pulling pressure which is going to pull the water to great heights. You know, beta, there are only two ways to conduct water. One is transpiration pull, second is root pressure. Root pressure is a weak positive force which is not able to push water to some good height. But transpiration pull, it's a very strong negative pressure which can pull the water to some great heights. So first statement is true. दूसरी statement, beta NCERT की line है. If you study the roles of transpiration in your NCERT textbook, this statement is quoted over there. That due to transpiration, one of the important function of transpiration is perform cooling. Yes or no? As there's loss of water, it's kind of plant is sweating. So what happens when you sweat? Your body temperature cools down now. Then same thing happens with plants as well. During transpiration, there is fall in the temperature approximately by 10 to 15 degree. This is the line from NCERT. कुछ सोचना ही नहीं था. So both the statements are true. Hence, the answer of this question will become B. B for ball. Yes or no? Understood everyone? Chalo. Next question. Very easy, beta. Question is asking you when the Earth Summit happened. Now you know there were two summits happened. One was Earth Summit, and second was beta World Summit. Earth Summit beta happened in the year 1992, whereas World Summit happened in the year 2002. Now, if question is related to Earth Summit that happened in Rio de Janeiro, right? Whereas World Summit held at Johannesburg, South Africa, right? But here question is with respect to Earth Summit, hence your answer will become B. Correct? Yes. Agla sawal. Which micronutrient is required for the splitting of water during photosynthesis? Very easy question, beta. So tell me which microelement is required for the splitting. You remember this question I did in my marathon. The second question that we discussed in the marathon class, right? So remember, ma'am told you that there is oxygen evolving complex that helps in the splitting of water, is made up of three elements: manganese, chlorine and calcium. So according to the option, answer will become B for ball. Very good, baby bear. Next question. Now here there's two statements given, assertion and reason. Hi Firdaus, how was your paper? I remember Firdaus was there in my marathon class as well. So very good beta, very good Firdaus. How was your paper? Now next question on the screen is, the assertion is given beta that ATP is utilized in two steps of glycolysis. ठीक है? Okay, yes, ATP rather in, there are three steps. Yes, true, ATP is utilized in two steps. Correct? Reason. First, ATP is utilized in converting glucose into glucose 6-phosphate, and second, fructose 6-phosphate is when converted into fructose 1,6 biphosphate. Yes or no? So both the statements are true, but कहीं से भी इनका कोई ऐसा लग रहा है कि भाई रीजन एक करेक्ट एक्सप्लेनेशन है असर्शन का, नहीं, ये चीज नहीं फील हो रही. रादर yes, you can say that both the statements are absolutely true. Very good. So the answer will become C. Both are true, but as such, reason is not the correct explanation of assertion. Next question. Large, colorful, fragrant flowers, they are used for the pollination of which kind of pollinating agents are requiring these many features? Of course, insect pollination, beta. Insect, they need large, brightly colored, conspicuous, full of nectar, fragrant, rich flower for their pollination. So simply answer will become B, right? Okay, beta. Next question. The next question says, again, it's assertion reason. See students, sometimes when assertion reason questions they come, they can be little bit tricky. It becomes difficult for the student to analyze that whether the reason is the correct explanation of the assertion or not. But here, this time, in today's NEET examination, I can 100% bet that none of the questions were at all tricky. All the assertion reasons, they were absolutely clear. And yes, I know that this time, many of you are going to score 180 on 180. Yes? Okay, very good. Thank you, Tabish. Thank you so much. Now, moving ahead with the next question, let's read what the assertion has to say. Assertion says that the first stage of gametophyte in the life cycle of moss is protonema. Bilkul sahi hai bhai. Yes or no? The first stage of gametophyte in the life cycle of protonema of mosses is protonema, which develops from the spore directly. Yahi second statement hai. Yes or no? So can't you say that both the statements are true? And yes, reason is the correct explanation of assertion. Yes, see, read the first statement. You yourself will understand. The first statement And the second statement is the continuation of the first statement, explaining the first statement itself, that the first stage in the lifespan of mosses is protonema that develops from the spore directly. So over here, answer will become B. Next question. Very good, Firdaus. Next. In angiosperms, haploid, diploid and triploid structures you have to find out, beta. See, synergids are haploid, antipodal haploids, polar nuclei haploid. So this cannot be the answer. Next, synergids haploid, primary endosperm, triploid, zygote diploid. Again, this cannot be the answer. Look into the sequence and then try to answer. You cannot change the sequence by yourself. Third, antipodals, haploid, synergid haploid, प्राइमरी एंडोस्पर्म, ट्रिप्लॉइड. फिर ही हमारा सेट नहीं हुआ. ओके बेटा, आई गॉट इट. नेक्स्ट, synergids haploid, zygote diploid, endosperm triploid. Yes, very good baby bear and Firdaus. The answer will become D in that case. Clear? चलो अगला सवाल. Expressed ESTs, they are, beta, you know, in Human Genome Project, there were two techniques. There were two group of scientists who thought that which technique should we use to decode the entire human genome. So the two techniques were, number one, ESTs, number two, sequence annotation. ESTs was specifically responsible for decoding that portion of human genome which is responsible for producing protein. Whereas sequence annotation believed that we should use such a technique which is capable of decoding the entire genome including both exon and intron. But EST was simply responsible for decoding that portion of genome which is responsible for producing protein. That means if ESTs, they are responsible for producing proteins in exons only. So answer will become all genes that are expressed as RNA. Correct? Next, given below are the two statements. You have to tell which is right and which is not. So this D cannot be the answer. Arthi, Dikshant, beta, D cannot be the answer over here because all genes, whether expressed or unexpressed, this is used for sequence annotation. This is for sequence annotation, not for EST. Correct? चलो आगे चलते हैं, अगला सवाल. Now if you read the first statement over here, beta, endark and exarch are the two types of primary xylem. They are the distinctions of primary xylem but not of secondary xylem. I'm sure many of the students must have not read this question properly and they must have marked this question wrong, what I feel personally, because many of the students must have ignored that secondary and primary word. But remember, protoxylem and metaxylem are the two types of primary xylem, not secondary xylem. So first statement is wrong, but if you read the second statement which is missing, no, it's not missing, it's over here. Statement 2, exarch condition is seen in case of roots, true. Remember beta, exarch is for root, right? And endark is for stem. याद रखना इस चीज को, right? Okay, Dikshant. So the answer will become statement 1 is incorrect, but statement 2 is true. Dikshant, which one you want to understand again? Ma'am, why दोबारा समझाना? Which why? I did not, ma'am. Why? मेरा wrong हो गया. दीक्षांत, कौन सा question कह रहे हो बेटा? ये तुम्हारा wrong हो गया. मुझे लग ही रहा था बहुत सारे बच्चे ये गलत करेंगे because they will ignore the word secondary over there. And secondary was the only catch. Instead of secondary, there should have been primary. Next question. What is the function of tassels? Baba Ray, this is a direct question from your NCERT. You remember where we were studying about wind pollination, anemophily. This is one of the feature of plants getting pollinated by wind. Plants pollinating via wind. They have to capture the pollen grain. So their inflorescence is quite exposed in the form of tassels. So tassels are responsible for trapping the pollen grain from the wind. है ना? Easy सवाल है ना? बिल्कुल सीधा NCERT से पूछा गया है, कोई confusion नहीं होगा. So answer over here becomes C. Next question. The process of appearance of recombination nodules occurs at which stage of prophase 1? Absolutely, again very easy question, very easy beta. During pectine stage of prophase 1 of meiosis 1, recombination nodules appear on the synaptonemal complex for crossing over. Right? सही बताया कि नहीं बताया? Yes. So over here answer will become B for ball. Next question. The phenomenon of pleiotropism. Now if you remember beta, Mendel's law of dominance, then Mendel believed that one gene will only control one character. However, when more studies were done, then we came to know that actually this statement is not true. One gene can control more than one character also, and this is pleiotropism. Very good Dikshant, very good everyone. So answer will become D, a single gene affecting multiple phenotype expression. Next question. Identify the correct statements over here. Now these statements are written with respect to the ecosystem function decomposition, in which there are 5 steps. Fragmentation, that is done with the help of detritivores, like for example earthworm. So this is true. Second, the humus is further degraded by some microbes during mineralization. Of course, during whole catabolism, humus is getting degraded till the time it does not produce minerals. So true. Third, water soluble inorganic nutrients, they percolate down and they reach to the deeper layer of the soil, and this is known as leaching. The second step of decomposition, again absolutely correct. So A, B and C, all the 3 statements are right. Fourth, detritus beta food chain. Yes, very good Firdaus. This question was also done in your class. See, I told you now that whatever this marathon is about, you will get so many questions of this marathon in your NEET examination, and see, that only happened. So many questions that ma'am already discussed in her marathon class, they were absolutely there in your NEET examination even without changing the options. Option भी नहीं change हुए थे और सारा का सारा भाई उसमें पूछा गया था. तो देखो कितना फायदा हुआ होगा उन बच्चों को, those who have attended the session, right? So now, fourth statement is wrong. Fifth, earthworm break down detritus into smaller particle by the process known as fragmentation. So D and E are wrong, hence answer will become B. अगला सवाल भाई. Cellulose does not form blue color with iodine because, now again this question is from zoology, but ma'am will tell you beta what happens. Cellulose does not have a helical structure. When you study the structure of sugar, cellulose, then cellulose, due to the lack of helical structure beta, it cannot hold iodine, and because it cannot hold the iodine molecule, hence it does not give iodine test. So answer of this question will become 4. Very good everyone. Abhinav, very good. अगला सवाल देखो भाई. अगला सवाल क्या कह रहा है? The next question says, again very easy. Dikshant, I was telling you that in cellulose, since cellulose is not having a helical structure, because of which it does not have the capacity to hold the iodine molecules. As a result, it will not give your iodine test, and that is why the answer of the question over there will become 4. अगला सवाल यहाँ पे पूछा है R stands for what? So beta, this R stands for respiratory loss. बेटा RQ क्या होता है? RQ is the volume of CO2 evolved upon volume of oxygen consumed. So this is used in respiration, but here ma'am ये तो गलत है. No issues Dikshant. Next question will become, the answer of this question will become 4. Respiratory loss. Next question, how many ATP and NADPH2 are required for the synthesis of one molecule of glucose? Very easy. Very easy. Very good, Firdaus. See over here. If I talk about one glucose synthesis, then this cycle is going to run 6 times. And if the cycle is going to run 6 times, then 18 ATP and 12 NADH2 will be required. Hence, the answer will become 3. C. Very good, everyone. Very good. Next question. In gene gun method, again this question is related to zoology, but still ma'am will tell the answer. In gene gun method, it is one of the technique of recombinant technology, beta. When you introduce DNA into host cell, तो कौन सा metal use करते हो? Of course, it is gold or tungsten because they are very thin, and through them you can you can penetrate the microparticles very easily through them, as they are highly malleable and ductile, and they can be, they can be beaten into a very thin sheets. So answer of the question will become D. Right? Next question. The next question says, beta, see, this question ma'am discussed again. ये फिर सवाल आपके मैराथन से ही पूछा गया है. You remember evil quadrant? There are 4 reasons for the loss of biodiversity. Number 1 was habitat loss and fragmentation. Number 2 was overexploitation. Number 3 was alien species invasion. And number 4 was co-extinction. But the first reason is habitat loss and fragmentation. This is first, this is second, this is third, and this is fourth. But according to the question, answer becomes B, not D, beta. Dikshant, did you not attend my marathon last day? Ma'am discussed this question in the marathon, bacha. Next question. Give the reasons below 2 statements. Okay, given below the 2 statements, you have to tell assertion and reason again. First, late wood has fewer xylem element with narrow vessels. True. Why? क्यों ऐसा होता है भाई? लेट वुड के पास क्यों कम जाइलरी एलिमेंट्स होते हैं? क्योंकि लेट वुड का मतलब हो गया ऑटम वुड. ऑटम वुड के टाइम पे जो टेम्परेचर होता है वो फेवरेबल नहीं होता फॉर द फंक्शन ऑफ कैम्बियम. सो यस, assertion, reason, both are true, and reason is the correct explanation of assertion. So here answer will become B for ball. Right? Next question. Unequivocal proof. Direct NCERT. Nothing you have to do over here, beta. Direct NCERT based question. See, the unequivocal proof that DNA is the main genetic material. It was proposed by Hershey and Chase. Right? Yes or no? Simple question. Nothing you have to do. Now, moving towards the next question. The next question says, beta, frequency of recombination between gene pairs on same chromosome as a measure of the distance between genes to map and their position on chromosome. ये किसने बताया था? This was done by the student of Morgan, that is Alfred Stewart went. So over here, answer will become 4. Next, the reaction center PS2. Now there are two photosystems, beta. PS1 and PS2. PS1 is responsible for trapping light of higher wavelength, hence it is P700. PS2 is responsible for trapping light of lower wavelength, hence it is P680. So here answer will become B for ball. PS2 680. Most of the questions are from marathon. Absolutely true, Abhinav. Very good. Next question. Family Fabaceae differ from Solanaceae and Liliaceae. Yes, true. On what grounds with respect to anther? So if you know, beta, in Fabaceae, anthers are diadelphous and dithecous. No epipetalis, no epiphyllous conditions. So here answer will become B for ball. Correct? Agla sawal. In tissue culture experiment, now see this question is slightly tricky. The question says कि अगर हम tissue culture में एक mesophil, oh I can see Vikas is back. Okay, welcome to the class Vikas. How was your paper? How can I forget you? See beta, mesophil cell, it is already a differentiated cell. Now question is saying if you want to use this differentiated cell for tissue culture and from this cell if you want to obtain callus, then how can you do this thing? कैसे करोगे भाई ये? तो क्या करोगे? Mesophil cell beta will undergo dedifferentiation. It will again regain the capacity to divide. It will regain the capacity to divide and then it will become meristem and then it will help in tissue culture. So the answer of the question will become C. Why? What happened, Vikas? Why are you not studying well? So if you don't study, गड़बड़ तो होगा भाई मामला, है ना? अगला सवाल. Which hormone promotes internode petiole elongation and deep water rise? Direct NCERT based question है. Answer will become D, ethylene. Correct? Next. The next question says, during the purification process of recombinant DNA technology, okay, this question is, for ma'am, because he starts doing, okay, give some motivation who don't do their exam well, sir, can you, see beta, don't worry, if you, see, this is not the end of the life. First of all, let me tell you, this is absolutely not the end of the life. This is just the beginning. अगर अच्छा नहीं हुआ है तो कोई टेंशन की बात नहीं है भाई. There's lot to do more in your life even after this. And if you can give one more attempt, see, just calculate your marks. If you're scoring some standard marks like 400, 500, then of course you can give yourself one more chance, right? Because sometimes it happens, maybe it's not a good day for you and maybe due to some reason you could not do well, and yeah, you can give yourself one more chance in that case. However, if you want to change your field and if you think that okay, that's it for myself, I want to change the field, I want to go in some different field and do something good, that also you can do, because this is not the end of the life. You have lot to do and lot to explore. So keep exploring, don't worry. Whatever the paper, जो भी हुआ है पेपर में, उसकी बिल्कुल चिंता मत करो. बस आगे का सोचो, जो हो गया उसकी चिंता मत करो. Don't worry about the things that has been done. Now you can't change your past. But yes, what you can do is make your future. So explore more. So just explore more of fields, and if you really want to change the field, then too you can do. But if you really want to give yourself one more chance, then too you can do. But stay motivated, no need to lose hope. This is not the end of the life, this is just the beginning for you. Firdaus. Okay, chalo, this is the zoology question. In zoology class you will discuss it when the zoology paper will be done. Moving towards the next botany question. Identify the pair of heterosporous pteridophytes among the following. So which are the heterosporous? Selaginella and Salvinia. Equisetum over here is homosporous. Lycopodium homosporous. Xylotum homosporous. So answer will become C for cat. Next question. Axial placentation is observed in, the simple answer is, thank you Vicky, thank you so much, is A. Why? Because in mustard you see parietal placentation. Wrong. Beans, you see marginal placentation. Lupin, marginal placentation. Wrong. Then in this P, again marginal placentation. Wrong. So the answer of the question will become 1 over here. अगला सवाल फिर से भाई assertion reason का है. Assertion says a flower is defined as modified shoot wherein the shoot apical meristem changes to floral meristem. True? What is flower? You know, when floragen hormone is synthesized in the leaves, then it travels from the leaf to the shoot apical tip. It transforms the shoot apical tip into floral tip and then flower is born. So in short, you can say that flower is a modified shoot. So first statement is true. Second, internode of the shoot gets condensed to produce. Now ये तो उसका explanation ही हो गया भाई कि कैसे काम हो रहा है. तो जब भी बेटा फ्लावर को बनना होता है, shoot modifies by internode getting condensed. This is the line from NCERT, and yes, over here both the statements are true and reason is the correct explanation of assertion. So answer over here will become B. Next question. Vikas, stop spamming. Next. How many different types of proteins does the ribosome consist of? This is directly NCERT based line. In ribosomes, we have 80 different types of protein. So over here, answer will become B for ball. Agla sawal. Malonate is the inhibitor of which enzyme? So let me tell you. Malonate is a chemical which inhibits the activity of succinate dehydrogenase, which is used in Krebs cycle as well as ETS. But you add malonate, this enzyme will not work. So answer will become B. Okay. Thank you so much, Vikas. Now I liked it. Now I like that your message. Thank you so much. Shahid, okay, love physics wala from Kashmir and lots of love to you as well. Now moving towards the next question. You just have to do the matching over here. Oxidative decarboxylation, you see in case of link reaction, enzyme pyruvate dehydrogenase. Next, glycolysis, it is also known as EMP pathway, Embedin-Meyerhof-Parnas. Correct? Oxidative phosphorylation. Oxidative phosphorylation is basically ETS, responsible for generating ATP. Tricarboxylic acid cycle is also known as citric acid cycle, where citrate synthase enzyme is used. Now see, A with 2, B with 4, C with 3. So here answer goes as 1. ठीक है? अगला सवाल. Again you have two statements and you have to tell which is true, which is not. So if you read the first statement, bacha, then Gauss experiment was simply, ma'am, Yakin English 2024. Soon it will be launched, Shubham. Don't worry. All the batches will come. Now it's the time for every new batch to restart. Surely you'll get the information about Yakin English as well. So don't worry. Stay positive. Now coming back to the statement number 1. Gauss competitive exclusive principle states that two closely related species competing for the same related species cannot coexist. Yes, absolutely true. First statement is true, beta. Why? Because according to Gauss experiment, two closely related species competing for the same resource cannot live together. They will suffer competition and the superior species will remain, whereas the inferior species will get eliminated, right? So first statement is true, but second is wrong. Why? Because herbivores are more adversely affected. As compared to carnivore, beta, herbivore are more affected, and the reason associated with it is what? The reason associated with it is as herbivore they only have one option to feed ना. herbivore से पेड़ पौधे खा सकते हैं, carnivore के पास इतने सारे options होते हैं to feed on. Correct? So answer will become C. Very good, Firdaus. Next question. Which of the following combination is required for chemiosmosis? Very simple. Ma'am has taught in the class. So answer will become B. Membrane, which is thylakoidal membrane, proton pump, gradient and ATP synthetase enzyme. All these four are the requirement. Next, given below are the two statements. One is labeled as assertion and reason again. If you see over here, beta, the first assertion statement says in Gymnosperms, pollen grains are released from the microsporangium. Yes. And by wind current, they are dispersed. True. Now, once they get dispersed from the wind current, beta, then the micropyle of the megasporangium take it, accept kar leta hai. Then there is the formation of pollen tube, and then that pollen tube carries the male gamete inside the archegonia for fertilization. But here it is written no pollen tube. So that is why this statement becomes wrong. Hence, answer will become D. Agla sawal. Which of the following answer is not correct? If you read all the four statements, beta, and you focus on the C statement, then in C statement, what is written? Algal blooms caused by excess of organic matter in what does not improve, beta, rather it degrade the quality of water. Correct? So over here, answer will become C. Agla sawal. You have two. Which of the following statements are correct about Klinefelter syndrome, which is actually allosomal trisomy? It is basically allosomal trisomy, 2n plus 1, and here one extra X chromosome develops apart from pair of autosome. This is the situation, and in this case it was not discovered by Langdon Down. Down discovered, very good Firdaus, by Down syndrome, wrong. Then affected individual becomes tall rather than short. This is also wrong. And E statement is true. Physical, psychomental, no mental retardation. And B is also true. So B and E. So answer will become D in this case. True? Yes or no, everyone? Chalo. Next question. Identify the correct statement, beta. If you see over here, in this question, lenticels are the lens-shaped openings permitting the exchange of gases. True. Bark formed early in the season is called soft bark, not hard bark. तो ये गलत हो गई statement. तीसरी, beta, bark is a non-technical but not technical. Here it should be a non-technical word. So that is why this statement also become wrong. But the fourth statement is true. So first and fourth, A and D. Hence answer will become C. Clear? Next, now you have to simply match the following. Very easy. This question we discussed tomorrow. Sorry, this question we did in our marathon. Yes? So M phase is the interval. G2 phase, protein synthesis. Quiescent phase, inactive phase. G1 is equational cell division. So A with 3, B with 1. Sorry, A with 3, B with 1, C with 2 and D with 4. But here I can't see any of such options. I think some misprinting is there. You just check it like this. There is some misprinting in the options. This is just the printing error. You see, M phase is the interval between, sorry, I did some mistake. Wait. So basically, beta, M phase is equational division, G2 phase protein synthesis, quiescent inactive, G1 interval. Now, this is how you have to match it, beta. and now the answer will become A4, B1, C2, answer becomes D. Clear? थोड़ा सा भाई starting में वो हो गया. अगला सवाल, match the list and select the correct options. So mutualism is such an interaction where both the species are equally benefited. So it's plus plus. A with 4. Commensalism, it's kind of a plus 0. One species benefited, another is neutral. Amensalism, minus 0. Parasitism, plus minus. So A with 4, B with 1, C with 2. So answer becomes C for cat. Next question. Now match the list. Again, this question also we did in our marathon. ये फिर मैराथन का सवाल है. चेक कर लेना मैराथन को कि मैम सही कह रही है कि नहीं कह रही है. ये सवाल मैराथन में था. Cohesion is attraction among water molecules. A with 2. Adhesion is attraction towards polar surfaces. Surface tension helps in maintaining the water in droplet form. Guttation, loss of water in the form of liquid phase. So here answer will become B. Correct? Next question. Main steps in the formation of recombinant DNA are given. So the answer of this question will become B. Sorry, it will become D. Why? Because the first step is isolation. Then second is cutting with the help of restriction enzymes. Then third is C, B. Then third is amplification using PCR. And then fourth is inserting. So here answer will become C, B, D, A. Last question. The last question says you have to again match the following beta. Iron is the activator of catalase. Zinc activates oxygen. Boron helps in cell elongation and differentiation, and molybdenum helps in component of nitrate reductase. So A with 3, B with 1, C with 4 and D with 2. So I hope you all have understood all the 50 questions of botany specifically, and thank you for watching all these lectures, and I hope it continues with the zoology as well. So thank you everyone guys, thank you so much. Wait for the zoology to resume.
Hello, my dear lovely students. Hello, guys. How are you? Hope everyone is doing good. Hope your exam went well. Now, I am here to discuss some questions from your zoology. Everyone ready? Tell me in the comment section, am I audible? Am I visible? Is everything fine? Yes, Josh. How's the Josh? Josh is also high. Everyone. So if I look at your question, a moment back, I was going through your question paper. Questions, they were very easily. In fact, 100%, that means all questions, they were directly from the NCERT. Are you guys ready? Can we start this session? Yes, guys, we are going to discuss today, we are going to discuss zoology. And those students who who have seen those marathon series where I have discussed the zoology questions, most of the questions, they were directly from those topics. Do you agree to those points? Yes, zoology, guys, zoology. So let's not waste any time. Let's start this session, and do tell me what is your score in the comment section so that we will definitely will check. Clear? Okay, buddies, let's start. Yes. So keep sharing your answers also. Now the first question, the very important and the very easy question, guys. The first question is, two lists they were given. Very easy, very easy. Now the first is the peptic cells. Guys, these are the peptic cells. So peptic cells, you know, they are responsible for secreting the proenzyme, which is a pepsinogen. Goblet cells, they secrete mucus. Oxyntic cells, they are responsible for the secretion of the HCl as well as the intrinsic factor, the Castle intrinsic factor, which is important for the absorption of vitamin B12. Now we have two, and the hepatic cell, you know, they help in the secretion of the bile juice. Isn't it clear? Like, it was a very easy question. Now, first option, peptic cell, option 3 is correct. Yes. So we are going with the option D in this question. Right, everyone? So option D was absolutely 100% correct. Now let's move on to the next question. Which one of the following common STD is completely curable when detected early and treated properly? So they have given a some list of the diseases. For example, HIV infection, genital herpes, gonorrhea, and hepatitis B. 3 diseases, guys, they have discussed about it in like NCERT talks about 3 diseases which are non-curable. In the non-curable, we have HIV, we have genital herpes, and we have is a hepatitis B. But question is a bit opposite. That means they are asking which is curable. Correct answer we have is the gonorrhea. That means causative agent. What's the causative agent of gonorrhea? I know it's not a revision session, but it's okay. It is Neisseria gonorrhoeae. So correct answer is option C. Did you mark it correct? Option C. Yes, buddies. Yes, buddies. Come on, come on, come on. Okay. Yes. Now, let's move on to the next question. Select the correct group. Now, we have to select the correct group of Australian marsupial. Clear? Australian marsupial, guys, you have to select. What did you write? What was your selection in this question? So, 2 groups they are given as far as the adaptive radiations they are considered. The parallel evolution they show whenever we talk about the marsupial and your placental mammals. But we have to only select the Australian marsupial. So in this case, direct question from your NCERT. What is that? The correct answer we have is a numbat, spotted cascus, and the flying phalanger. All these are what? These are your Australian marsupial. Guys, right? No, student, in this case, the correct answer we have is this. Here, when we talk about the mole, the flying squirrel, both of this, they are what? They are the placental mammals. So, this is not the correct answer. So, this is the incorrect answer. Clear? Now, let's move on to the next question. I won't take much time. We are revising everything. We are going through the question in a very fast way. And just tell me what is your score. Paper was quite easy. If I look at the paper was very easy. And if you have gone through the concept, you are, in fact, in zoology, you will be able to attempt all the questions. 100% the questions, they were directly from NCERT, not from any XYZ group. Any XYZ, in fact, I'm all good, Rahul. So, like questions, they were directly from NCERT, your 11th and the 12th class NCERT. Not even a single question was from outside. So, I was so happy to look at the question paper. Okay, now, next is match the list and list 1 and list 2. This question we have already discussed in that marathon session, where the P wave, QRS complex and the T wave was given. And guys, if you have gone through the paper, if you have seen, like if you have matched a single thing, you will definitely get an answer. Definitely. Now, first one is given as a P wave. You know the P wave is responsible for the depolarization of the atria. So, for the P wave, for the A, correct answer we have is a 3. So, this gives us an idea, like this is the correct answer. Right? And I'm 100% sure many of you must have guessed it right. Now, Q wave, the second one, you all know the Q wave is the one which is responsible for the beginning of the systole. That means a contraction is about to happen, ventricle contraction. The QRS complex, this is responsible for the depolarization of the ventricle. Yes, for the T wave, we have discussed about it, that it is responsible for the repolarization of the ventricle. And in fact, at the time of discussion, I told you ECG is one of the topic where one question will surely come. Yes, definitely. Now, let's move on to the next question. We have discussed about all these diseases. If you have gone through the session, this was a question in your marathon session also. The first one is a ringworm. Yes. The name indicates it's a worm, but this is because of the fungal disease. You know, ring guard. You must have seen that advertisement in the TV, ring guard. So this is basically because of some, you can say fungus. The correct answer we have is a trichophyton, epidermophyton, microsporum. 3 example NCERT mentioned about. Now for the A, the correct option we have is a 2. So that means these 2 options, they are correct. Second one we have is a filariasis. This is the causative agent is Wucheria bancrofti. Second name is Wucheria malleae. And malaria, you know, this is caused by Plasmodium vivax, Plasmodium falciparum. Next one we have is a pneumonia. Causative agent we have is Haemophilus influenzae or Streptococcus pneumoniae. 2 example NCERT talks about. Clear? So the first 2 for the B, correct option we have is a 3, and both of this is correct. For the C, correct option we have is a 4. So we are going with the option B in this case. So what is the correct answer? Option B. Clear? If you are 2023 NEET aspirant, that means you must be going through your score. And if you are NEET 2024 aspirants, you can see, you can in fact solve many of the questions from the 11th standard because you have already completed your 11th standard. Questions, they were directly from NCERT. Those who are like, definitely, who are like that we'll go through this topic in depth, or we should talk about more about this topic. See, nothing comes outside NCERT. Yes, and in fact, in the zoology part, many questions, they were from botany. Now, let's move on to the next question. Given below are the 2 statements. So, the 2 statements they were given. Now, we have to select the correct answer. Now, statement 1, vas deferens receive a duct from the seminal vesicle and opens into urethra is ejaculatory duct. Is this statement correct? Yes, guys, this is 100% correct. Yes, 100% correct. No doubt in this topic. Now, next one is a statement number 2. Statement of number 2, the cavity of the cervix is called cervical canal. So, cervix form a cavity, that is cervical canal, right? And along with the vagina, it forms a birth canal. This is given in the NCERT only, correct? This is also a correct option. This is also correct. This is also correct. Both of them, they are correct. So, we are going for the option B in this. So again, I'm saying I was so happy looking at the paper that the questions, they were directly from your NCERT only, not from any other book, directly from your NCERT. They were very easy. See, this statement is directly given in NCERT. Now, let's move on to the next question. Given below are the 2 statements. Again, we have 2 statements. The first statement is RNA mutates at faster rate. We all know the RNA mutates at a faster rate. This is absolutely correct, 100% correct. Now, statement 2, viruses, they have a RNA genome. We all are acquainted with the viruses now. So, viruses, they have a RNA genome. We are well acquainted with the virus because of the SARS-CoV-2 virus. And they have a RNA genome. They have a shorter life, span, mutate, evolve faster. Yes, they have a RNA as a genome and they mutate at a very fast rate. And that is the reason every now and then we are getting different strain. Here, yes, AFMC 2023 aspirant, tell me, how was your paper? It went well. Now, both of them, they are absolutely right. So, option B is the right over here. What was? Yes, absolutely right, guys. Absolutely right. Now, let's move on to the next question. Radial symmetry. Radial symmetry is not found. So, that means we are talking about the false statement. They are not found. The first one is given as a echinodermata. In the adult echinodermata, we have radial symmetry. Whereas in the larval form, we have is the bilateral symmetry. So this cannot be a right answer because they have radial symmetry. Ctenophora, they also have a radial symmetry. So this cannot be a right answer. Hemichordata, they have a bilateral symmetry. So Coelenterata is also not the correct answer. Yes, isn't it easy? Very easy. In fact, questions, they were very, very, very easy. Now, let's move on to the next question. Given below are the statement. One is labeled as assertion and other is labeled as a reason. Clear? Your favorite assertion, reason type of question. Assertion A, nephrons are of 2 types. Yes, we all know nephrons, they are of 2 types. The cortical nephron, guys, we have cortical nephrons and we have juxtamedullary based on their relative position in cortex and medulla. right? So where actually they are located, that means which duct is present more towards the medulla region or which is present more towards the cortex region. On that basis, we have two type of nephron, the cortical nephron and the juxtamedullary nephron. Now next one, reason. So this is 100% right. This is 100% right. Clear? Already given in NCERT? Absolutely right. Now let's look at the reason. Reason. Juxtamedullary nephron, they have a short loop of Henle. Is that so? Does juxtamedullary nephron has short loop of Henle? Short? No. It has long loop of Henle, whereas cortical, they have short loop of Henle. Short loop of Henle. Here, so that means this particular statement, this statement is incorrect. So assertion is right, reason is wrong. Assertion is right and reason is wrong. So let's look at the correct option. Yes. So we are going for the option A. A is true and the reason is false. Clear? Yes, buddies, is it clear? Now, moving on to the next question. Vital capacity of the lung. Vital capacity. That means this is that capacity which is responsible for keeping us alive. Vital capacity. And I told you there is a volume of air which we inhale or which we exhale. What do we call it? We call it tidal volume. And suppose we inhale at a forceful rate. So that is the inspiratory reserve volume and the expiratory reserve volume. So if we are going to add all of this, the inspiratory reserve volume, the expiratory reserve volume and the tidal volume, what do we get? Is the vital capacity. And what is left behind is your residual capacity. And residual capacity is the one which we cannot measure. With the help of spirometer, we cannot measure the residual capacity. So correct answer we have is IRV, ERV and the tidal volume. So IRV is inspiratory reserve volume, expiratory reserve volume, and this is the tidal volume. Here, tidal volume is a normal respiration that we do. Some amount of gases we exhale, some we inhale. That is the tidal volume. But when a force occurs, so that means expiratory reserve volume and inspiratory reserve volume. Here, so everything comes under your vital capacity. Now, moving on to the next. Otherwise, what will happen? The lungs will burst, and that we don't want. Now, which of the following is not considered as a part of endomembrane system? So in endomembrane system, correct answer we have is endoplasmic reticulum. We have is Golgi, but chloroplast is not there, peroxisome is not there, and this is not there. So ACE, option C is correct. Option C is correct. Yes, yes, buddies. Now let's move on to the next question. List 1, we have to match list 1 and the list 2. Again, guys, list 1 and the list 2. Tell me, what is the correct answer? Here they are talking about your lac operon. Guys, they are talking about the lac operon. Right? In the lac operon, when we talk about the structural gene, 3 structural genes are present. Lac Z, lac Y, and lac A. Right? Clear? Clear, clear, clear. So when we talk about the beta galactosidase, the correct answer we have is lac Z, right? Lac Z. So for the D, the correct option we have is a 1. So option C, C looks correct. But see, I told you that like even if you know a single option, you can easily go through these list 1 and list 2 type of questions. Now, when we talk about, guys, let's talk about this gene A. We all know the gene A is responsible for the formation of transacetylase, Y for the permease, I is for the repressor protein. Yes, it forms a repressor protein. This is the correct answer, option C. Now, again, match list 1 and the list 2. Right? 2 lists are given, list 1 and the list 2. Clear? Now, heroin. We all know heroin. Heroin is responsible for the slowing down the body function. It slows down the body function, right? It suppresses the body activity, heroin. Now, next is a marijuana. It affects the cardiovascular system. Guys, these sentences, they are from nowhere. They are directly from your 12th class NCERT chapter, Human Health and Diseases, Drugs and Alcohol Abuse. Directly from that topic. Now, when we talk about the cocaine, what is that cocaine? Cocaine interferes with the transport dopamine. Clear? Yes, Gulshan? Yes, I'll tell you. Morphine, you all know during the war, earlier time during the war, people, they like those soldiers, they are given morphine just to suppress the effect of pain. They act as a painkiller. What's the correct answer? Option B. Yes, Gulshan, option B is absolutely correct, student here. Yeah. Now, next one, match list 1 and the 2. And guys, this we have discussed in the session also. In that session, we have discussed about the questions, the detailed question, the marathon session, where we had that thing we have talked about the joints. Yeah. So first one is a type of joint. So on the one side, a type of joint is given, and in the second side, where they are actually found is given. So cartilaginous joint, where are the cartilaginous joint found? Cartilaginous joints are those joints which are found between the adjacent vertebrae. We have cartilaginous joint. Ball and socket. So when we talk about the humerus in the pectoral girdle, we have is this. Next is a fibrous joint between the skull bone, saddle joint between the carpal and the metacarpal of the thumb. Yeah. For the 1, correct option we have is a 2. So these 2 options C correct. For the B, we have is a 4. Absolutely right. For the C, correct option we have is option 1. So your Chavi ma'am's option, your C option is absolutely clear. Let's have a look once again. Yes, a D option. Yes, option C correct. Now, again, match list 1 and 2. So 2 lists are given. List 1. Here they talked about the certain hormones. Here the list 1, that means the organ on which they are acting or the organ from which they are secreting. Now CCK, you all know they act on the pancreas so that they can secrete the pancreatic, you can say secretions. PIP, gastric glands. Atrial natriuretic factor, this is secreted by heart, right? ADH, antidiuretic hormone, that is for the kidney, right? So for the CCK, correct option we have is a 4. For the B, correct option we have is 3. Yes. For the B, correct option we have is a 3. For the C, we have is the option 2, ANF, atrial natriuretic factor. And when they are secreted in response to high blood pressure, so that we can reduce the blood pressure. ADH is responsible for more and more water reabsorption from the DCT and the loop of Henle and collecting the. Which of the following is a wrong statement is a correct statement, you have to find out. Algal bloom decreases the fish mortality. Is that so? Algal bloom increases the fish mortality. Eutrophication referred to increase in domestic sewage and water waste in the lake. No, eutrophication leads to the aging of the lake. Not correct. Magnification referred to increase in concentration of toxicant. Yes, absolutely right. So whenever the toxicant level is increasing in successive trophic level from one trophic level to another, this is termed as biomagnification. Slowly and slowly it is increasing. What do we call it? Biomagnification. Example, a very beautiful example is given is of DDT. Now, a large amount of nutrient waste. Now next one, match 1 and the 2. Guys. Yeah, match 1 and the 2. Again, some options are given. So it is, yes, we have studied this chapter in botany, but it is given in the zoology part, but it's okay. Question was, again, a super easy, and these examples, they were from your NCRT only, not from anywhere. Now, a leopard and the lion in the forest and the grassland. Leopard and the lion, what will be type of interaction? Definitely competition. No doubt in this. Competition. Next one. Cuckoo laying egg in the crow nest. This is a brood parasitism. Now, fungi, the root of the higher plant in mycorrhiza. Right? What do? They help in, they are the mutualism is happening. Mutualistic behavior. Both are being benefited. Now, next is a cattle ergot and the cattle in the field. So, one is affected. So, this is a kind of a positive and zero kind of interaction where one is affected. One is benefited, another is neither benefited nor harmed. You all know about it. So A, B, C. So correct option is given in front of it. So A, B, C, D. I guess option yes. Option B is correct. Yes, buddies. Option B is correct. Yeah. Yes. Yes. Option B. Now, in which blood corpuscle the HIV undergo replication and produce progeny viruses, right? So when I talk about when we talked about the HIV virus, I told you, yes. Listen, so Shurendra, I'll tell you the questions. They are same. Either we talk about this G1, G3 or G6, whatever you are saying. So, but the question, sometime it is like question, yes. So sometime it is like reshuffling is there in these. And if you'll go through the question paper, if you'll go through these questions, definitely you can easily calculate your marks. Clear? Here we are discussing, guys, G3. Now, correct answer we have is T helper cell. So what do they do is they decrease the level of, they decreases the number of the T helper cell. So the person immune system is affected, immunocompromised. Yes, T helper cell. Many times, guys, we have talked about it. So T helper cell, whose number is reduced? T helper cell number. Now, let's move on to the next question. Match list 1 and the 2 with respect to human eye. Yeah. Now, first list 1, list 2, fovea. Guys, we have discussed about it. Fovea. So, those who have they seen our AITS paper, All India Test Series, this question was directly from there. Now, fovea is a point of greater visual acuity or the resolution, right? Greatest visual acuity and resolution. Iris is a visible colored portion of the eye which regulate the diameter of the pupil. Absolutely right. Blind spot is a point where the optic nerve, right? Optic nerve leaves the eyeball, right? Photoreceptor cells, here the images are not formed. Photoreceptor cells, they are absent. Sclera is a layer, the external layer of the eye, which is formed by the dense connective tissue. Yes, Kulshan? Yes, Vaibhav? Very nice. Now, correct answer for the first correct answer we have is option 3. Yes. So correct. See, like if you know about, at least know about the fovea, you can give a correct answer. That is option B is correct. This is the cheat which I am discussing right now. And you all must, like, this is a basic strategy. Even they know about, even the student, they know about the first or the second option, first and second A and B. They can easily guess C and D option. No question, no answer was confused or overlapping. Now, moving on to the next is, given below are the two statements. Again, two statements they were given. The first is, in prokaryotes, the positively charged DNA. Positively charged DNA? No. So, DNA is always negatively charged. DNA is always negatively charged. Yes. DNA is held with some negatively charged protein. Incorrect. Now, statement 2, in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleus. This is absolutely right. Clear? So, statement 1 is wrong. If you look at the statement 1, that is wrong. Statement 2, which is given over here, it is starting from here. This is absolutely right. Clear? So, statement 1 is incorrect and statement 2 is true. If you are from any other, like if you are matching your question, guys, from G1 or the G2, please go through the options also. So option reshuffling is also seen. So it's not the case that for this question in this paper, the option 1 is correct. This option 1 over here is statement 1 incorrect, but statement 2 is true. This particular option can be option C or D in different papers. So please, guys, go through the question properly and go through the answers of options also properly. Moving on to the next, given below are the two statement. Guys, again, two statement question, one of the very favorite question. Now, ligaments are dense irregular connective tissue. Is it a dense irregular connective tissue? No, they are dense regular connective. Yeah. Now, cartilage, they are saying they are dense regular. No, they are specialized connective tissue. Both the options, they are wrong. Yes. Both statement 1 and 2 are false. Both the statements, statement 1 and 2, both are false. Yeah. Now, let's move on to the next question. Which one of the following symbol represent mating between relatives in human pedigree analysis? In human pedigree analysis, what is the case? Yeah. So, what was the correct answer? Let me look at the option. Just 1 second. I want to go through the correct option. 1 second. Is that so? I guess there is some problem in the typing part. Yes. Yes. So, yes, this is right. Option C is correct over here. So, because we are talking about the closely relative, the close relative. So, this is a correct option, right? But when we talk about a normal meeting, in the normal meeting, this is a correct one. The normal, because they are linked, because they are relative to each other. So, we use this symbol. Now, which one of the following technique does not serve the purpose of, this is directly given in NCERT, does not serve the purpose of early diagnosis of disease in early treatment? What's the right answer? That is a serum and the urine analysis. Serum and a urine analysis gives us an idea, it gives us information when disease is already there in the body and we go for it after we got symptom. So, after symptom only, we'll get a positive test in urine and the blood analysis. But before symptom, that means if pathogen is present in the body before any symptom, yes, recombinant DNA technology plays a very important role. With the help of ELISA, with the help of polymerase chain reaction, we can easily detect. So what is the right option? Option C. Option C. And what is that? Serum and the urine analysis. Moving on to the next is test 1 and the 2. Again, test 1 and the 2, two options they are given. The first one is given as tinea. So they are talking about the excretory structure. We all know tilia, they are famous for the protonephridia or the flame cell. That means for the A, the correct option we have is a 3C. Isn't it easy? Okay, let's go for the other option also. Paramecium, they are having contractile vacuole. Periplaneta americana, that means your favorite cockroach, they have uricose glands. For example, next we have is a ferritima. Characteristic feature is they have nephridia. Now for the B, let's go for other option also. B2, we have is a C4 and D1. Yeah. Now next one, again guys, two statements. One is labeled as assertion and other is labeled as reason. Now, assertion, amniocentesis for sex determination is one of the strategies for the reproductive and child healthcare program. Is that so? So, in the reproductive and the child healthcare program, yes, amniocentesis is there, but amniocentesis was not for the determination of sex. That was not for the sex determination. Basically, it was like that we should not use, we should not go for the sex determination with amniocentesis. This was banned in India. So this is incorrect. So that means assertion is wrong. Ban on amniocentesis, check increasing menace on female fetocyte. Yes, this is absolutely correct. So here the assertion is false and the reason is true. Right? So we went for option 1. Was it easy? Yes. In fact, guys, all options, all the questions, they were very easy. Very, very, very easy. Yes. So this was all about this session where we have discussed some questions from the zoology. Some more questions, yes, are also there. That will be discussed by another teacher. So all the very best. Do tell me what was your score? How much did you attain? Did our marathon session benefited you? If yes, do tell me in the comment section. Take care, guys. Irrespective you get, irrespective how many questions they are right or wrong. Just be focused, just be calm. It's okay, guys. Do not stress yourself. Take a chill pill, relax yourself, and enjoy this moment. Take care. Bye-bye, everyone.
Hello students, how are you all? I'm sure all of you are so amazing. After exam, feeling so relaxed. So how was your exam? So write down in the chat box how was your exam. So I hope you have done awesome. And today we are just going to discuss questions of zoology. After 175, Chavi ma'am has already discussed some questions with you, 25 questions with you. And after 175, I'm going to discuss other question with you. Code number is G3. So let me take you ahead. This is the question number 1. So question was so easy. So I am sure you have done it nicely, right? So there must be like rush of the emotions. Some of you must be happy. Some of you might be super excited. Some of you might not have done that much good. So you have to decide for yourself what you have to do, right? So let's discuss the question. This is the question 175 of the G3 paper, code G3. Given below are two statements. So you just tell me what will be the answer here. Statement 1 is electrostatic precipitator is most widely used in the electrostatic precipitator is most widely used in the thermal power plant. And statement 2 is electrostatic precipitator in thermal power plant removes ionizing radiation. So chalo, write down in the chat box what will be the answer. So let me see how many of you have done nicely. So write down in the chat box. So let me see your chats. So write down in the chat box. So what will be the answer here? Hello students, what will be the answer here? So I hope that you know the answer. Electrostatic precipitator is most widely used in thermal power plant, which is very correct. This answer is absolutely, this statement is absolutely true. But electrostatic precipitator in thermal power plant removes ionizing radiations? No. What does it remove? Particulate matter, right? So this option is absolutely incorrect here. So this statement is incorrect. Statement 1 is correct. So what will be the answer? Absolutely. Akib, yes, answer will be 1. Yes, Jia, answer will be 1. Statement 1 is incorrect, but statement 2 is correct here. So I hope this question is clear. This is the next question. Once the undigested and unabsorbed substances means fecal matter. So once the fecal matter enter the cecum, their backflow is prevented by, what will be the answer? So you know very well, pyloric sphincter will not be the answer because they are talking about the fecal matter. And fecal matter will be gone to the cecum, this region of large intestine. And obviously, this is the part of small intestine that is known as ileum. This is ileum and this is the cecum region of large intestine, right? So obviously, food will come here, fecal matter will come here, and we are having the sphincter here in between the ileum and the cecum. So this sphincter is known as ileocecal valve. So why we called it valve? Because it will prevent backflow of the fecal matter in the ileum of small intestine region, right? So ileum is the part of small intestine. So correct answer will be C here. Gastroesophageal sphincter is present between the stomach and the esophagus. Sphincter of Oddi is present there in the pancreatic region. Hepatopancreatic opening is basically guarded by sphincter of Oddi. And then we are having pyloric sphincter, which is present there in between the intestine and stomach, right? So correct answer will be this. So let's move toward the next question. This is the question number, this is the next question, match list 1 and list 2. So this question from the reproductive health, write down the answer. So I'm just looking at your chat. What will be the answer here? Yes, absolutely. So vesicectomy, that means vas deferens. Tomy means surgical removal. So we just cut the vas deferens and just tie it up. That is the part of the surgical method. Male sterilization. Coitus interruptus, you know, is the part of natural method, natural contraceptive method. Cervical caps are barrier method and Saheli is oral method. So what will be the answer here? A goes with the third, B goes with the fourth, C goes with second, and D goes with first. So correct answer will be C here, right? So absolutely correct. So let's move towards the next question. This is the next question from the biomolecule chapter. So you just tell me what will be the answer. Let's write down the answer here. So full Josh, with full Josh, it should be seen here, right? That you have done really amazing in your test. So low temperature, statement 1 is low temperature preserve the enzyme in a temporary inactive state, whereas high temperature destroy enzymatic activity because proteins are denatured by heat. And second statement, when the inhibitor closely resembles the substrate and its molecular structure and inhibit the activity of enzyme, it is known as competitive inhibitor. So when we see the first enzyme, we know, when we see the first statement, we know that low temperature, in the low temperature, enzyme is not destroyed. It has lost its property or its activity for some time. As it gains the normal temperature, it will regain the activity also, right? So yeah, this is basically given that they become temporarily inactive in low temperature. But at the high temperature, we know enzyme hydrogen bond in the enzyme structure is destroyed. So it undergo denatured process, means now it become permanently inactive. So no matter if the temperature brings to the normal temperature, this activity cannot be regained, right? So this is absolutely true. Statement 1 is correct. Statement 2 is inhibitor closely resemble the substrate, competitive inhibitor. So we know they are talking about the competitive inhibition where substrate and the inhibitor look same, and they compete for the active site of enzyme. So suppose this is the active site of enzyme. This is the active site of enzyme. Both this substrate and inhibitor fight for this active site of enzyme. And when inhibitor bind with it, then no product will be formed. This enzyme cannot form any product, right? So product will be formed only when substrate is attached with enzyme. So this is absolutely true. So which answer will be correct here? Both 1 and 2 statements are true. That means B will be the answer here. Let's move towards the next question. Which of the following is not a cloning vector? So what will be the answer here? Write down the answer in the chat box. It's very easy question of biotechnology. What will be the answer here? So answer will be probe, because probe is nothing but the radio-labeled nucleotides, right? Which we use radio-labeled nucleotides. And that is complementary to the DNA or RNA that we have to detect, right? So it is complementary sequence, or it forms the complementary bond with the single-stranded DNA or RNA, so that we can obviously get to know the sequence of the DNA. For that we use the probe. It is not a cloning vector. Rest all of them are the cloning vector. So PBR322 is also cloning vector. Yeast artificial chromosome is also cloning vector, and bacterial artificial chromosome is also the cloning vector, right? So these two are used in Human Genome Project, and this you must have done in your biotechnology chapter. So correct answer will be probe here. Let's move towards the next question. Which of the following function is carried out by the cytoskeleton in a cell? Direct NCERT question. So all the questions are from NCERT except one. So question is the transportation, nuclear division, protein synthesis, motility. So what will be the answer? Write down in the chat box. What will be the answer? So absolutely, my chat box is flooded with the answer. What will be the answer? Carried out by the cytoskeleton in a cell. What is the function of cytoskeleton? So obviously motility. It provides motility. So not transportation, not nuclear division. Protein synthesis will not be the answer. So cytoskeleton help in the motility, which is given in our NCERT, right? So this will be the answer. Next question, statement 1, a protein is imagined as a line. They are talking about the primary structure of protein. So we know the left end of the protein have the N-terminal, because amino group is basically free here. And here the last amino acid, or this right end basically indicating last amino acid, representing last amino acid. So they are having the COH in the end. So obviously we called it C-terminal. So a protein is imagined as a line, left end represented by first amino acid, true. And it is called C-terminal, no, it is called N-terminal. And the right end represented by last amino acid, very true. And they are having the C-terminal free, means COH group free. So we called it C-terminal, not N. So this is wrong statement. Adult hemoglobin consists of 4 subunits. So what are those 4 subunits? 2 alpha and 2 beta. So 4 polypeptide chains individually coil and then come together to arrange to form the hemoglobin structure, right? So this statement is absolutely true. So correct answer will be statement 1 is false and statement 2 is true. So A will be the answer here, right? Now let's move ahead. What will be the answer of this question? Write down in the chat box. What will be the answer of this question? Yes, absolutely, Akib. So what will be the answer here? Given below are two statements. One is labeled as assertion A, other is labeled as reason. So endometrium is necessary for implantation of blastocyst. Is it true? Yeah. It is absolutely true. Endometrium lining of uterus is required for blastocyst implantation. In absence of fertilization, corpus luteum degenerates that cause disintegration of endometrium. This statement is also true. But they're talking about the implantation in above line in the assertion, but in the reason they are not explaining the reason of this. So both the statement individually are correct, but reason is not correct for assertion, right? So in that ways, which one will be the correct here? Both assertion, reason are true, but reason is not explaining the assertion, right? So correct answer will be C here. So in absence of fertilization, we know corpus luteum disintegrated. And when corpus luteum disintegrated, what will happen? Obviously, no progesterone will be released. So when there is no progesterone, means no uterus lining will be maintained. And uterus lining will be broken down to cause the menstrual bleeding, right? So that they are talking about absence of fertility here. They are talking about the fertility here, means implantation here. So both of the statements are true individually, but reason is not correct for assertion here, right? So now you tell me the answer of this question, easiest one. Tell me the answer here. What will be the answer here? Absolutely, broad palm with single, with palm creases is visible in a person suffering from Down syndrome. This is the correct question again, right? So broad palm with palm creases is visible in a person suffering from Down syndrome. So correct answer will be B here. Next question, which of the following statements are correct regarding female reproductive cycle? In non-primates, so what will be the answer? Write down in the chat box. What will be the answer here? Write down in the chat box. Absolutely. So in non-primate female mammals, cyclical changes during reproduction are called estrus. Very true. First menstrual cycle begin at puberty is called menopause. No, it called menarche. So this is incorrect option. Lack of menstruation may be indicative of pregnancy. Yes, it's true. Cyclical menstruation extend between menarche and menopause. Yeah, it starts from the menarche and ends at menopause. So that is the that is about the cyclical menstruation. So what will be the answer? A, C and D is correct. So correct answer will be A here, right? So can we go ahead? Can we go ahead? Yeah. So this is the other question. Which of the following statements are correct? So which of the following statements are correct regarding skeletal muscle? Muscle bundle are held together by collagenous connective tissue layer called fascicle? No. All the muscle bundles are known as fascicle, right? Fascicles are basically muscle bundle and they are collectively bound by the fascia. Fibrous cord known as fascia, not fascicle. Sarcoplasmic reticulum of muscle fiber is a storehouse of calcium ion. Very true. Striated appearance of the skeletal muscle fiber is due to distribution pattern of actin and myosin. Yes. So obviously this thin and thick protein actually make the bands, right? Striated means band. So those bands which are visible in the skeletal muscles are due to arrangement of actin and myosin. M line is considered as functional unit of contraction called sarcomere. No, M line is basically bisecting the actin, right? So myosin. So myosin protein is basically bisected by M line. This is not functional unit of contraction. Sarcomere is functional unit of contraction of muscle, right? So they have given it wrong. So what will be the answer here? B and C are correct, right? So yeah, B is correct and C is correct. So correct answer will be C here. So let me take you ahead now. This is the other question. What will be the answer? Write down in the chat box. What will be the answer here? Yes. So which of the following is characteristic feature of cockroach regarding sexual dimorphism? Presence of anal cerci, it is present in male as well as female, right? The dark brown body colors and anal cerci, so it's common for both. Presence of sclerites means their body like covering, so obviously it's present in all cockroaches. But anal style is present in the male only, right? So correct answer will be C, repeated question. So let me take you ahead. Which of the following statement are correct? So what will be the answer here? Students, write down in the chat box. What will be the answer here? Which of the following are correct? Excessive loss of body fluid, that is water, from the body switching off? No. It will switch on osmoreceptors so that body fluid can be maintained. It will be increased by the osmoreceptor because it sends signal to the hypothalamus, and hypothalamus sends signal to the posterior pituitary gland to release which hormone? ADH hormone. And that ADH will reabsorb water in the body back, right? So ADH facilitate water reabsorption to prevent diuresis, means loss of water. Yeah, it prevent loss of water, true. ANF causes vasodilation. Yes, that's why it decreases BP, right? So yeah, this is true. ADH increases blood pressure. Very true. ADH responsible for decrease? No, it is responsible for increase in GFR. So you just tell me which statement are correct here? B, C and D only, right? So correct answer will be C here. Is it clear? Is it clear? So let me take you towards the next question. So you tell me the answer here. Logistic growth is the limited growth, right? So we are having the limited resources and obviously limited growth will be there. Exponential growth will be there when there is the unlimited resource availability. Expanding age pyramid means percent individual of pre-reproductive age is largest. So yeah, children are more in comparison to old and the adult, right? So means after a few years, their population will be expanding population. Stable means pre-reproductive percentage of individual of pre-reproductives means the children and the adult are having the same ratio. So that means after few years, the population size will be stable only, right? So what will be the answer here? A goes with second, B goes with first. So obviously this will be the answer. C goes with third and D goes with fourth. So yeah, correct answer will be B here. So let me take you towards the next question. Unique mammalian characteristics are. So what are the unique character of mammals? Pinna, they have pinna, yes. They are monocondylic, no. Mammals are dicondylic. And mammary glands. So they are talking about the characteristics means that is present only in mammals. So only in mammals have hair, their body is covered with hair. They have pinna and they have mammary glands. So these are some unique character means that is present only in mammals are these three characters, right? So correct answer will be C here. Let me take you towards the next question. So you have to tell the sequence of mRNA. Oh, so they have given the sequence of mRNA. And you have to tell the sequence of coding strand, right? So you know that coding strand is basically what we called is basically same as mRNA except there is the T in the DNA, right? So coding strand is the DNA strand which is having the same sequence as mRNA except T. So what will be the answer? 5 A T C. So this will be the answer here. It's understood, right? So correct answer will be D here, right? So let me take you to the next question. So I hope all of you have done this correct in the exam also. These are easiest question, direct question from NCERT, right? So given below are two statements. Statement 1, during G0 phase of cell cycle, the cell is metabolically inactive. No, it's metabolically active. It's not proliferating, it's not dividing, but it is metabolically fully active. And statement 2 is centrosome undergoes duplication during S phase of interphase. Yes, so during S phase, centrioles get duplicated, centrosomes get duplicated, right? So statement 1 is incorrect, statement 2 is correct. So correct answer will be A here, right? So let me take you towards the next question. So these questions are really easy, direct from NCERT. Next question is correct statement with reference to core data. Write down in the chat box. Write down in the chat box from the animal kingdom. Presence of mid-dorsal solid double. No, they have single dorsal hollow nerve cord. So this is absolutely wrong. They have closed circulatory system. Presence of paired pharyngeal gill slits. Presence of dorsal heart. No, they have ventral heart. They are triploblastic, yes, and they are coelomate, not pseudocoelomate. So this is also incorrect. So what will be the answer here? B and C. So correct answer will be C here, right? So what will be the correct answer? C answer will be correct here. Let me take you towards the next question. You tell me the answer here. So write down the answer in the chat box. Yes, what will be the answer here? So mast cell you must have done present where? It is present in the areolar connective tissue. So you just tell me the function of mast cell also. Inner surface of bronchioles have the ciliated epithelium. Tubular part of nephron, that is DCT, PCT, have cuboidal epithelium. And blood is the specialized fluid connective tissue. So A goes with second, B goes with first, C goes with fourth, and D goes with third. What will be the answer here? Answer will be D. What will be the answer? Answer will be D here, right? So let me take you towards the next question. Which one of the following is not an advantage of inbreeding? So what will be the answer here? Write down in the, so it was the tricky question. They are talking that which is not an advantage of inbreeding means it happens in inbreeding. It is the problem of inbreeding, but not advantage. It's a disadvantage, right? So what will be the answer here? It decreases the productivity of inbreed population after continuous inbreeding. Yes. So we know that it causes inbreeding depression, which is not the advantage of inbreeding method. Rather, it is the disadvantage. So they are asking you disadvantage of the inbreeding. So what is the disadvantage? Tell me. Which is the disadvantage? Obviously, inbreeding depression, right? They show inbreeding depression if we continuously inbreeding them. So that means it's not advantage of this process. We are not getting advantage of it. It's disadvantage. It decreases homozygosity. This is absolutely wrong about the inbreeding. And obviously, it's not advantage anyway. So correct answer will be A, out of all, right? It exposes harmful recessive genes that are eliminated by, so elimination of the less desirable gene and accumulation of superior gene. This is the advantage. So out of all, we will go with the A option, right? So is it true? So any doubt in this question? So I'm sure that you have got the question that they are talking about the disadvantage. So you have to opt the appropriate option out of all, right? So this is not anyway, this doesn't happen in the inbreeding, right? So this happened and this is disadvantage. So this is the next question. Which of the following statements are correct? Basophil are most abundant cell of the total WBC. Do you agree? Write down in the chat box. Do you agree? So obviously basophil is not the most abundant WBC, rather it's neutrophils, right? That means PMNL cell. Neutrophils are the most abundant cell of WBC. They secrete, basophils secrete histamine, serotonin, heparin, very true. It is involved in the inflammatory response, very true. Have kidney-shaped nucleus? No, it is having S-shaped nucleus, 3 lobes in the nucleus, right? Kidney-shaped nucleus seen in the monocytes, not in the basophil. So this is wrong. Basophils are granulocyte. So do we agree with it? No. It's not a granulocyte, rather it is granulocyte, right? So obviously, eosinophil, neutrophil and basophil are granulocyte. So correct answer will be B statement is correct and C statement is correct. So correct answer will be D here. B and C only. Got it? So let me take you towards the next question. So you just write down the answer in the chat box. So only one student seemed very super excited. I guess he got like more than 700. Huh? What about you? Have you got more than 700 and plus 650 above? Definitely he get. That's why he's super excited. And that's really nice. That's really nice. Right? So which of the following are not under the control of thyroid hormone? So you just tell me which is not the function of thyroid hormone. Write down in the chat box. Maintenance of water and electrolyte balance. It is the function of thyroid gland. Regulation of BMR. It is also function of thyroid hormone. Normal rhythm of sleep and wake up cycle is not function of thyroid hormone. Rather, it is the function of pineal gland, right? Melatonin hormone. So obviously, it's the function of melatonin hormone. And development of immune system is done by thymus gland, and the thymus secrete thymosin to mature T lymphocyte. So obviously this is also not the function of thyroid gland. This is also not the function of it. Process of RBC formation, yes. It also, thyroid hormone also help in the erythropoiesis, right? So correct answer will be, means which is not the control of the thyroid hormone, is B and no, C and D. So correct answer will be C and D. So C and D is not the function of the thyroid hormone, right? Rest all the options are correct. Next question, select the correct statement. You just tell me what will be the answer here. I'm just seeing you. Yes. So now I can see many students become super excited. So it means like you have done really nice in the exam, is it? So keep it up. So all the best. So may you get the dream college. So tetrad formation is seen during leptotene, is it? Tetrad formation is seen during leptotene. It's absolutely wrong. During anaphase, centromere split and chromatid separate. Very true. Terminalization takes place during pectine. No, not at all. It happens during dikinesis, right? Nucleolus, Golgi complex, ER are reformed during telophase. Is it? It's true. So they are asking correct statement. Crossing over takes place between sister chromatid of chromosome. No. Crossing over takes place between non-sister. So this is absolutely wrong. So you have to tell the correct answer, that is B and D only. So this is the correct answer, right? Agree? What will be the answer here? So B and D only, that is C, right? So let me take you ahead. What will be the answer here? Part of human brain that helps in regulation of sexual behavior, expression of excitement, pleasure, rage, fear, etc., etc. So what will be the answer here? It's very easy. We know our emotional brain is limbic system, along with hypothalamus control all these activities, right? So our limbic system and hypothalamus together is basically controlling all these activity, all these emotions, right? So these are emotional brains. So let me take you ahead. I guess this is the last question of this code. Yeah. So in cockroach, excretion is brought about by phallic gland? No. So conglobate gland present in the conglobate gland present where? So it is present in male, and obviously it helps in secretion of outer layer of spermatophore that you must have done in your class. So it doesn't have any role in excretion. Uricose gland, yes. Nephrocyte, yes. Fat body, yes. Collateral gland, no. It is present in female. and it secrete ootheca, right? It form ootheca. That means egg case around the egg. So B, C and D will be the answer. So correct answer is D here. So it was really easy paper, right? So I hope you have done well, and those kids who have not done well, it's okay. You just take time and decide for yourself what you have to do, right? So always, there is always, always a hope, right? So I'm sure that you are doing really well. So there is always blessing in disguise, right? So have a great day. See you all, and all the best for your result now. Bye-bye, guys.
Hello, 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 hello. Hello, 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 hello. Am I audible? All of you? Can you hear me? Here we go. Hello, 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 hello. Welcome back. So once again, a very warm welcome to all the champions. And as I promised, the complete paper, all the nearly 48 questions out of 50 are from exactly what you have studied. So let's begin the discussion. It was actually a very delightful moment when I saw the paper, and exactly word to word, some of the questions, the statements, the questions, the formulas, the concepts that we have seen, exact the same paper has came in this NEET 2023. So it's really, I'm really happy with the paper. So let's get started anyhow. So here we go. We are beginning with the discussion of physics. The first question in front of you is this. So have a look at the question, what it says. Two bodies of masses m and 9m. Okay, the masses are m and 9m are placed at a distance of r. The gravitational potential, they are asking about the gravitational potential on the line joining the bodies where the field equals to 0. So let's, the first step that we always do, let's make a small sketch. The masses have been given to you as m and 9m. What are the masses being given to you? The masses have been given as m and 9m. Okay, fine. Where on this line the net field, the net force can be 0. It can be 0, let's say somewhat over here. Okay, fine. So this distance is x. This distance is going to be r minus x. What is the expression for the field? What are we going to do in this question? We are going to solve this question in two steps. First step, find the location where the field is 0. And second step, find out the potential at that location. What I say? First step, find the point where the field is 0. Second step, find the potential at that point. So we are saying that E1 equals to E2. Or let's call it as, this is the point, okay? So field 1, E1 or I1, whatever you denote it, equals to E2. So Gm divided by x square equals to G times of 9m divided by r minus x the whole square. Okay, fine. So this gets cancelled out. This gets cancelled out. And what are you left with? You are left with multiply 9 over here. This is 3x the whole square equals to r minus x the whole square. So what are you left with? You are left with r, x equals to r by 4. You are left with x equals to r by 4. So one location is r by 4. The other location is going to be what? It's going to be r minus r by 4. So what is the potential going to be at this point, is at this location? Let's go and stand over here. Stand over here at this point. So what are you going to say? You're going to say that the potential is V1 plus V2. Okay, fine. You have written V equals to V1 plus V2. What is V? V is minus G m divided by distance. Distance is how much? Distance is r by 4. I multiply straightforward r by 4 over here. Minus G m divided by distance. It's r minus x. R minus r by 4. That is 5r by 4. What is the mass over here? The mass is 9m. What is the mass over here? The mass is 9m. So what are you left with? What are you left with? Let's solve it. Let's have a look over here. This is r minus x. That means r minus r by 4. This is 3r by 4. Okay, this is 3r by 4. So G 9m divided by 3r by 4. So this 3 gets cancelled out. This 3 gets cancelled out over here. This is 12. This is 16. So minus 16 Gm divided by r. Do we have some option like this? Do we have some option like this? Definitely yes. What's the option? The option is D. So the answer for the first question is D. Any doubt in the first question? Any doubt, any sort of confusion in the first question? Let's come to the second question. Have a look at this question. What it says? It says that in hydrogen spectrum, the shortest wavelength corresponds to maximum energy. In the Balmer series, it is given as lambda. So the first thought that comes in our mind is, let's have a rough representation of the spectral lines. Okay, fine. That's cool. So we know that these are the quantum numbers 1, 2, 3, 4. If you talk about the shortest wavelength of Balmer series, Lemon, Balmer, Paschen, Brackett. Okay. So the shortest wavelength of Balmer series is when the transition is from the extrememost orbit, that means from infinity to n equals to 2. Okay, this is for Balmer. This is the shortest wavelength for Balmer. I am calling it as lambda BA. Call it as lambda BA. Okay, fine. This is given as lambda. The shortest wavelength of Brackett, that means from infinity to n equal to 4. What is the expression? The expression is 1 by lambda, lambda for Balmer. This has been given as lambda. R bracket 1 by n1 square minus 1 by n2 square. Okay, fine. That's cool. That's fine. So what are we left with? We are left with lambda Balmer. Solve it and you get it as 4 by R. Expression number 1. That's okay. That's fine. This is Balmer and this is Brackett. So let's call it as BR. Bracket is BR. Okay. Bracket is BR. This is BA. Okay, fine. Next expression. 1 upon lambda Brackett is R bracket 1 by 4 square minus 1 by infinity. So what is the next equation that you're going to get? You're going to get lambda for Brackett is 16 divided by R. Okay, that's fine. So these are the two expressions that we are left with. These are the two expressions that we are left with. Divide both of them. Divide both of them. Straightforward, what do you get? You get lambda Brackett is 4 times of lambda Balmer, and lambda Balmer is lambda. So the answer is 4 times of lambda. So another simple question. Second question, answer is 3. Have a look at the question. Have a look at the solution. Have a look at the answer. Let's come to the next question. Third question in front of you is this. Read the question. A very simple question from optics. What the question says, light travels a distance x. Okay. Light travels a distance x in time t1 in air and 10x in time t2 in some another medium which is dense. Okay. Okay. What is the critical angle for the medium? First of all, what is the expression for the critical angle? The expression for critical angle is this. Okay, fine. That's cool. Now let's apply the logic over here. The speed in air, we know that is c. What is speed? That's distance upon time. So this is the expression number 1 corresponding to air. Light is traveling in air. So the expression corresponding to air has been given in front of you. Simple. Next point. In some another medium, let's call the speed is how much? The speed is v, or let's call it as v2. Okay. So what is v2 or what is v medium? I'm calling it straightforward as v medium. Okay, that's 10x, that means distance, divided by, what is the time? Time is t2. Okay. These are the two expressions. Are we aware about the expression for mu? Yes, we are. Mu is nothing but c by v. Mu for any given medium is nothing but c divided by v. So can we divide the two expressions and find the expression for mu? Can we divide these two equations and find the expression for mu? Definitely we can. So divide both of them. What are you left with? This, divide both of them. This is x gets cancelled out. This is t2. T2. What is it? This is, just a second. This is x divided by t1 and this is 10x divided by t2. Okay, fine. So this is t2 by 10 t1. This is mu is t2 by 10 t1. And what is sine theta c, or simply what is theta c? That's sine inverse of 1 by mu. So what is 1 by mu? 1 by mu is 10 times of t1 divided by t2. 10 times of t1 divided by t2. Have a look at the situation. Have a look at the answer. So what answer are we left with? Is there some option that is matching? Sine inverse of 10 t1 divided by t2. Is there some option which is matching? Definitely yes, there is one option, that is A. Another simple question. I would call it a very simple question. Okay, so this is the solution in front of you. Have a look at the question. Have a look at the solution. If there's any sort of doubt, let me know. or else we are getting ahead to the next question. The next question in front of you is this. Okay, have a look at the question. Magnetic energy stored in an inductor. What are we talking about? We are discussing inductors. Okay, the inductance has been given to you as how much? That has been given to you as 4 micro Henry. Okay, what is the current? Current has been given to you as 2 ampere. You need to find out the magnetic energy. What is the expression? Straightforward, 1 by 2 Li square. Straightforward question, one of the easiest question that can happen with NEET examination. So this is a straightforward 1 by 2 into 4 into 10 to the power minus 6 into I square is 4. So what are we left with? What are we left with? We are left with 8 microjoule. Answer is A. Have a look at the question. Have a look at the solution. This is in front of you. Okay. So let's take the discussion ahead. Here we go. Here we go. Here we go. Here we go. This is micro, not milli. 2 options were given exactly looking very identical, but the answer is A. Okay, fine. Come to the next question. What the next question says? Amount of energy required to form a bubble. The first thing that should strike in your mind when you listen the word bubble is that there are 2 surfaces. Master, what are you saying? I am saying that remember the difference of drop versus bubble. What was the difference of drop versus bubble? Very clearly in every single session I shouted on this difference, that there is only one difference between a drop and a bubble, that there is liquid air. Only one interface, and for a bubble, air, liquid. This is liquid and this is air. Air, liquid, and liquid air. Getting what I'm saying? Air, liquid, air. There are 2 surfaces. There are 2 surfaces. This is the only difference between a bubble and a drop. What we need to find out? We need to find out the amount of energy required. What is the amount of energy required? The work done, the surface energy, that's T times of delta A. Okay. Surface tension into area. There are 2 areas. Area number 1, which is inside. Area number 1, which is inside. Area number 1, which is inside. Area number 2, which is outside. So what do we get? What do we get? We get 2 times of 4 pi r square. 2 times of 4 pi r square. Why this 2? I hope that the logic is absolutely clear. So this is nothing but T, surface tension, has been given to you as 0.03. That means 3 into 10 to the power minus 3 into 2 into 4 into 3.14 into what is R? That's R square. That's 4 into 10 to the power minus 4. So how do we solve this? 3 2 the 6, 4 the how much? 6 4 the 24 into 4, 96. So this is 96 into 3.14 into 10 to the power how much it is? This is 10 to the power minus 7. 10 to the power minus 7. So if I roughly take it as 100, this is something around 300 into 10 to the power minus 7. So the only option that is matching, from here is this one. Okay, multiplied by 2, divided by 2. So what are you left with? This is 100 into 3, 300. Okay. Oh, sorry, this is 10 to the power minus 3. 3 to the 6, 4 to 24, 96. Okay, yeah, fine. The only option that matches is this much. Surface tension. Okay. 2 into 4 into pi into r square. R square is 4 into 10 to the power minus 7. 3 to the 6, 6 4 the 24, 24 into 4, 96 into 3.14, nearly 300. And this is 10 to the power minus 7. Okay, that's fine. So this is 1010, 10 to the power minus 4 minus 5. Yeah, D is the correct answer. How many of you got this? Done. Let's come to the next question. The next question in front of you is this. What the question says: the potential energy of a long spring stretched by 2 centimeter is U. If the spring is stretched by 8 centimeter, 8 centimeter, the potential energy stored in it will be. What is the expression for the potential energy for a spring? This is simply half kx square. Okay, this is simply half kx square, isn't it? Yes. So we are writing that the potential energy U, that is half into k into x square. How much is x? X is 2 centimeter. That means 0.02 meter square. Fine. Now what are they saying? They are saying that the spring is stretched by 8 centimeters. So U dash is going to be how much? 1 by 2 times of k into this is 0.08 the whole square. So the distance is how much? It's 4 times. If I simply take the relation U is proportional to x square, so U dash by U is x dash divided by x the whole square. If you make x dash as 2 times of x, it's going to be 4 times. The energy is going to be 4 times. If you make x as 4 times of the original, energy is going to be how much? It's going to be 16 times. So the answer is 16. Answer is going to be 16. Simple. Yes or no? Tell me quickly. Done. Let's come to the next question. The answer is 16U. Another question. A football player is moving. Where the football player is moving? He is moving towards the south and suddenly turns eastward. The football player is moving towards the south, and what happens? Suddenly he moves eastward. Okay. This is the initial. This is the initial direction. This is the final direction. This is the initial direction. And this is the final direction. Okay. What do you need to find out? You need to find out the force. What is force? Force is rate of change of momentum. What is force? Force is rate of change of momentum. What is change in momentum? That's final momentum minus initial momentum. That's final momentum minus initial momentum. Okay. So can you write this as, final momentum plus minus of initial momentum? Can you write that the force is nothing but final momentum plus negative times of the initial momentum divided by the time? That means the vector sum of final and minus initial. So if this is the direction of initial momentum, this is the direction of final momentum. What is the direction of minus initial momentum? This is the direction of minus initial momentum. That means minus I is along this direction. I repeat. If this is initial, this is final. This is initial, this is final. This is minus initial, opposite to initial, isn't it? So the resultant of initial and final lies somewhat over here. What direction it is? South, east, north. So the force is acting along what direction? The force is acting between north and the east. That means it's along the northeast direction. Okay. Simple logic. Tell me quickly, yes or no? Are we clear with this point? Are we clear with this logic, all of you? In the chat, just a yes or no. Can we move ahead? Any kind of confusion with the discussion? No? Perfect? Okay. Great. So here we go. Here we go. Next question. The ratio of frequencies of fundamental harmonic produced by an open pipe to that of the closed pipe. We are talking for open pipe. We are having the discussion for open pipe and closed pipe. Okay. Open pipe and closed pipe. Okay, fine. Great. So what is the formula for the frequency for the open pipe? Tell me. That's V divided by 2L, isn't it? N times of V by 2L. What is the formula for closed? That's 2N minus 1 V divided by 4L. A quick yes or no. Tell me quickly. Yes or no. Done. So what has been given? They have said that you need to find out the ratio of the fundamental frequencies. So what are we going to do? We are going to substitute n equals to 1 over here in both of the expressions. So we are left with f naught, that means fundamental frequency for open is going to be what? It's going to be V divided by 2L, and fundamental frequency for the closed is going to be n equal to 1. That's 2 minus 1 is how much? 1. V by 4L. Okay. So if you divide both the expressions, what are you going to get? You're going to get the ratio of fundamental frequency for open is to close. V by L gets cancelled out. You are left with 2. So the answer is 2:1. So the answer is C. Answer is C. Have a look at the question. Have a look at the solution. Are we very clear with the point? Let's take the discussion ahead. Here we go. Next question from current electricity, the magnitude and direction of the current. They are talking about the magnitude and direction of the current in this given circuit. Great. A very simple question. Let's apply the Kirchhoff voltage law. Let's apply the KVL. How do we apply the KVL? As I say, go and stand at any one of the point. Go and stand over here. Start jumping from this point to other points. Okay. So if you go along this direction, this is the direction of current. Along the direction of current, the potential decreases. Or let's say the current moves along the direction of reducing potential from higher to lower. So this is going to be minus of I into R. R is 2. Great. Minus 2 plus, this is going to be plus 10. Plus 2 minus, this is going to be minus 5. Minus I into 1. Come over here. Minus I into 7. Come again at the same point. Complete the loop, equals to 0. So what are we left with? 2i, 3i, 7i. That means minus 10i. 10 minus 5 plus 5 equals to 0. So I equals to 1 by 2 ampere. That means the answer is 0.5 ampere. And the direction is from A to B through E. So the answer is C. The answer is 3. Yes or no? How many of you got this answer? Great, 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 great. Have a look again at the question. Let's have a sip of water. You also drink water. Yeah. Okay. So here we go. Next question in front of you is this. An AC source is connected to a capacitor. Nice question. This was a nice question. Connected to a capacitor due to the decrease in the operating frequency. Let's draw a simple circuit. This is the AC source, and this AC source is connected with the capacitor. This is the AC source and this is connected to the capacitor. Great. Great. Alternating source. And you know that what is the resistance offered by the capacitor? That's called the capacitive reactance. That's called the capacitive reactance. What is the expression for XC? That is 1 by omega C. What is omega? Omega is 2 pi F. 2 pi sorry, FC. So what is happening in the situation? What is happening? Due to decrease in frequency, frequency is decreasing. So what is going to happen? The capacitive reactance is going to increase. And if the capacitive reactance increases, if the resistance increases in a circuit, current decreases. So what are you going to say? You are going to say that the current decreases. What current is it? This is the conduction current. Sir, but they are not asking about the conduction current. You are giving the answer for conduction current. They are asking about the displacement current. First option, capacitive reactance remains constant. Wrong. Capacitive reactance decreases. Wrong. Displacement current increases or decreases. You have given the answer for conduction current. But we know that the conduction current equals to the displacement current. Yes or no? Definitely yes. So if the conduction current is decreasing, the displacement current is decreasing. Cool. So the displacement current is going to decrease. So the answer is 4. The answer is going to be 4. The next question in front of you is this. Have a look at the question. The error in the measurement which arises due to unpredictable fluctuation in temperature and voltage. Remember the day we discussed errors. I clearly gave this chart to you. Errors are of two types. Errors are of two types. One are systematic. And another is random. Another is random. In systematic error, what I gave? I gave instrumental error. Instrumental error, personal error, and experimental error. And what are random error? That occurs due to any irregularity. There is no pattern that is being followed over there. So the answer is going to be random errors, and the answer is nothing but A. A is the correct answer. Okay, so every question till now is from your notes. Any student who has just gone through the notes, before the NEET, I guarantee 44 to 45 questions correct out of these 50. Okay. Next question coming in front of you is this. Read the question, what it says. A very basic, a very simple question. The angular acceleration of a body moving along the circumference of a circle. If a body is moving, if a body moves this way, if a body moves this way, if a body moves in a circle, so we know that the angular displacement, theta, omega, and alpha, all three of them are axial vectors. What are they? They are axial vectors. Okay, so the answer is going to be along the axis of rotation. Along the axis of rotation. Cool? Clear? Let's take the discussion ahead. And here we go. The next question in front of you is this. Have a look at the question. If the galvanometer G does not show any deflection in the circuit, then the value of R is going to be. What are they saying? They are saying that the galvanometer does not show any deflection. That means there is no current in the galvanometer. That means the potential of this orange dot and this orange dot is same. So let us apply the nodal method. If I take this point to be at 0 potential, this point to be at 0 potential, this point is also at 0 potential, this point becomes at 100 volt. Okay, and since no current is flowing in this branch, this is 2 volt, this is also 2 volt. Okay, fine. So now what I'm going to do, I'm going to make two expressions out of these resistances. I repeat, first expression, I am going to write for this particular resistance. I am straightforward going to write that V equals to I times of R. V between these 2 points, let's call them as A and B. Okay. This point is also B. This point is C. Great. So VA minus VB. 100. Oh, sorry. This is 10 volt. Okay, this is 10 volt. I'm really sorry. This is 10 volt. This is 10 volt. So 10 minus 2, that is 8, equals to I times of 400. So I is 8 divided by 400. Okay, that's great. This is the expression for current that we have just found, ampere. Okay, unit ampere. Apply the expression over here. The same current goes through this circuit because no current is going in the right part of the circuit. The current in this whole loop is 0. Okay, so current is not going over there. So I'm writing that the same current should flow through this particular branch. So this is 2 minus 0. That means V. B minus VC. VB minus VC equal to I. I is how much? I is 8 divided by 400 times of R. Okay. So how much are we left with? This is 2. 2 4s are 8. This is 100. So R is equals to 100 ohm. So this was again a very simple question and the answer is D. and the solution is in front of you. Have a look at the question, have a look at the solution. Cool. Let's take the discussion ahead. Here we go. The next question in front of you is this: the resistance of a carbon resistor determined by the color codes B. Roy of Great Britain has a very good wife. She wears gold, silver, and sometimes no color. Okay. So these correspond to the tolerances, and these are the digits, and the expression for resistance is A into B into 10 to the power C plus minus the tolerance, plus minus the tolerance, plus minus T. Okay. So what has been given to you? It has been given to you B. Roy of Great Britain. Okay. They are asking about the color in the third strip. They are asking about the color in the third strip. 2 into 10 to the power 22 into 10 to the power 3 plus minus 5. So it is going to be 2, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. 5%, 10%, no color, 20%. Okay. So this is red, second strip red, third strip orange. So if this is the carbon resistor, red, third strip is orange. Okay. Third strip is orange. So the answer in front of you is orange. Is this point very clear? Simple question. Answer is orange. Here we go. Let's take the discussion ahead. The next question coming in front of you is from thermodynamics. A Carnot engine has an efficiency of 50% when its source is at a temperature of 327 degrees centigrade, the temperature of the sink. So what is a Carnot engine? Simply an engine, the ideal engine that gives you maximum efficiency while working between a source and a sink, a higher temperature source and a lower temperature sink, isn't it? This is what we call as engine, and what it does, it gives you some useful amount of work done. What is the expression for efficiency? That's work done upon heat input into 100. That's simply 1 minus T2 by T1. This is the fraction. If you multiply it by 100, you get the percent. This is source. Sorry, this is lower temperature. That means sink. This is source. Higher temperature. Source. So efficiency has been given to you as 50%. That means 1 by 2 equals to 1 minus sink upon source. What has been given to you? Source temperature is 327. Higher temperature is 327. You need to find out the lower temperature. Let's call it as T dash. Divided by the source temperature. Add 273. Add 273 to this. What do you get? This is 600. This is 600. So what are you left with? You are left with T dash divided by 600 is 1 minus 1 by 2. That is 1 by 2. So T dash is how much? T dash is 150 Kelvin. Do we have some option like this? No, we do not have any option like this. This is 150 Kelvin. Oh, sorry. This is 300 Kelvin. Sorry. This is 300 Kelvin. If this is 300 Kelvin, what is it in the degree centigrade? Minus 273. That means 27 degrees centigrade. B is the correct answer. Another simple question. Another simple solution in front of you. Have a look at the question. Have a look at the solution. I hope this was correct. You did this correct. Now let's come to the next question. The net magnetic flux through any closed surface. Gauss's law of magnetism. If you take a small magnet inside a closed surface, you know that this is South Pole, this is North Pole. The magnetic field lines from north are outside, somewhat like this. The magnetic field lines in the south enter somewhat like this. So the total magnetic flux through the surface is going to be 0. The total magnetic flux through the surface is going to be 0. So the answer is going to be B. Okay. Next question in front of you is this. Have a look at the question. A full wave rectifier circuit, semiconductors, full wave rectifier circuit consists 2 PN junction diodes. Yes, definitely. One we keep as forward bias, another we keep as the reverse bias. There is a center tap that keeps on switching between the two diodes. I hope you remember this. There is a center tap, transformer, capacitor and a load resistance. Which of these components remove the ripple, AC ripple, from the rectified output? So this is a very direct question. The capacitor and filter which is used in the circuit is used to remove the AC ripple. What? Capacitor and filter. Okay. This is the answer. Is the answer. I hope that the discussion is cool, that the discussion is fine. Let's come to the next question. Another question in front of you is this. All of you, do the question. Read the question. The minimum wavelength of X-rays produced by an electron accelerated through a potential difference of V volts is proportional to. You know that E, that is nothing but HC divided by lambda, equals to E times of V. So from here you can say that lambda is inversely proportional to V. So the answer is going to be C. So the answer is, just a second. So the answer is going to be yeah. What I said? The answer is going to be C. Let's come to the next question. Let's come to the next question. Another question in front of you is this. This was a tricky question. This was a nice question. I'm making a star over it. A lot of students got confused in this question. The common feedback that I got from the students. The temperature of a gas is minus 50 degrees centigrade. Temperature is minus 50 degrees centigrade. To what temperature the gas should be heated so that the RMS speed is increased by 3 times? See, you are having some amount X. Increase this amount, increase to 3 times, increase this amount to 3 times. What do you get? You get 3x. You have some amount X. There is a difference. Try to understand. There is a difference. Try to understand. You have some amount X. Increase this amount by 3x. So you get X plus 3x. Oh my God. X plus 3x, that means 4x. Lot of students got confused in this. Tell me how many of you got confused in this? This is where 90% of the students got confused in this small word by. So we know that V equals to root 3 R T by M. So V is proportional to root T. That means V1 by V2. That means V1 by V2 is root over T1 by T2. So you can clearly say that V1 by V2 is how much? V2 is 4 times. It is increased by 3 times. So that means V2 is 4 times of V1. So you can say that 1 by 4 equals to root over T1 by T2. So T2 is 16 times of T1. How much is T1? T1 is 273 minus 50. That means 223. Multiply 223 by 16 and subtract it from 273. So how much is 16 into 223? 16 into 223. Do this small calculation. I'm using calculator for quick calculations. Okay, you do it by yourself. So this is simply 16 into 223. This is 3568 minus 273. I'm writing 3568 in Kelvin. 3568 in Kelvin. Subtracted by 273, you get 30. Just a second. This is 273 minus 15. 273 minus 50. 223 into 16, 3568 minus 273. Okay, fine. This is 3295. I did a small calculation error. 3295 degree centigrade. This is Kelvin. This is degree centigrade. This is the answer in front of you. Okay. You got confused in this? You got confused in this? I knew that. Okay, so here we go. Let's come to the next question. The another question coming in front of you is this. Have a look at the question. In series LCR circuit, the inductance L is 10 millihenry, capacitance C is 1 microfarad, and the resistance is 100 ohm. The frequency at which the resonance occurs, the resonance frequency they are talking about. What is the expression for resonance frequency? That's 1 by root over LC. Okay, so what is L? What is L? L is 10 millihenry. Okay, what is C? C is 1 microfarad. Okay, so this is 10 to the power minus 2. This is 10 to the power minus 2. This is 10 to the power minus 6. So omega is 1 by root over 10 to the power minus 2 into this is 10 to the power minus 6. So this is 10 to the power minus 8. This is 10 to the power minus 4. Omega is 2 pi f. Omega is 2 pi f. This is 10 to the power minus 1 upon 10 to the power minus 4. Take it upside, you get something like this. You get the expression something like this. So frequency is 10 to the power 4 divided by 2 pi. Okay. So you can write 10 to the power 4 as 10 to the power 2. That means 100 into 10 to the power 2. Smart calculations. 2 into 3.14. 2 into 3.14, nearly 6.28. 6.28. Divide 100 with 6.28. Divide 100 with 6.28. This is going to be a term of nearly 16, less than 16. So 15.9 into 10 to the power 2. You can write it as multiply and divide by 10. You can simply write it as 1.59 into 10 to the power 3. That means 1.59 kilohertz. 1.59 kilohertz. What do you say? Simple question. Another very simple question it was. Answer is in front of you. Note down the question, note down the solutions. Let's come to the next question. How many of you got this correct in the chat? In the chat, tell me. How many of you got this correct? Next question in front of you is this. Read the question. A 12 volt 60 watt lamp, 12 volt 60 watt lamp is connected to the secondary step down transformer whose primary is connected to AC mains of 220 volt. Okay, that's fine. Assume that the transformer to be ideal. What is the current in the primary winding? What is the circuit being given to you? See, this is the AC supply. Okay, it's given something like this. The system has been given to you something like this, and there is a lamp that's being connected. Somewhat like this. Okay, this is the circuit in front of you. 12 volt 60 watt lamp. 12 volt 60 watt lamp. And what is the power rating of this AC source? This is 220 volt. This is 220 volt. This is 220 volt. Okay, that's fine. What you need to find out? You need to find out the current in the primary winding. You need to find out the current in the primary winding. Is the power being given to you in the secondary? Yes. Potential being given to you in the secondary? Yes. So you can, let's call it as 1. Section 1, primary, secondary. Power is V times of I. So can you find out the current in the secondary? Definitely yes. What it's going to be? It's going to be P2 divided by V2. That means 60 divided by 12. This is 5 ampere. This is 5 ampere. Okay, that's fine. So once you found the current, you know that the power is transferred undiminished. So you can say that P1 equals to P2 Okay, P1 equals to P2. P is VI, V1, I1 equals to V2, I2. That's fine. So you can clearly say that V1 divided by V2 times of I1 equals to I2. So what is the expression for I2 that you're going to get? V1 divided by V2, that's 220 divided by 60, isn't it? Into 5. How much it is? 220. Oh, sorry, 60 divided by 220. Really sorry. I2, 60 V1 divided by 220 into 5. Isn't it? So how much are we left with? Just a second. V1. Sorry. There's a slight mistake. Just a second. V1 is how much? V1 is 220. That's fine. Divided by V2. V2 is how much? Okay. We have done a mistake over here. I'm really sorry for this mistake. This is what? This is power. This is power. This is 60 watt divided by 12 volt. 60 watt by 12 volt. This is fine. This is fine till here. Current is fine. Okay. Now what we need to do? We need to find out the current. So this is going to be I2 equals to I1, that is 5 into V1 divided by V2. 5 times of V1 divided by V2. Now it's fine. This was the mistake was due to this small error that we did. We just wrote the unit as wrong. We wrote unit as volt over there. Okay, it was a very simple mistake. I'm really sorry for that. So that's how much. This is 60 divided by 220. So let's find out the value of 60 divided by 220. That means nearly of 6 divided by 22. So 3 divided by 11. Okay, 0.0222. That means 0.27. Nearly 0.27 ampere. 0.27 ampere. B is the answer. It was a nice question. Okay. How many of you got this correct? Have some water. I'm also having some water. You also drink some water. It's good to be hydrated. Okay. The answer is 0.27 ampere. Let's take the discussion ahead. Next question in front of you is this. Work functions of cesium, potassium and sodium are being given to you 2.14, 2.30 and 2.75. If incident electromagnetic radiation has an incident energy of 2.20 electron volt, which of the photosensitive surfaces emit photoelectrons? So we know that for photoelectric effect to happen, if this is a material and these are the electrons being embedded in the system, these are the electrons in the free electrons in the metal. If the light is coming, so what is the required condition? That the energy of the incident light should be greater than the work function of the material. E should be greater than the work function for the material. Then only photoelectric effect is going to happen. So what can we say? We can say that incident energy 2.20 is greater than the work function only for this fellow. Who is this fellow? This is cesium. So it is going to happen, the photoelectric effect is going to happen only for cesium. So the answer is going to be cesium only. This is the answer in front of you. How many of you got this correct? How many of you got this correct? Got this correct? Sure? Nice. Okay, fine. Great. Great. Next question in front of you is this. A very simple question from electric charges and fields. Question of dipole. What it says, an electric dipole is placed at an angle of 30 degrees with an electric field of intensity. Electric field intensity has been given to you. What is the electric field intensity? 2 into 10 to the power 5. What are they talking about? It experiences a torque. It experiences a torque of how much? 4 Newton meter. 4 Newton meter. Okay, that's fine. It experiences a torque of 4 Newton meter. That's great. Calculate the magnitude of charge on the dipole. Calculate the magnitude of charge on the dipole if the dipole length is 2 centimeter. So you know that if this is a dipole, what actually a dipole system is? Minus q and plus q separated by some length L. So what is the dipole moment? The dipole moment is charge into separation. I'm talking about the magnitude of dipole. This is charge times the separation. Okay, that's fine. What is the expression for torque? Tau is P cross E. That means P E sine of theta. What is torque? That's been given to you as 4. Let's find out the dipole moment from here. E is 2 into 10 to the power 5 into sine 30 is 1 by 2. This gets cancelled out. So dipole moment is 4 into 10 to the power minus 5. Okay. And what is dipole moment? Dipole moment is simply charge into separation. So P equals to Q times of L, or let's say Q times of D. So this is 4 into 10 to the power minus 5 equals to Q into length is how much? 2 into 10 to the power minus 2. So what are you left with? What are you left with? This is 2. And this is 10 to the power how much? What do we need to find out? Just a second. Tau, that is 4, is P E into sine 30. That's 1 by 2. So this is 2. So P is 4 into 10 to the power minus 5. That's fine. Okay. What are we left with? This is dipole moment equals to Q into separation. Length is 2 centimeter. So this is 4 into 10 to the power minus 5. That's Q into 2 into 10 to the power minus 2. So this is 2 into, yeah, correct, 10 to the power minus 3. Sorry. It was correct. It was correct. We did correct. I was just cross-checking the calculation. Okay. So this is Q is 2 into 10 to the power minus 3. That means 2 millicoulomb. How many of you got this? How many of you got this? Got this? Correct? Great. Okay. A is the answer. Let's take the discussion ahead. The next question coming in front of you is this. Read the question. A plane electromagnetic wave, a question from EM waves, traveling in free space. The electric field component oscillates sinusoidally at some frequency, and the amplitude for the electric field, that is E naught, has been given to you as 48 volt per meter. Then the amplitude of the oscillating magnetic field is going to be, what is the amplitude of oscillating magnetic field going to be? You know the relation E equals to BC. So B is E divided by C, isn't it? So what is E? What is E? E is 48 divided by C. What is C? C is 3 into 10 to the power 8. So 8 gets up. This is 10 to the power minus 8. 16 into 10 to the power minus 8. Multiply by 10. Divide by 10. So this is 1.6 into 10 to the power minus 7. Answer is D. Another simple question. Another very simple question. Done with this, all of you? Let's take the discussion ahead. In the chat, tell me, how many of you got this? Okay. Here we go. The next question in front of you is from errors. What the question says, a metal wire has a mass. What is the mass being given to you? Mass has been given to you as 0.4 and plus minus the error in mass is 0.002 grams. Okay, this is grams. Radius has been given to you. The information for radius has been given to you. 0.3 plus minus 0.001. Okay, that's fine. And the length in front of you is 5 plus minus 0.02 centimeter. The maximum possible percentage error in the measurement of the density. Maximum possible error in the density. So what is the density? If this is the wire, this is the wire in front of you. Let's say the length is L, the radius is R. So you know that what is density? Density is mass divided by the volume. And what is volume for this given system? The volume is area into length. Area is pi r square times of L. So you can straightforward write that the expression for density, the percentage error for density is going to look something like this. This is delta m by m in 200 plus 2 times of delta r by r in 200 plus delta L by L into 100. Isn't it? Cool. So substitute the data. Substitute M, substitute delta M, R, delta R, L, delta L. Calculate it. You are going to get something nearly 1.6%. Calculate and match the answer. You're going to get 1.6%. Have a sip of water. Done. Done. Here we go. The next question in front of you is this. What the question says: the half-life of a radioactive substance is 20 minutes. In how much time the activity of the sample drops to 1 by 16? That means if the initial number of sample is n naught, this is the initial number of sample. Okay. Finally, it is going to be n naught by 16. This is final number of samples. You know the expression. N equals to n naught divided by 2 to the power small n, where small n is nothing but the number of half-lives. T divided by T half. What is this? This is number of half-lives. Oh ho. Isn't it? Number of half-lives. Okay, that's fine. So what to do over here? Let's substitute the data. N is what? N is n naught divided by 16 equals to n naught divided by 2 to the power n. 2 to the power n. Let's find out the value of n. N is equals to 4. 2 to the power 4. So how many half-lives? T divided by T half. Okay. Number of half-lives. Sorry, this is going to be T half by T. This is going to be T half by T. Okay. Simply I'm writing number of half-lives. I'm writing it as number of half-lives, n. So this is going to be what? This is going to be T divided by T half, number of half-lives. If half-life is 20 minutes, you get T as 80. There are 4 half-lives. So what it says, the half-life has been given to you as 20 minutes. And 4 half-lives is going to give you as 4 into 20. That means 80 minutes. 80 minutes. So this is going to be, answer is A. 80 minutes is the answer. 80 minutes is the answer in front of you. Simple question. Let's take the discussion ahead quickly. What do you say in the chat, all of you? So here we go. The next question in front of you is this. Read the question. The ratio of radius of gyration of a solid sphere of mass m, radius r, about its axis to the radius of gyration of a thin hollow sphere about the same axis, same mass, same radius. So what it gives you? There are 2 spheres. One is solid, another is hollow. Same mass, same radius. One is solid, another is hollow. This is solid. This is solid sphere. This is hollow sphere. So what it gives you? It gives you radius of gyration I equals to m times of k square. What is I? I for solid is 2 by 5 m r square. This is going to be m k square. And I for hollow is 2 by 3 m r square. This is m times of k square. This is k1, this is k2. So divide both the expressions. What do you get? Divide both the expressions. What do you get? This gets cancelled. This gets cancelled out. So K1 by K2 is root over, this is 3 to the 6, this is 10, that means 3 by 5, but with a root, and it has not been given in the root has not been given, so this is going to be a bonus for you. This is going to be a bonus for you. Okay, so this is a bonus question. Root 3 by 5. Okay. Even if you didn't do this, you got the marks over here. Okay. Next question is a very simple question. These two are in parallel. So this is going to be 3 and 6 connected in series. This is 3 microfarad. This is 6 microfarad. So the net capacitance is going to be 3 into 6 divided by 3 plus 2, 3 plus 6. That's going to be how much? Is going to be 2 microfarad. So the answer is B. Simple. Let's take the discussion ahead. Read the next question. In Young's double slit experiment YDSC, they are talking about YDSE. Okay. Screen is moved away from the plane of the slit. Angular separation of the fringe remains constant. If you take the slit away, what happens for the angular separation? What is angular separation? Angular separation is beta divided by capital D. That's beta divided by capital D. Beta is lambda d by small d. You have to divide it by capital D. So what are you left with? You are left with lambda by d. You are left with lambda by d. Okay. So if the screen is moved away, if you change capital D, there is going to be no effect on theta. So the first statement is absolutely true. The first statement is absolutely true. If the monochromatic source is replaced by another monochromatic source of larger wavelength, angular separation is going to decrease. Angular separation is going to decrease. Angular separation is going to decrease? I don't think so. This depends directly upon lambda. So the second statement is false. Second statement is false. First statement is true. Second statement is false. Okay, so the answer is going to be C. The answer is going to be C. Very nice. Another question in front of you is this. Read the question, what it says. A bullet fired from a gun at a speed of 280 meter per second in the direction of 30 degree above the horizontal. The maximum height attained by the bullet. They are talking about the maximum height attained by the bullet. So what is the maximum height of a projectile? The maximum height for a projectile is given by what expression? This is U. You have projected it somewhat like this. So what do you get? What do you get? You get that H max is U square sine square theta divided by 2G. Okay. G has been given as 9.8. I am solving with, let's take 9.8 only. So U square is going to be how much? That's 280 into 280 into sine square theta. That means 1 by 4 divided by 2 into 9.8. So nearly I'm taking as 10. I'll be reducing the answer to some certain amount. Okay. So the answer is going to be, this gets cancelled out. This is 140. This is 270. Okay, this is 70, and this is how much? This is 140. Okay, 7 4 is 8, 28, 7 1 is 7, 8 9. So this is 980 meter. So I said that this, you have increased the denominator, so the answer is going to be reduced. So you have to increase it slightly. The answer is 1000 meter. Or simply take it as 9.8, you get exactly the same answer. Okay. Note it down. The answer is 1000 meter. Cool with this? Done with this? Let's take the discussion ahead. All of you. Here we go. Next question coming in front of you is this. Read the question. Photovoltaic devices can convert optical radiation into electricity? Photovoltaic. Photovoltaic, a device that converts the radiations of light into current. A device that converts the radiation into current. So photovoltaic device can convert optical radiation into electricity. Yes, that's true. Zener diode is designed to operate under reverse bias in breakdown region. That's also true. This is true. This is true. So what's the answer going to be? Both the statements are true. 1 and 2 are correct. So the answer is B. Easy question. Direct, simple theoretical question. Statements that we studied during studying the chapter. Isn't it? So let's take the discussion ahead. And the next question coming in front of you is this. Venturi meter works on the principle. What principle? Bernoulli's principle. The sum of the kinetic potential and the pressure energies for a flowing fluid remains the same. So the answer is Bernoulli's principle. Integral E dot ds is 0 for a surface. What it indicates? It indicates that for a given surface, if you say for any closed surface, what is this? What is this integral? What this integral indicates? It indicates closed integral, surface integral. So what it means for a surface integral, the number of field lines which are entering equals to the number of field lines that are going out of the surface. So the answer is going to be number of field lines entering the surface must be equal to the number of field lines leaving it. Answer is C. Okay. Next question in front of you is this. Let a wire be suspended from the ceiling and stretched by a weight W attached to the free end. What are they saying? They are saying that the question says that this is a wire. Okay, this wire is being suspended by a weight of how much? By a weight of W. This wire is being suspended by a weight of W. So what happens? If W is the weight if W is the force that the weight is applying, that the mass is applying, this mass to the wire, then equivalent amount of force is applied by the ceiling, by the top on this particular wire. So the situation for the wire looks something like this. The situation for the wire looks something like this. This is the force. That's equal to W. This is the force. That's equal to W. So what is the stress going to be? Stress is going to be force divided by the area. The restoring force is going to be developed throughout the structure, and that's going to be same. So the answer is going to be W by A, and the answer is C. And the answer is C. Okay, fine. Let's take the discussion ahead. Next question in front of you is this. Have a look at the question. What the question says: a bullet from a gun is fired on a rectangular wooden block with a velocity u. When the bullet travels 24 cm through the block along the length horizontally, velocity becomes u by 3. Let's say that this is the block. This is the block. Okay. Bullet enters from this point. It travels some distance and the velocity has been reduced from u to u by 3. And what is the distance being given to you? What is the distance being given to you? The distance has been given to you as how much? That's 24 centimeter. That means 0.24 meter. That's fine. Okay. So let's find out the retardation that's being offered by the block to this bullet. So we can say that V square equals U square plus 2AS. What is S? S is 0.24 meter. That's great. So let's find out the retardation. What's going to be the retardation? U by 3 minus U. Minus of 2U by 3. Minus of 2U square by 3. Okay. Divided by 2A. Yeah. Divided by 2A. This 2 gets cancelled out. and the acceleration is 0.24 meter. Write it like this only. It's going to get cancelled anyhow. It's going to get cancelled. Write it this way only. Cancel this too. So this is the expression for acceleration. After this, what happens? After this, the bullet travels certain amount of distance and it comes to rest. The bullet travels some distance and the final velocity becomes 0. So that's what they are asking you. That's what they are asking you. That what is the total length of the block? So let's call it as x. So you can say again v square minus u square. V square equals to u square plus 2as. Final velocity 0. Final velocity 0. Have a look over here. Ankita is saying ये अंग्रेज लोग पढ़ते हैं. यहाँ अंग्रेज लोग पढ़ते हैं. Anyways, it's English channel, so we have to speak in English. What can we do? Okay, no other option. You just focus over here. So what is this? This is v square equal to u square plus 2as. This is 0 equal to u square. That is u square by just a second. U by 3. So v square is u square by 9. I'm really sorry. This is u square by 9. U square by 9. Really sorry. So this is going to be v square equal to u square plus 2as. V square is how much? V square is u by 3. That means u square by 9. So this turns out to be how much? This is minus of, subtract them, 8u square by 9. Okay. Divided by 2 into 0.24. This is the value of a. So this is 4u square by 9 into this particular factor. So what are you going to get? You're going to get v square equals to u square by 9 minus 2 into a. A is how much? A is how much? A is 4u square divided by 9 into 0.24 times of s. So let's find out the value of s from here. Straightforward, this u square gets cancelled, 9 gets cancelled. So you're going to get s equals to 0.24 divided by 8, that is 0.03 meter. This is x. This is the additional distance that the body has traveled. This is x. So the total distance, x is how much? 0.03 meter. Okay, so the total distance is 0.24 plus 0.03. That means 0.027. That means 0.027. Okay, so how much it is? This is 27 centimeter. 27 centimeter. Okay, 0.027 meter. That means 27 centimeter. Next question in front of you is this. Read the question, what it says. A horizontal bridge is built across a river. The student standing on the bridge throws a small ball vertically up. Let's say this is a bridge and this is you standing over here. This is a bridge and yes, this is a bridge and this is you standing over here. On the bridge, what you have done? You have just thrown a ball upward. With what velocity? With some velocity of 4 meter per second. So let's say that your initial velocity is upward. So what the ball is going to do? The ball is going to get up and then come down somewhat like this. The ball is going to rise up and then fall finally down. You need to find out the height of the tower. You need to find out the height of the tower. Ball strikes after 4 seconds. The height of the bridge above the water surface is going to be what? So what can we say? Let's say that s equal to ut plus half a t square. Okay, you need to find out s. So s is ut plus half a t square. Substitute the data with proper sign conventions. U is upward, while the acceleration due to gravity, the net displacement, everything is downward. So let's take u as negative. Let's take u as negative and rest everything is positive. So what do we get? What do we get? We say s, that's downward. That's what we need to find. U is minus of 4 into time. How much time has been given? 4 plus half into, let's take g as 10. It has been given. T square is how much? T square is 16. So this turns out to be 8 into, this is this is 8 into 10, 80 minus 16, that's going to be how much? That's 64 meter. Another simple question, another simple answer. Is this fine? Are we done with this, all of you? Answer is D. Let's take the discussion ahead. Another question in front of you is this. This is from current, sorry, this is from magnetic effects of current. Yeah, magnetic effects of current. What the question says, a very long conducting wire is bent in a semicircular shape from A to B. Okay, that's fine. So the current in this semi-infinite wire has been given to you. Current in this semi-infinite wire has also been given to you. This is the direction of current. This is the direction of current. This is another segment. This is another segment. Semicircular. Semi-infinite, semi-infinite, semicircular. Okay, fine. That's cool. So let's go and straight over here, stand over here at this point. Tell me first of all the directionalities. Tell me what are the directions of magnetic field going to be. If this is the element number 1, if this semi-infinite wire, I call it as 2, semicircular, I call it as 3. So the direction due to this semi-infinite wire, hold this wire like this, it's going to be inside. It's going to be inside the plane. So the magnetic field due to 1 is in. Hold this wire this way, the magnetic field due to 3 is also in, and the magnetic field due to this semicircular wire is out. What it is out. So I'm writing that, see, I'm giving you a very simple method. Directly you would get the magnitude as well as direction. A very OP technique I'm giving you. A very awesome technique I'm giving you. Write down the magnetic field inward. Write down the magnetic field outward. Inward versus outward. How much is the inward magnetic field? Inward magnetic field is B1 plus B3. And both are equal. So directly I am writing 2 times of what is the magnetic field for semi-infinite wire. Magnetic field from semi-infinite wire is mu naught I divided by 4 pi r. Yes or no? Mu naught I divided by 4 pi r. Okay, fine. So what are we writing? We are writing this is mu naught I divided by 4 pi r. Okay, so this is mu naught I divided by 2 pi r. Net, this is mu naught I divided by 2 pi r. Great. What is the magnetic field outside? B out. That's going to be mu naught I divided by, this is how much? Mu naught I by 2r is for the complete wire. Yes or no? So what it's going to be? This is going to be mu naught I by 4r. So who is greater? Who is greater? Have a look over here. This is nothing but mu naught I divided by 2 pi r. And this is mu naught I, and this is mu naught I divided by 4r. So 2 into pi is 2 into 3.14. That means 6 point something. That means 6 point something, isn't it? Or let's write it as, let's write this expression as 2 times of mu naught I divided by 4r only. Let's write it this only. So clearly you can see that the magnetic field outside is greater than the magnetic field inside. So the direction is going to be away from the page. That means out of the plane. So into gets cancelled out. Net magnetic field is going to be magnetic field out minus magnetic field in. So I am writing it as magnetic field out, that is mu naught I divided by 4r minus magnetic field in, that is 2 times of mu naught I divided by 4 pi r. So I am taking this term mu naught I 4r as common. Mu naught I 4r 4r as common. So you take mu naught I by 4r common, you get 1 minus 1 by 2 pi. 1 minus 1 by 2 pi. That means the answer is D. It was a nice question, nice concept. Okay. So you got this answer correct. If you got it correct, congratulations, and let's take the discussion ahead. Have a sip of water. I'm also having a sip of water. You also drink. My water is finished. No worries. Let's solve the questions. I'll bring the water later on. Read the question. What the question says: net impedance of the circuit. Net impedance of the circuit. What is the expression for impedance? Impedance is root over R square plus XL minus XC the whole square. So R has been given to you as 10 ohm. XL is omega L, that has been given to you. Omega is 2 pi f. 2 pi f, this is f. Okay, this is f, frequency. So 2 pi f into L. L is how much? L is 50 divided by pi, and unit is millihenry. So this is 10 to the power minus 3 Henry. That's great. So what are you left with? Pi gets cancelled out. So this is 2 into 50, 100. 100 into 50, 1000. 100 into 50, 10,000 minus 1000. This is 10. This is 10. This expression is 10. This is 10. Okay. And let's find out XC. XC. XC is 1 by omega C. Omega is 2 pi f into C is, how much is C? C is 10 to the power 3 divided by pi. Microfarad. Micro is 10 to the power minus 6. It gets up. This gets 10 to the power 6. Okay. So pi gets cancelled out. This is 100. 100. This is 10 to the power 5. Oh, sorry. This is 5. This is 10 and this is 5. I'm really sorry. This is 5. And this is 100. This is 10 to the power 3. So 10 to the power 3 divided by 10 to the power 2. You are left with 10. So XC is 10 and XL is 5. So this is root over. This is root over R square. R square plus XL minus XC the whole square. So 100 plus 5. That means 100 plus 5 square. That's 25. Root 125 is 5 root 5. So the answer is 5 root 5 ohm. Answer is 5 root 5 ohm. Okay, that's fine. Let's take the discussion ahead. The next question in front of you. Just read the question, what it says. Radius of innermost orbit of the hydrogen atom is 5.3 into 10 to the power minus 11 meter. R equals to 0.529 or simply 0.53 times of n square divided by z. Isn't it? So multiply by n square. They are asking for the third orbit. Third orbit. So what is going to be R dash? That's going to be 9 times of 0.53. Isn't it? 9 times of, yeah, 0.53 into 10 to the power. This is going to be 0.53 angstrom, na? Angstrom. I'm giving the answer in angstrom. So how much it's going to be? This is going to be nearly 4.77. Nearly, see, if this would have been 10, this would have been 5. So this is lesser than 5. This is nearly 4.77 angstrom. You don't even have to calculate smart calculations. You're going to mark the answers in seconds. Okay. Yeah. So here we go. The next question in front of you is this. Read the question. This was a nice question. This was a nice question. How many of you got this correct in the chat? In the chat, all of you, how many of you got this question actually as correct? Because I really found this a decent question, a nice question. So what the question says that a satellite is orbiting above the surface of Earth. Okay. Just above the surface of Earth. Just above the surface of Earth. This is Earth. This is Earth. Okay. This is Earth. And this is the satellite that's orbiting very close to the surface of the Earth. That means the radius and the separation between the Earth and the satellite can be taken nearly as same. nearly as same. So we know that the centripetal force is provided by the gravitational force of attraction. And what is the centripetal force? The centripetal force is m r omega square. That's g m divided by r square. We have taken the radius of Earth and the distance of the satellite from the center of Earth as same. Why? Because of this word just above the surface. Okay. Omega is how much? Omega is gm by r cube to the power 1 by 2. This is the expression for omega. This is the expression for omega. Time period is 2 pi by omega. Time period is 2 pi by omega. I am substituting omega over here. I am substituting omega over here. So the expression looks something like this. This is the expression for time period. This is the expression for time period. They are talking about a particular quantity, that's 3 pi divided by gd. So what is going to be d? D is the density. What is going to be d? D is the density. D is the density. And what is density? Density is mass divided by the volume. Density is mass divided by the volume. Density is mass divided by the volume. Volume is 4 by 3 pi r cube. Yes or no? So see, what the logic says. The logic says that let's eliminate the root from here. If I eliminate the root, I get t square equals to 4 pi square into r cube divided by gm. This is the expression for t square. So now I need to find out the expression that relates 3 pi gd. So let's multiply these both expressions. Or let's divide these expressions. Okay. Let's divide these expressions. Let's multiply these expressions. That makes no difference. Okay. You can get the expression. You can get the answer. Okay. Or let's substitute the value of r cube from here to here. Whatever you feel comfortable. Whatever you feel comfortable. Find out the value of r cube. Keep it over here. Straightforward what I am going to do. I am going to multiply t square with d. I am going to multiply t square with d both the sides. I am multiplying density both the sides. So this is t square and I have multiplied d equals to 4 pi square into r cube divided by gm and d multiplied, that's m divided by 4 by 3 pi r cube. So what are we left with? What are we left with? M gets cancelled out, 4 gets cancelled out, pi gets cancelled out, r cube gets cancelled out. So t square equals to pi into 3 divided by d into g. So 3 pi by gd is nothing but t square. So the answer is t square. T square is the answer. It was a nice question. I said that it was a nice question. So tell me now how many of you got this as correct. Let's take the discussion ahead. Here we go. The next question. Yeah, this was a very simple question. What this question says, it's relating the temperature coefficient of resistances. So we know that if this is a resistance, you raise the temperature. So if you raise the temperature, the resistance increases, and the law says that delta R by R equals to alpha delta T. So we are writing the expression. Resistance of platinum wire at 0 degree centigrade is 2 ohm and 6.8 ohm. Yeah, 6.8 ohm at 80 degree centigrade. So the first expression corresponding to this is delta R. Just a second. Resistance of platinum wire at 0 degree is 2, and at this is the temperature coefficient. Yeah, so let's compare this. Okay, so delta R, that means 6.8 minus 2, that means 4.8 divided by the temperature initial, that's 2, equals to alpha into delta T. Delta T is 0. The difference of 0 and 80. 80 minus 0, that's 80. So what are you left with? You are left with 2.4 divided by 80. Okay, so this is how much? This is 24 by 100. So 3 divided by 100. So you are left with 3 into 10 to the power minus 2 per degree centigrade. 3 into 10 to the power minus 2 per degree centigrade. That means 0.02. How many of you got this correct? How many of you got the same answer? Yeah, there is some misprinting. I got confused because of this dot. ये जो dot है ना, I got confused because of this dot. This was the misprinting over here. Okay, actually it was a full stop, but yeah, I got a bit confused of this. It should have been a comma over here, like this. Okay. Yeah, so is this point very clear? Got the answer as correct? See, every single question is from the notes. Every single question, Yakin English, I taught in Yakin English, and I bet Yakin English, every single question out of 50, I have calculated 48 questions out of 50 are from your notes and are from the KKF that I have provided you. So out of 48, you have to attempt only 45 questions. Anyways, the next question, calculate the maximum acceleration of a moving car so that the boy lying on the floor of the car remains stationary. This is the floor of the car. Okay, let's say this is the floor of the car. And this is a boy which is lying. Let's say this is a boy. Doesn't look like a boy, but let's say this is a boy. Okay, so what is happening? This car is speeding. This car is accelerating. So if this car speeds up, the boy has a tendency to slip backward. So if the boy is slipping backward, okay, there is going to be a frictional force along this direction. So we are going to say that the frictional force must be, equal to the force applied if what? If the body is stationary. So I wrote something wrong, backward. Yakin English. You won't say, yeah, that's fine. Sahi to likha hai. Yeah, so F equals to M times of A. What is frictional force? Maximum frictional force, that's going to be mu times of N. N is mg equals to M times of A. So this M gets cancelled out. This M gets cancelled out. A equals to mu times of G. Mu is how much? Mu is 0.15 into 10. 1.5 meter per second square is the answer. Okay. Let's take the discussion ahead. Another question. Simple question. 10 resistors. Each of resistance R are connected in series. 10 resistors. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. Connected in series. So what to do? 10 resistances connected in series. So what is the R equivalent? That's going to be 10 times of R. Okay. And let's say the current in the first situation is I. The current in the first situation is I. So I equals to E divided by 10R. Okay, that's fine. In the next situation, they are connected in parallel to the same battery. They are connected in parallel through the same battery. Okay. They are connected in parallel through the same battery. R through the same battery. So what is going to be the R equivalent in this situation? R equivalent is going to be R by N. Okay, that's fine. They are in parallel, na? R equivalent is R by N. So what is the current? The current increases by N times. So I dash is going to be N times of I. I dash is going to be N times of I. So I is what? I is V. divided by R equivalent. R equivalent is what? R equivalent is R by N. Okay. This is N times of I. I is what? I is what? I is E by R. E by R. So N, so what we need to find? We need to find out the value of N. Yeah. So what is N from this equation? From here, what is N? N is nothing but R divided by R equivalent. Okay. Just a second. Let's write it this way. Let's write it this way. Don't get confused. Let's write it this way. You have written I equals to E divided by 10R. I equals to V by R in the first situation. This is the first situation. Fine. In the second situation, I dash is simply N times of I. It is N times of I. And what is I? What is I? It's E divided by 10R. Okay. So what is I dash? What is I dash? I dash is E divided by R dash. What is R dash? R dash is N times of R. That means 10 times of R. So how much is the value of N from here? How much is the value of N? That's going to be 10 into 10. That means 100. That means 100. Substitute it over here. Substitute it over here. I dash E. Okay. $100. Let's come to the next question. Done. Next question in front of you is this. Read the question. In the figure shown, what is the equivalent focal length? Now have a look at the situation. There are 3 lenses. 1, 2, and the third one. Lens number 1, lens number 2, and the lens number 3. Lens number 1, lens number 2, and the lens number 3. Yes or no? So you know that the equivalent focal length is going to be 1 upon f1 plus 1 upon f2 plus 1 upon f3, and you know that f1 and f3 are going to be same. So let's individually find out the values of f1, f2 and f3. f1, mu2 by mu1 minus 1. Mu2, that means mu of lens. I'm writing the formula over here. I'm writing the formula over here and substituting the values directly. Mu2 by mu1 minus 1, 1 by r1 minus 1 by r2. Okay. So this is going to be mu2. That means mu of lens 1.6 by 1 minus 1 bracket 1 by r1 minus 1 by r2. 1 by r1 minus 1 by r2. What is it going to be? This is going to be minus 1 by 20. Okay. So what are we left with? We are left with f1 equals to minus of 20 divided by 0.6. 0.6 is 6 by 100. That means minus of 100 by 3. Minus of 100 by 3. This is f1. You have found f1. You have found f3. Both are same. The expression for f2 is going to be 1 by f equal to mu2 by mu1. That means 1.5 minus 1. That is 0.5. Bracket 1 by r1. This is plus 20. Minus 1 by r2. That means minus 20. So this is 2 by 20. 1 by 10. So f2 is what? f2 is 10 by 0.5. 10 by 0.5. That means this is going to be 20. This is going to be 20. Okay. You need to find out the net focal length. You need to find out the net focal length. So what it's going to be? f1 plus f3. That's going to be same. That means 2 times of. That means fnet 2 times of minus 100 by 3 plus 20. What I said? 2 times of 100 by 3 plus 20. So what are we left with? What are we left with? Minus 200. This is minus 200 plus 3 2s are 60 divided by 3. Just a second. Just a second. Just a second. This is 2 by 10. 2 by 20. That means 1 by 10. Reciprocate it. Divided by 0.5. This is 20. That's fine. This is for this lens. Oh, I'm really sorry. I'm really sorry. I'm really sorry. We are doing a silly mistake over here. I'm really sorry. This is 1 upon f equals to 1 upon f1, 1 upon f1 plus 1 upon f3. That means 2 times of 1 by f1. Okay. That is minus 3 by 100 plus 1 by 20 plus minus of 3 by 100. Okay, so what are you left with? You are left with minus of 6 by 100 plus 1 by 20. So 1 by f is going to be 100. This is minus 6 plus 5. So f is going to be minus 100 centimeter. This is the answer in front of you. How many of you got this answer? How many of you got this answer? Got? Nice, 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 nice. Note it down. The answer is D. All of you note it down quickly. Nice question. Answer is D. The answer in front of you is D. In the chat, tell me how many of you got this question as correct. Okay, so the next question coming in front of you is this. What the question says: two thin lenses of same focal length. One is convex, another is concave. One is convex, another is concave. Are placed, the focal lengths are same. So you know that f1 you are going to write as f. f2 you are going to write as minus f. So 1 upon f equivalent equals to 1 upon f1 plus 1 upon f2. f1 is f, f2 is minus f. So this gets cancelled out. Answer is 1 upon f is 0. That means don't mark it as 0. The answer is infinity. Infinite. Answer is A. F is infinite. 1 by f is 0. Next question in front of you is this. This is a nice question. A simple and nice question. So the time period over here has been given to you as 8. Time period is 8. Yes or no? Omega is 2 pi by t. That means pi by 4. Okay, you need to find out what? You need to find out acceleration. Minus omega square into x. Minus omega square into a. What is a? This is the a, maximum displacement from the mean position. Omega square is minus pi square by 16 into a. What is a? A is 1. Answer is minus pi square by 16 meter per second square. This is the answer A. How many of you got this as A? Okay. The next question is this. Electric dipole placed as shown in the figure, minus q plus q. You need to find out the potential at this particular point. So go and stand over here at this point. You need to find out the potential. Let's call this as charge number 1. Let's call this as charge number 2. So V is going to be what? V is going to be what? V is going to be V1 plus V2. What is V1? Minus of K Q divided by R is 8 centimeter. R is 8 centimeter. So 8 into 10 to the power minus 2 meter plus K Q divided by R, that's 2 centimeter. Okay. Okay. Fine. So what are we left with? This is going to be, take out K Q 10 to the power minus 2. It gets up and becomes 10 to the power 2. So what are you left with? You are left with minus 1 by 8 plus 1 by 2. Isn't it? KQ by 5 plus 8, 5 plus 3, 5 minus 3. That's fine. So this is going to be KQ 10 to the power 2. This is 8. This is minus 4. Sorry. This is minus 1 plus 4. So this is 3 by 8 KQ into 10 to the power 2. 3 by 8 KQ into 10 to the power 2. This is the answer. This is the answer. That's what they are asking. The electric potential in 10 to the power 2 volt. Okay, that means multiplied by 10 to the power 2 volt. This is 3 by 8 KQ. So this is the answer in front of you. Let's take the discussion ahead. The next question in front of you is this. So this is from logic gates. NOT gate, NOT gate, NAND gate. Directly writing the outputs. Okay, let's take 0 and 0. So I'm making it over here A, B, and this is your final output Y. A, B, and your final output Y. So take 0, 0. What it makes to 0, this makes 1, this makes 1. 1 and 1 goes over here. If it would have been AND gate, the answer would have been 1. Since it's NAND gate, the answer is 0. So for 0, 0, what do you get? You get the answer as 0. Now take the next term as 0 and 1. This turns out to be 1. This turns out to be 0. So when 1 and 0 reaches the NAND gate, if it would have been AND gate, the answer would have been 0. 1 and 0, the answer would have been 0. Since it's NAND gate, the answer is going to be 1. So you have taken 0 and 1. If, I repeat, this becomes 1, this becomes 0, 1 and 0, the answer would have been for AND gate, A and B, 1, 0. The answer would have been 0. This makes the answer as 1. Another input, 1, 0, 0, 1. Take 0, take 1. AND gate makes 0. This makes 1. So the answer for 1, 0 is 1. Take 1, 1. Take 1, 1. Take 1, 1. Okay, what it makes? It makes 0, 0. Take 0, 0. For AND gate, the answer would have been 0. For NAND gate, the answer would be 1. So the only option that matches is 0, 1, 1, 1. 0, 0, 1. 0, 0, 0, 1, 1, 0, 1, 1. Yes, the answer is C. The answer is C. Yes or no? Yes or no? Tell me quickly. 3 is the answer. Sachin Kumar is saying national language क्या है सर मेरी जान इंग्लिश का चैनल है करना पड़ता है धंधा है काम है ठीक है. Okay, come over here. Okay, here we go. The next question in front of you is this. Read the question, what it says. A wire carrying a current I. A wire carrying a current I along the positive x-axis has a length L. It is kept in the magnetic field. This is the magnetic field in front of you. Magnitude of the magnetic force. You know. Goxder is saying it is not short dipole. I'll have a look at the short dipole. Don't worry. Goxder. That. Okay, don't worry, I'll give the answer. So yeah, what I'm saying, the wire is being kept somewhat like this. This is a current carrying wire. The current is along the positive x-axis, so this is I cap. Okay, the length is L, and the magnetic field has been given to you as 2I cap plus 3J cap minus 4K cap. Magnitude of magnetic force, you know that the expression for force is I L cross B. I L cross B. So what is the direction for the L? L is along the I cap because the current is along the x direction. So the unit vector along the x direction is I cap. Yes or no? Definitely yes. So what are we going to write? We are going to write that L cross B, I cap minus J cap, K cap. I cap minus J cap, K cap. What is I cap? I cap is 1, unit vector along the x direction, J and K. L is 0. L cross B. What is B? 2, 3 minus 4. Let's take the cross product. Let's take the cross product. I cap becomes 0. J cap term becomes minus j times of minus 4 minus 0. And the k cap term becomes 3 minus 0. That means this is going to be 4j plus 3k. 4j plus 3k. This is the expression for B. So the magnitude of B is going to be 5 root over 3 square plus 4 square. Okay, this is going to be 5. So the force is going to be I into L cross B. Sorry, this is L cross B. This is not B. This is L cross 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 B, and this is the magnitude of L cross B. Okay. So I into L cross B. L is a unit vector along the x direction, cross B. B is how much? B is 5. So the answer is 5IL. The answer is 5IL. Done, 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 done. Yeah. So now tell me what question you were talking about. Goxder, that it is not short dipole, it is in centimeter. Yeah, where are you? Where's Mr. Goxder? Which question, my dear? What question are you talking about? What question are you talking about? This one? Mr. Goxder. Are you talking about this question? Goxder? What question are you talking about, Mr. Goxder? Tell me quickly, are you there in the chat? I don't think he's there. Anyways, so students, first of all, what marks are you getting? Tell me quickly, what marks are you getting? How many correct you're getting? My favorite subject is physics and hero is social. That's good. That's good. Yeah. So anyways, this actually a very happy moment for me and for every PVN, because you just saw that out of 50, nearly 48 questions were exactly from the notes that we have discussed. Exactly from the notes that we have discussed, isn't it? So let's meet forward someday again. So I wish you all the best in your life, and we would be meeting soon. Okay, take care. Goodbye. Thank you. Have a great day ahead, and the chemistry discussion is about to come. Have great days ahead. Okay, chemistry discussion is about to come. Don't leave the streaming. I'm leaving. Chemistry sir would be joining soon. Take care. All the best. Keep shining. Keep rocking. Thank you. And namaskar from KK.
Hello everyone, how are you all doing? Hey guys, can you hear me? Can you all see me? If you all can hear me and if you can see me properly, just write in your comment box that you can see me and hear me properly, everyone. Okay, I'm just waiting for your response, everyone. Can you see me? Okay. Excellent, guys. Well, I'm hoping that you all are doing great. This is Sagar Darvi, your chemistry teacher, and I welcome you to Physics Wallah for the detailed discussion of NEET 2023 paper that you guys have written just few hours back. Well, guys, I'm also hoping and I'm sure that you guys must have had a wonderful day so far. And why not? For this 2 years, you all have been preparing, you all have been putting all the efforts that you could do, and finally it's the time to fetch what you all deserve in terms of your results. Great, guys. So we all are aware that today we are meeting here for the discussion of NEET 2023. So now we'll be starting with the remaining subject out of 3. So you already had a discussion of biology, followed by physics, and now we're going to start discussion of chemistry. To begin with, we'll be first discussing the organic chemistry part. So without wasting a single minute, guys, we are starting straight away with the discussion. And the first question right on your screen. Well, you all can see the question says, in Lawsonia extract of organic compounds, both nitrogen and sulfur are present. Which of the following complex gives blood red color with Fe+3 ion, that is ferric ions? Well, guys, I'm sure you people have already identified that this question is straight away from the qualitative analysis that you study in organic chemistry. Yes? Well, guys, so we know that qualitative analysis basically is the process where we identify the presence of element in an unknown organic compound. Yes, there are several tests. We identify the presence of carbon, hydrogen, nitrogen, sulfur, halogen, etc. Whereas in order to find out, or in order to see the presence of elements such as nitrogen, sulfur and halogen, we conduct a test called as Lawsonia test. So this is what the question is all about, that they are asking, in Lawsonia extract an organic compound where there is presence of both nitrogen and sulfur, which of the following complex gives blood red coloration with Fe3+ ion? Well, so whenever we carry out Lawsonia test, we prepare an extract, which is what you call it as Lawsonia extract, or sometimes it is also called as, in simple words, sodium fusion extract. So if there is a presence of nitrogen along with the carbon, and when we heat that with sodium metal, we get the formation of complex called as sodium cyanide. Whereas the presence of sulfur along with the sodium forms a compound called as sodium sulfide. And those two complexes, or the compounds, we further treat with some of the reagent. But when a compound, an unknown organic compound, contains nitrogen and sulfur together, then a sodium metal upon combination with carbon, nitrogen and sulfur results in the formation of a compound that is sodium thiocyanate, and this complex then further reacts with Fe3+ ions, which results in the formation of blood red coloration. So if you see, it was a very straightforward question, straight away informative. You can see here, I suppose this should have been NaCNS, that is sodium thiocyanate, which further upon reaction with Fe3+ ion results in the formation of blood red coloration. So guys, keeping a fact in mind that you guys have been attending sessions since a very long time, you discussed all the questions of biology followed by physics, and now I'm sure that you guys must be tired, so we are not taking much of a time and moving forward with the next question, number 2 from organic chemistry. Well, the question is from a chapter biomolecule. We come across a biomolecule called as nucleic acid. So this question straight away belongs to nucleic acid. The question says you have been given two statements. The first statement says a unit formed by the attachment of base to 1' position of a sugar is known as nucleoside. Well, guys, when you talk about nucleoside is a combination of sugar, which is a pentose sugar, we call it as ribose. Precisely speaking, the beta ionomer of ribose sugar along with base. So yes, first statement is absolutely true. You'll find that this base is attached to a sugar at 1' position. So this is absolutely correct statement. A unit formed by attachment of base to 1' position of sugar is known as nucleoside. So statement A is absolutely correct. Now let's have a look at the statement number 2. Statement number 2 says when a nucleoside is linked to phosphorus acid at 5' position of a sugar moiety, we get a formation of a nucleotide. Well guys, let me tell you, nucleotide is a combination of 3 units together. Two, out of which 2 we have already mentioned over here, which is sugar plus base. Now base is connected to sugar at 1' position as mentioned in statement A, whereas in the same sugar at 5' position there has to be attachment of phosphoric acid and not phosphorus acid. But if you look at statement 2 says there is attachment of phosphorus acid, which is not correct. It has to be a phosphoric acid, which means statement 2 is not a correct statement. So now we just have to look at these 4 options, and whichever option states that statement A is correct, but statement 2, that is number 2, is incorrect, is going to be our correct answers. So guys, which option says so? Statement A is correct statement. Statement A has to be correct, but statement 2 is incorrect. So statement 2 is false. So among all the options, it is option number 4, which is an appropriate option. Because in order to form a nucleotide is basically a monomer of nucleic acid. So nucleotide contains a base, which is a pentose sugar. It contains sugar, which is a ribose sugar, which is further attached to a base at 1' position and phosphoric acid at 5', not phosphorus acid. So everyone, the right choice is answer option 4. Shall we proceed then, everyone? Yes? Excellent. Guys, the next question says, consider the following reaction and identify the product in the given question. Well, if you observe the substrate which is given to us, it is some alcohol, which is then treated with HBr, and they are asking us to find out product P. Well, HBr is a halo acid, which is a proton donor. So this will dissociate to give you H plus ion. In a given substrate, since we have alcoholic OH, where oxygen being electron rich, it acts like a nucleophile. So the step number 1 of reaction between HBr with an alcohol is the protonation of alcoholic OH. So the lone pair of oxygen will attack at H plus, and that will be further converted to OH2 plus. Now we all have studied that OH alone is not a good living group, but OH2 plus, that is water, is a good living group. So in the next step, what happens is there is departure of water as a living group. So if water leaves, you will see this carbon, which is a secondary carbon atom, will now be converted to its corresponding carbocation upon loss of bond pair between carbon and oxygen. Guys, if that happens, what is going to be our next carbocation? Well, I'll write it down. It will be CH3, CH, which is further connected to CH3, and there we get the secondary carbocation CH plus, which is in turn further connected to CH3. Well, now when we studied general organic chemistry, we have learned that whenever there is a formation of carbocation, the first thought that should come in our mind is that carbocation the stable one, or is that carbocation not stable but has the ability to rearrange itself to form a stable carbocation. So here, if you observe carefully, the existing carbocation is secondary, adjacent carbon being tertiary. There is a scope of rearrangement via hydride shift. So H minus will shift to this carbocation, which will in turn convert this carbocation into even more stable carbocation, which is a tertiary carbocation. So you will see there is a formation of tertiary carbocation CH2. This becomes after hydride shift, and we got formation of tertiary carbocation. Once we get formation of tertiary carbocation, now this is the stable one. Once we get formation of stable carbocation, the next step is straight away attack of Br minus, that is nucleophile at this carbon. Attack of Br minus at this carbocation will result in the formation of a product, which will be 2-bromo 2-methyl butane. 2-bromo 2-methyl butane. So let us observe, where do we see the same product? Isn't that the option 2 here? Yes, it is, everyone. Option 2, 2-bromo 2-methyl butane. So the question was from chapter alkyl halide, which involved, or you can say from the chapter of alcohol, which involves formation of carbocation, followed by rearrangement via hydride shift, and then the attack of a nucleophile. Right, everyone? Excellent. Guys, next question. Next question on your screen. It is an informative question from a chapter polymer which asks, which among the following molecules upon polymerization produces neoprene? Well, what is neoprene? In the chapter polymer, after classification, we study several types of polymers. Addition polymer, condensation polymer, biodegradable polymer, right? So when you talk about addition polymer, we come across several addition polymer, one of which the important category is rubber. So we further study rubbers are of two types, natural rubber and synthetic rubber. When you talk about natural rubber, we talk about cis polyisoprene and trans polyisoprene, which we also call it as gatta parcha. And then there is a synthetic rubber. If you talk about the basic example of synthetic rubber, we talk about neoprene, we talk about Buna S and Buna N, that is SBR and NBR. Now, when we talk about this synthetic rubber, neoprene, this neoprene is also known as polychloroprene, which indicates that neoprene is made up of polymerization of many chloroprene units, and chloroprene is nothing but 2-chloro 1,3 butadiene. So when they're asking that neoprene rubber is made up of polymerization of which of the following monomer, we need to understand that neoprene is made up of polymerization of chloroprene, and chloroprene is 2-chloro 1,3 butadiene. Where do we see 2-chloro? Well, guys, 1, 2, 3, and 4. Here is 2-chloro 1,3 butadiene, and this is what we also call it as chloroprene. Chloroprene is used as monomer in the preparation of formation of a synthetic rubber called as neoprene. Hence, the right choice is answer option C. Perfect. Yes, I can see, guys. Wonderful job. I can see in the comment section that many of you have already identified the right choice. Excellent. Well, guys, next question for you all on your screen. Identify the product in the following reaction. Well, I wouldn't say it was difficult question, but a little tricky. I have seen some of the students have committed some silly mistake over here. Let us see what was that. Well, the starting compound given to us is benzene diazonium chloride salt, benzene N2 plus Cl minus. If you see, they are saying we have to treat this further with Cu2Br2HBr. So it is nothing but, guys, if you all remember, benzene diazonium halide salt shows two types of reaction. Type 1 reaction where there is a replacement of N2, and type 2 reaction where there is retention of N2, like a coupling reaction. Whereas the first part over here is talking about the Sandmeyer reaction. Sandmeyer reactions involves replacement of N2 with the halogen which is taken in the form of salt of a copper. So Cu2Br2, in which bromine will substitute this N2. So we say the first compound, or you can say intermediate obtained, would be this N2 will be released, instead of which will be getting formation of bromobenzene. So first condition is resulting in the formation of bromobenzene. The second condition that they're saying whatever product we are getting, treat that with Mg in presence of dry ether. Well, guys, the substrate now we have is haloarenes. We are treating that with magnesium metal. So reaction of alkyl and aryl halides with the magnesium metal in presence of dry ether. Generally we use diethyl ether, that results in the formation of a very famous reagent that we use in the preparation of several other organic compounds. And that compound is nothing but a Grignard reagent. So bromobenzene upon reaction with magnesium metal will result in the formation of phenyl magnesium bromide. Phenyl, well, as of now, just for the sake of representation, I'm showing bond pair over here. Phenyl magnesium bromide. However, we know that across this carbon and magnesium metal, there is going to be more of ionic character, as a result of which we always consider this bond pair in favor of phenyl ring, existing this in the form of pH minus and MgBr plus. Now, in general, when you talk about any Grignard reagent with whatever value of R in this R mgx, we always consider this as R plus and mgx, R minus, I'm sorry, R minus mgx plus. This R minus being a conjugate base of hydrocarbons such as alkane or alkene or even benzene. Because all alkane, alkene and benzene being a weaker acids, their conjugate bases are always a strong base. So guys, since this pH minus C6H5H, C6H5 minus being stronger conjugate base, whenever you allow it to react with anything that is protic, all strong conjugate base will have a tendency to abstract a proton immediately. Hence, when this phenyl magnesium bromide is further hydrolyzed, meaning when it is brought in contact with water, we know the fact that water is a polar protic solvent, always ready to donate a proton. So this pH minus will attack at H plus, and that attack takes place, everyone, phenyl ring upon accepting proton will straight away result in the formation of an aromatic compound, which is nothing but benzene. So everyone, if you just check all the options given to you, you will find that option 3 is the most appropriate option because it is saying the product is benzene. So we were given benzene diazoxide chloride salt followed by the Sandmeyer reaction forming bromobenzene. Upon reaction with magnesium metal in presence of dry ether gave us phenyl magnesium bromide, and that further upon reaction with water gave us benzene. Alright everyone, very well. Next question for you all on your screen. Complete the following reaction. Cyclohexanone is given. We are treating that with SCN. We all know the fact. We studied this reaction under the heading of nucleophilic addition reactions of carbonyl compound, which we call it as characteristics reactions of aldehydes and ketones. Generally, such reaction, especially reaction of HCN, we carry out in presence of an alkaline condition. So, let's say if I dissociate this as H plus and Cn minus, cyanide being nucleophilic in nature, and across carbon and oxygen, oxygen being more electronegative, will acquire delta minus, leaving delta plus charge on carbon. Delta plus charge on carbon indicate that it is electrophilic center and hence invite attack from a nucleophilic species. So here in SCN, what is nucleophile, dear students? Yes, cyanide is our nucleophile. So let's say cyanide attacked here. So carbon will become pentavalent. So pi bond will break towards O, making it O minus, which will then attack at H plus. So see, this is going to result in the formation of an intermediate compound called as cyanohydrine. So I'll call this product as cyanohydrine of cyclohexanone. Now this cyanohydrine of cyclohexanone is further treated with concentrated H2SO4, and the important condition mentioned is heat. Guys, we know the fact concentrated sulfuric acid acts like a dehydrating agent, and especially when that reagent is heated with an alcohol, it undergoes dehydration. So, ideally speaking, if we have got OH, carbon containing functional group is alpha, then I have got two adjacent carbon, beta. Now both being equivalent, I am highlighting one of the H. And this is being dehydrating agent, it is going to carry out dehydration of OH and adjacent beta carbon. So this sigma bond will shift here, resulting in the formation of double bond between alpha and beta carbon. Now ideally speaking, the product should have been cyanohydrine, cyanide at the carbon of cyclohexene. But when you look at all the options, we don't really see anywhere there is a cyanide, which means we have to assume that we are carrying out acid hydrolysis of cyanide. Although no water mentioned over here, looking at all the options given, I'll consider that this step is a hydrolysis of cyanide in an acidic condition. So one of the very important conversion, I'm sure every teacher must have told you, cyanide upon acid hydrolysis converts into carboxylic acid. That's what we will consider. Cyanide upon acidic hydrolysis will get converted to carboxylic acid. So the cyanide will be now converted to another functional group, which is carboxylic acid. But we know the fact there is unsaturation here. We will be getting COOH. Cyclohexene carboxylic acid is your product. So everyone, which option states so? Here it is, answer option 1. So although hydrolysis, water is not mentioned over here, Looking at all the options, we'll consider this condition as acid hydrolysis of cyanide. All right, everyone? Yes, perfect. Great, guys. Moving forward to the next question. Some tranquilizers are listed below. Which one of the following belongs to barbutirates? We all know the fact that barbutirates are the derivative of barbutyric acid. And if you all remember, if you have followed all our previous series of NEET Selection or Yakin or any other batches, I have been telling you guys repeatedly, never underestimate those small chapter which comes after nitrogen containing compounds. So be it biomolecule, polymer, environmental chemistry, or even chemistry in everyday life, you will find at least 2 questions from the set of those 3 to 4 chapters. See, so far as many questions we have solved. We got a question from biomolecules, we got a question from polymer. Now we have got question from, yes, chemistry in everyday life. You will find one more question from environmental chemistry as we proceed further. So here, this question is from chemistry in everyday life, and they're asking which of the following is tranquilizer. Well, when we studied this chapter, and in the chapter when we studied different types of class of compounds or the medicines, we come across a compound called as tranquilizers, the type of drugs. In tranquilizer, when we studied the derivatives of barbutyric acid, I had told you a simple trick to remember all the derivatives of barbutyric acid by the word called vanillas. Vanillas. Now, what is that vanillas, everyone? Well, the trick was V stand for veronal, which is our right choice. V stand for veronal. A stand for amityl. N stand for nembutyl. I, we don't consider anything. L for luminal. A, we have already seen amityl, and S stand for sequinal. So basically, it was a simple trick for us to remember all the barbutyric acids. So, veronal, amityl, nembutyl, luminal, and sequinal. I and this A, you can ignore it. So among all the options which we studied or we remembered, one of the question was straight away from the name, which is here, veronal. So veronal is a tranquilizer, belongs to category of derivative of barbutyric acid. So we call it as barbutirates. So everyone, informative question for you all. Answer option A, or 1 you can call it. Well, another important chapter, carbonyl compounds, or we call it as aldehydes and ketones. This question straight away from aldehydes and ketones. And what are they asking? They have given you a compound where you can see there are 2 ketone group. Upon arrow condition mentioned is zinc amalgamine concentrated HCl. Guys, can you recall what reagent is that? Can you write in the comment section, everyone, what reagent is that? Come on. Well, zinc amalgamine concentrated HCl is the reducing agent known as Clemenson's reducing agent. So basically what we're going to carry out here is Clemenson's reduction. Clemenson's reduction uses a reagent which is an acidic reagent, and that is useful for the conversion of carbonyl carbon atom into hydrocarbon, or you can say methylene group. The same conversion can also be carried out using Wolff-Kishner reagent. The only difference lies in the condition. When you talk about Clemenson's reagent or Clemenson's reducing agent, to carry out this conversion, we use acidic condition. Hence, if our substrate is acid sensitive, we do not use this. Whereas for the conversion of carbonyl carbon into CH2, we can also use Wolff-Kishner, but that use alkaline condition. So if your substrate is base sensitive, we don't really use Wolff-Kishner reducing agent. But now straight away, they are saying that you have been given Clemenson's reducing agent, what will be product? Like we said, carbonyl carbons will be reduced to hydrocarbons. So if you observe, this C double bond O should be converted to CH2. I can see that here. And even this C double bond O should be converted to CH2, which I can see over here. So keeping all the options in mind, I can say the answer is option 2. Isn't it, everyone? Yes, perfect. I can see many of you have been participating in this paper solving. Guys, I'm happy to see all that, and I'm sure you guys have done that well in your actual exam as well. Superb, guys. You all are rock stars. Come on, the next question for you. The question says the weight of 2 moles. The weight of 2 moles of the organic compounds which is obtained by heating sodium ethanoate with sodium hydroxide in presence of calcium oxide. Well, I have been always telling you that the thumb rule in organic chemistry is whatever is given to you in the form of a sentence or a statement, put it down in the form of reaction. Many a time you'll realize that when you try to do or solve some reaction in an air, probably you get stuck somewhere. But if you write it down, so many things you will be able to recall, because in most of the cases, reactions we remember by visuals. Let's do that here as well. So what are they asking us? They are saying 2 moles of organic compounds. 2 moles of organic compounds which is obtained by heating sodium ethanoate. Okay. Ethanoate is a conjugate base of ethanoic acid. Ethanoic acid is 2-carbon carboxylic acid, meaning they're talking about sodium salt of acetic acid. That's sodium ethanoate. Yeah. You have to treat that with sodium hydroxide. All right. So I'm heating that with sodium hydroxide in presence of CaO. I'm sure you all are aware that this mixture collectively we call it as soda lime. So I'm not discussing mechanism right now. That is not really important for us as far as the paper solving is concerned. So I'm directly going to write down the product by keeping a simple trick in our mind, which every teacher must have taught you, that soda lime upon heating with sodium salt of carboxylic acid undergoes decarboxylation, where we eliminate sodium carbonate. Na2CO3, if I eliminate, this H will be directly associated to this methyl group, which will hence result in the formation of methane. If you look at this methane contains 1 carbon atom, atomic mass 12, 4 hydrogen, atomic mass 4, so 12 plus 4, 16 units. But that 16 belongs to 1 methane. In the question they are saying the weight of 2 moles of product obtained of this reaction. So 2 moles of methane, 2 moles of methane means the mass they are talking about, or the molar mass they are talking about, is 32 units. So where do you see the option? Yes, here it is, answer option 3, 32 units. Yes, everyone? Great, great. Well, it's going to be 32, not 18. If anyone is asking in the comment section, guys, it's 32 because they're talking about 2 moles. Molar mass of 1 mole of methane is 16. For 2, it's going to be 32. That's how the option is 3rd. All right, next question. Again, an informative question from haloalkanes and haloarenes. They have given us some structure and they're asking us to find out the given molecule is example of what kind of alkyl or aryl halide. Well, when you talk about any carbon atom of the benzene, we call it as phenylic carbon or phenylic carbon. The carbon which is outside the ring but directly connected to benzene, this carbon we call it as benzylic carbon. But right now we don't have to look at any of those carbon because the question is with respect to what halide is that. So the whole idea is look at the carbon to which halogen is connected. So halogen is connected to this particular carbon atom. This carbon itself is an sp3 hybridized carbon, in turn connected to sp2 hybridized carbon. Any such sp3 hybridized carbon which is directly connected to sp2 hybridized carbon, dear students, we call it as allylic carbon atom. We call it as allylic carbon atom. Further, the degree of allylic carbon may vary depending upon that carbon is directly connected to how many other carbon. For example, here, this allylic carbon is further connected to 2 other carbons, so we call it as secondary allylic carbon atom. So when a halogen is connected to allylic carbon, we call it as allyl halide. So looking at all the options available to us, if you see option number 4, it perfectly says it is allylic halide. No other option says it is allylic. So for the given structure, halogen being connected to sp3 hybridized carbon, which is further connected to sp2 hybridized carbon, we call it as allylic carbon. Precisely, it is secondary allylic. Hence, our answer option 4 is perfect answer, allylic halide. Yes, everyone? Are we all getting this? Yes, perfect. Yes. Superb. Next question, the number of sigma bonds, pi bonds and lone pair of electrons in a pyridine respectively are. Guys, you can't have question easier than this. Pyridine, such a common structure. We have been using this molecule as a base in so many reactions. Wherever see excess of acid is obtained as a byproduct, in order to neutralize that acid, we take a help of pyridine. In fact, When you studied reactions such as Darzan's process, you use pyridine. So you took alcohol plus thionyl chloride in presence of pyridine. You get the formation of alkyl chloride with the product of inversion of configuration. Which means, I'm just trying to tell you that this reagent we have used it several times earlier. So how does it look like? Well, pyridine looks like this. It's a 6-member cyclic heteroaromatic compound. Heteroaromatic compound. Now they're asking in this molecule how many are sigma bonds, pi bonds and lone pair. Well guys, now what happens is, because we have a tendency of writing structure of aromatic ring just like that by putting the benzene ring, we often forget that every carbon is tetravalent, so the fourth bond has to be satisfied by the hydrogen atom. And because we never practice of writing this hydrogen atom, we forget that there are going to be additional sigma bond because of the presence of hydrogen atom over here. Now, let's count the number of sigma bonds. So where do you see sigma bonds? Well, with the pink color, I'm writing sigma bonds 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11. 11 sigma bond. When I see that, I can easily rule out option A and 1 and 3, which means it has to be 2 and 4. Now guys, we know that the third option they're asking about is lone pair. I know the fact nitrogen carries 1 lone pair, meaning the third has to be 1, meaning I can also rule out option 2. But just to verify, you can see that again, we have got 11 sigma bonds, pi bond. Yes, we have got 1, 2 and 3 pi bonds. 3 pi bonds and 1 lone pair, which is right here. What is the correct option? Answer option 4. Yes. Superb. Guys, next question on your screen. Which of the following reaction will not give formation of primary amines? And the first reaction itself is an interesting reaction, because several times we have been using lithium aluminium hydride as our reducing agent. And in most of the cases, when we use lithium aluminium hydride as a reducing agent, generally the substrate that we use are either aldehydes, ketone, acid, chlorides, carboxylic acid or alcohols or esters. And in all these cases, when you use lithium aluminium hydride, we get the formation of product which is alcohol. So we never really think of what happens when amide is reduced with lithium aluminium hydride. So let me tell you, lithium aluminium hydride is a reducing agent that reduces amide into amines. So the product is going to be CH3CH2 NH2. So here the product is primary amines. H3 CH2 NH2. So this is unlike what happens in other reduction of aldehydes, ketones, acid halides or even esters, where you get alcohol as the product, but not here. Next reaction, Br2KOH given along with the amide. Guys, what is this reaction? This is yet another very famous reaction. I'm sure whichever booklet or the notes you must have solved, at least 2 to 3 questions you must have found on this reaction. And this is nothing but Hoffman's bromamide degradation. Hoffman's bromamide degradation. So if this is Hoffman bromamide degradation, the simple trick that we do is we assume as if this CO have been lost, meaning this is undergoing decarboxylation. So upon loss of this CO, the only product left will be CH3NH2. So even this is forming primary amine. Next one, acetonitrile with lithium aluminium hydride. Well, cyanide will be reduced with lithium aluminium hydride followed by hydrolysis. So even this is going to produce ethylamine, CH3CH2NH2. So guys, all these option 1, 2 and 3 are resulting in the formation of primary amine everywhere. We have to just find out where they do not form primary amine. So if you look at this molecule, nitrogen is not at the terminal position like it was in all other cases. Which means if we carry out reduction of this molecule, instead of forming primary amine, this is going to result in the formation of secondary amine. So here the product that we are going to get is secondary amine, and this is our right choice because this is the only reaction which is not resulting in the formation of primary amine. So our right choice is answer option 4. Yes, everyone? I don't really see any question was that challenging yet, except this simple reaction, probably if you are not able to recall. Very well, guys. Next question. Well, once again, from general organic chemistry, or you can call it as from aromatic hydrocarbon. The question says, consider the following compounds or the species, and they're asking how many of these compounds are following Huckel rule of aromaticity. Well, let me remind you, when you talk about any given compound is aromatic, we simply apply and check whether it is following the 4 criteria or not. And what are those 4 criteria? I'll write in shortcut. If any compound is cyclic, planar and completely conjugated, CPC, cyclic, planar and completely conjugated, and following Huckel rule of aromaticity, which is 4n plus 2 pi electrons, then that compound we call it as aromatic. Where the value of n 0, 1, 2, 3, so on and so forth. Which means if I put the value of n as 0, 4 into 0, 0, meaning 2 pi electron. If you put the value of n as 1, 4 ones are 4 plus 2, 6 pi electron. If I place the value of n as 2, 4 2s are 8 plus 2, 10 pi electron. So simply, if any molecule is following the first 3 criteria, cyclic, planar, completely conjugated, and having 2 pi electrons, 6 pi electrons, or 10 pi electrons, or like that, we say that compound is following Huckel rule of aromaticity, and hence it is aromatic in nature, right? So I want you guys to have a look at the first molecule, naphthalene, having 5 pi bonds, meaning it has 10 pi electrons, which is, yes, it is a Huckel number. And all other criteria have been satisfied already, that it is cyclic, planar and completely conjugated. So yes, it is going to follow Huckel rule of aromaticity. The next compound, this lone pair that you see as of now in the given structure, they appear like they are in sp3 hybridized orbital. But do not forget that this lone pair are in conjugation with the pi bond. If you delocalize them, then the corresponding structure formed is going to be a permissible resonating structure, which means this lone pair are delocalizable lone pair. When any lone pair delocalizes, upon delocalization they form a pi bond. Hence, I am not going to count this electron pair as a lone pair, rather I'll count this as a pi bond. So 2 pi bonds which you already see, this lone pair upon delocalization going to form a pi bond. So I have got altogether 3 pi bonds. 3 pi bonds meaning it has 6 pi electron, which is again a Huckel number. And of course, all 3 criteria have been followed: cyclic, planar, because this 4 carbon you already see are sp2 hybridized. This upon delocalization is again going to form sp2 hybridized carbon only. So yes, this molecule is aromatic. Well, here we have got 4 pi electrons. So this molecule is anti-aromatic. If any compound follows these 3 criteria, cyclic, planar, completely conjugated, and having 4n pi electrons, that is having pi electrons in a multiple of 4, you call it as anti-aromatic, which is highly unstable compound. So this is not following Huckel rule. Likewise here, 1 pi bond, 1 electron pair. This electron pair is also in conjugation with the pi bond. Hence it is delocalizable. So I'll count this also as a pi electron. So 1 pi bond, this will form another pi bond, 2 pi bond, meaning 4 pi electron. 4 is not the Huckel number. This is also not satisfying the Huckel rule. Here, pi bond in conjugation with vacant orbital in conjugation with the pi bond. So I've got 2 pi electron. Everyone, if I put the value of n as 0, I'm going to have 2 pi electron, which is Huckel number. So this is satisfying the Huckel rule of aromaticity, but not this. Sixth is a very typical structure. I am sure your teacher must have taught you that this is not anti-aromatic, although it looks like. The reason being, the actual structure of this molecule, which is cyclooctatetraene, exists in a tub-shaped structure, the way I have drawn here. Because of which the whole molecule is not planar at all. If it is not planar, the pi bond cannot delocalize, because for delocalization to take place, there has to be planarity in the ring, which means this molecule, although theoretically looks like it is anti-aromatic, it is not, which means I am not going to count this as a part of even anti-aromaticity. And if you talk about Huckel rule, it is not following Huckel rule either. So this is not our correct choice. And there is an anthracene molecule given to us 3, 1, 2, 3, 4, 5, 6, 7. 7 pi bond meaning 14 pi electrons. Keeping the value of N as 4, 3. Keep the value of N as 3, 4, 3s are 12 plus 2, 14 pi electrons. Meaning the molecule, the number of molecule following Huckel rule are first, second, fifth, and the seventh one, which means there are total 4 molecules, meaning the right choice is answer option 2, which is following Huckel rule of aromaticity. Right, everyone? Okay. Moving forward with the next question. Guys, if you look at the next question given over here, identify the major product obtained in the following reaction. Well, if you just look at the question given to you, the reagent that they have mentioned is ammonical silver nitrate, AgNH3 twice plus and 3OH minus. This reagent is nothing but a Tollens reagent. This is nothing but a Tollens reagent. And guys, we know that Tollens reagent, Fehling solution or Benedict solution are the reagent which we use to distinguish aldehydes from the ketones. So only aldehydes can be oxidized by this Tollens reagent. So in the given substrate, this is ketone. So ketone will not be oxidized, but aldehydes will be oxidized to carboxylic acid. But not to forget, as soon as this will become COOH, carboxylic acid, since condition being alkaline, this OH minus ion will react with COOH and will abstract the H plus ion. So instead of getting COOH as the product, we are going to get COO minus as our product. So keeping this keto intact, and this becomes COO minus. Where do you see that? Here it is. This keto is intact and this is becoming COO minus, meaning the answer option 4th is an appropriate choice. Okay guys, next question. Trying to speed up a bit because yet there is discussion of physical and inorganic chemistry to come. So guys, next question, identify the final product D in this question. Alright, so they have given us aldehyde. With lithium aluminum hydride, we know the fact lithium aluminum hydride is a reducing agent, so it will carry out reduction without any change in the number of carbon atom. So aldehyde having 2 carbon, aldehyde upon reduction gives you alcohol with 2 carbons. So we will be getting CH3CH2OH. I'm highlighting one of the H, keeping the next reagent in mind. So we have got ethyl alcohol. Aldehyde reduced to alcohol with the same number of carbon. H2SO4 being dehydrating agent, it will eliminate water, sigma bond will shift back. Between the 2 carbon will be getting double bonds. So B obtained here is ethene. CH2 double bond CH2. That ethene obtained is further treated with HBr. So it is simply an electrophilic addition reaction. Well guys, upon addition of HBr to it, let's say the pi bond shifted on one of the carbon, making it anionic, which attacked at H plus, and this cationic carbon is then attacked by Br, so you'll be getting CH3 single bond CH2Br. That is ethyl bromide. So C here is ethyl bromide. CH3 CH2Br. This ethyl bromide is then further treated with bromobenzene in presence of sodium metal. Guys, what reaction is that? If one of the substrate is alkyl halide, other is aryl halide in presence of sodium metal, yes, we call it as Wirth-Fittig reaction. Wirth-Fittig reaction will result in the formation of alkyl benzene. So you can simply consider reaction like this: ethyl bromide plus sodium metal. Let's say we are taking 2 molar equivalent of sodium metal and bromobenzene. So while representing the product formation, we eliminate NABR here and we'll be getting ethyl benzene as our product. So where do you see that ethyl benzene? Here it is. Option B, indicating formation of the desired product, that is ethyl benzene. So option 2 is an appropriate choice. Guys, next question. Well, you have been given an ether and they are saying you have to treat that with HI. So this is action of halo acids on ether, a very common reaction. and we have studied all the possible cases. The possible cases says that whenever you have an ether, the step number 1 will be the lone pair of ether will attack at H+, resulting in the formation of oxonium ion, so you'll be getting OH+. Now whenever you make this OH+, since oxygen is becoming trivalent, any one of these two bonds has to be broken, either this or this. But bond breaking is only favored when the corresponding carbocation form is stable. So let's say if I break this bond towards oxygen, you will see this carbocation form is going to be a benzyl carbocation, and benzyl carbocation being resonance stabilized, we will form a carbocation, which means the breaking of either this or this bond can happen only when the corresponding carbocation form is either tertiary or resonance stabilized. Like here it is a resonance stabilized, which means since there is a formation of carbocation, this reaction is following SN1 reaction mechanism path. And once carbocation is formed, now this I- will attack here to give you benzyl iodide, and if you see this product is phenol. Benzyl iodide and phenol. So guys, benzyl iodide and phenol have been given in answer option 4. Hence, this is our right choice. Guys, are you all understanding here? Yes? Perfect. Great. The next question. Well, guys, among all the questions which were given, I think this was the only question which probably must have given you little hiccups, because there is again a little contradiction in terms of what exactly are they trying to say in this question. Why? I'll explain it to you. They are saying which among the following will be the most readily dehydrated under acidic condition. We know the fact that whenever we have been given any alcohol under acidic condition, we expect protonation of alcohol to take place, and upon protonation of alcohol we get formation of ROH2 plus, and OH alone is not a good living group, but as soon as it becomes OH2 plus, it is a good living group. So the bond breaks and it results in the formation of carbocation. This formation of carbocation is found to be the rate determining step and the slowest step, which means the rate of a reaction depends upon which of the following alcohol is going to give you relatively stable carbocation. Now I said that probably this question might have given you hiccups because in many of the cases you must have considered the formation of thermodynamic stability of product. I mean, which alkene formed is more stable. So that way if you see the stability of alkene, you might end up writing 4 as your answer option, because if you remove OH from here and H from here, you'll be getting formation of double bond, which will be in conjugation of NO2. But considering the kinetics of reaction, because rate determining step involve formation of carbocation, we have to see among all the option given which alcohol is going to form relatively stable carbocation. So guys, if you check option 3 here, in this which we have got diol, if we protonate this OH and if departs from here, resulting in the formation of carbocation here, this carbocation which is formed will have hyperconjugation stability of 3 alpha H's from here and 2 alpha H's from here, altogether 5 alpha H's. No doubt that to this carbocation there is minus I of oxygen which is destabilizing the carbocation. But in all of the cases, if you consider the formation of carbocation where OH is present, there is even stronger electron withdrawing group present. And hence, in my opinion, these options 1, 2 and 4 will produce relatively less stable carbocation. And the relative stability of carbocation of option number 3 is going to be more stable the carbocation, faster the rate of formation. Hence, I would say option 3 alcohol will undergo dehydration readily. So guys, answer option for me here is number 3. Next and the last question from organic chemistry. Like I said, never underestimate those small chapters, guys. So this question is from environmental chemistry. It says, given below are two statements. Statement 1 is the nutrient deficient water bodies leads to eutrophication. Statement 2, they are saying eutrophication leads to the decrease in the level of oxygen. I will add on a point to this. Dissolved oxygen, we call it as DO, in the water bodies. In the light of above statements, choose the correct answer from the options given to you. Guys, I want you all to first recall what is the phenomena called as eutrophication. Well, let me tell you. eutrophication is the process where basically there is excessive growth of plants on the surface of water, or you can say rather excess of algal growth on the water. Now, because of this excess of growth of algae or plants on the water surface, it does not allow the enough concentration of dissolved oxygen in the water. As a result of lesser concentration of dissolved oxygen in water, it becomes harmful for the aquatic life. Like many of the fishes cannot survive because there is no enough dissolved oxygen present. So remember, the excess of growth of plant takes place due to nutrient rich bodies, especially elements such as nitrogen and phosphorus. So here they are saying nutrient deficient. So this is an incorrect statement that they're saying. It has to be nutrient rich water bodies that leads to eutrophication. So statement 1 is incorrect, but 2 is correct absolutely, because 2 says eutrophication leads to decrease in the level of dissolved oxygen. Yes, and that eventually results in the harmful effect on the aquatic life. So statement 1 is incorrect, but 2 is correct. Statement 1 is incorrect, but 2 is correct. Statement 1 incorrect, but 2 is correct, which means answer option 1 is a correct answer. So guys, that was all about the chemistry, or rather I'll say organic chemistry paper from NEET 2023. Well, all the questions I feel were quite moderate. Most of them were quite easy. The only question which I found was little difficult or something which was challenging was the dehydration of alcohols. So guys, that is it from my end as far as organic chemistry is concerned. From the team of Physics Wallah, I'm hoping that you had a wonderful day today and you guys are going to shine like a diamond. I'll see you soon in the next class. Thank you so much everyone for attending this class. Thank you everyone.
Oh, so sorry, I was on mute. So can you hear me now? No, this is not bio, this is chemistry. So I'll start chemistry. I've started inorganic chemistry. Can you hear me now? So we are starting inorganic chemistry. So if you can hear me, just let me know in the chat so that I can know that I'm actually audible to you. Okay, so let's just start. So my first question was, amongst the following, the total number of species not having 8 electrons around the central atom in its outermost shell. So there are two possibilities here. The first one is where the central atom has less than 4 bonds. That means the total number of atoms is less than 8. It is not 8. And the other possibility is where you have more than 8. In both the cases, in less than or more than, you don't have 8 atoms around the central atom in the outermost shell. So let's just start with the first one. So first one is ammonia. So you have NH3. So let me just write it in a different way so that it is very clear to you. So you have nitrogen, to which 3 hydrogens are connected. I'm not drawing the exact structure. I'm just drawing some rough structure. So nitrogen has one pair of electrons, 2 electrons on the central atom. So if you count the number of electrons, you have 2 electrons here, 2, 4, 6, and 8. So this has complete octet. So this cannot be your answer. The second one is AlCl3. So aluminium is from group number 13. It has 3 bonds with 3 chlorine. Now it has 1 vacant orbital. So total number of electrons around the central atom is 2 from here, 2 more from here, and 2 more. Total it is 6 electrons. So total 6 electrons, which is not 8. So this is one of your answer. So let's just start with the next one, which is beryllium chloride, BeCl2. So again, beryllium has only 2 bonds and no lone pair. So these are the first 2 electrons and the next 2, total 4 electrons, which is again not 8. So this is your answer. The fourth one is CCl4. It has 4 bonds with the central atom. It is not Cl, it is C at the center. So the fourth chlorine is here. So the central atom, central carbon now has 4 bonds. So it has 4 bonds. So just count the number of electrons, 2, 4, 6 and 8. So total 8 electrons are there. So this has 8 electrons. So again cannot be your answer. The next one is PCl5. So let's just write PCl5. Phosphorus, 2 chlorine on the vertical bond and 3 on the equatorial bond, Cl, Cl and Cl. So this is the structure of PCl5. Now again count the number of electrons. These are your first 2 electrons. Next 2 electrons, 2, 4, 6, 8 and 10. Total you have 10 electrons here. So 10 electrons is again not 8. So this is also your answer. So out of these 5 compounds that were given to you, how many are the answers? There are only 3. So it is option 2, which is 3. Option is 2, answer is 3. So I hope that is clear. Let's just move on to the next question. So the next question is the correct order of energies of molecular orbital of N2. Now the first thing that you have to do is count the number of electrons in the molecule. One nitrogen, first of all, nitrogen has atomic number 7. So each nitrogen that is there in the molecule will have 7 electrons. So there are total 2 nitrogen atoms. So total 7 into 2, which is 14 electrons are there. Molecule has 14 electrons. Whenever you have 14 electrons, there is something called sp mixing taking place. Sp, I should actually write a hyphen there. Sp mixing takes place, and because of this sp mixing, the energy of pi orbitals will be less than sigma bonding orbitals. So pi bonding orbitals will have less energy as compared to sigma bonding orbital. That is true for 2px and 2pz. So this order will be there. Pi will have less energy and sigma will have more energy. Everything else here is exactly same. So you cannot actually differentiate them based on the first 4 orbitals because they are exactly same. After that, your pi bonding orbital will have less energy than sigma. That is true here, but not true in this case. So not the answer. In the first case also, if you see, the sigma has very high energy. It is even more than like anti-bonding orbital, which again is not possible. So this is also not your answer. And if you see this option, you have again sigma less energy as compared to pi. So not the answer. So the only one which is true is option number 2. So option number 2 is the answer. So let's just move on to the next question. The solubility of Cu2 plus is more than Cu plus. Now, how do you find what is the exact reason behind this? The reason is basically solvation of these ions in the water. Reason for this to be more stable is solvation energy. So basically what happens is you have Cu2 plus, and Cu2 plus actually attracts water. Now water has dipole. This is your water molecule. There is a partial charge separation here. There is oxygen, which is more electronegative as compared to hydrogen. So there is partial negative charge. And because hydrogen is less electronegative or more electropositive, it will have partial positive charge. Now this copper 2 plus will attract the oxygen of water. So water will be attracted towards Cu. So because of the partial negative charge. And because of this, there will be some energy released, and that energy released because of this interaction of water and the ion is called solvation energy, because water is the solvent. In case of water, specifically it is called hydration energy. Hydration energy. Now look at the options. The reason is hydration energy. Because of this, Cu2 plus getting dissolved in water and the interaction of water with the Cu2 plus ions. Let's move on to the next one. So here again we have a reference in NCERT. So if you see this, this is your reference from NCERT. So what happens here is, it is clearly written. The reason here is basically inert pair effect. Inert pair effect. And what happens because of inert pair effect is, if you go down the group, the stability of lower oxidation state increases and stability of higher oxidation state decreases. That is very important for us. So I'll just write that statement. Stability of lower oxidation state, which in group 13 is +1. So stability of lower oxidation state increases. So, and you can also see thallium is more stable in +1 oxidation state as compared to +3 oxidation state. So let's just get back to the previous question. I mean, this question that we had. All you have to do is find the oxidation state of every metal atom that is present in this options, and then wherever you have group 13 element at +1, it should be lower in the group, and if it is in +3, it should be higher in the group to be more stable. So this is TL +1, this is +3, so it is thallium +1 +3. This is also +3 +1. +3 is less stable than +1. So this cannot be your answer. So first thing that I'll do is eliminate this option. Then go for the next option. The first one was true because +1 is more stable. Lower oxidation state, the stability of lower oxidation state increases if you go from top to bottom in the group. So this is more stable. That is very obvious. Let's move on to option 3, where the oxidation state is +3 and +1. So now what is more stable here? Because I know that down the group, stability of +1 increases and you have +3 given as more stable. So this is also incorrect statement. Let's just move on to the next option, which is like +1 and +3. Aluminium is the second element of the group where +3 charge should be more stable again. Because of that reason, this option also cannot be the answer. So option 1 is the only option where you have the correct statement, which is lower oxidation state is more stable for any element that is present lower in the group. So it is option 1. Let's move on to the next question. Next question is, which of the following statement is not correct? So what I'll do is, I'll find the correct statements and just eliminate them, and then we'll get the incorrect statements together. So I'll start with the first statement, which is hydrogen is used to reduce heavy metal oxides to metal. Yes, this is true. This is used in tungsten. So in elements like tungsten is a very heavy element. So in elements like tungsten, all you have to do is pass this hydrogen gas through this oxide of metal that you have, and it will be reduced. So I know that is true. So this statement is a true statement, so cannot be my answer, because in the question they have asked the statement are not correct. Because it is not correct, so this first one cannot be my answer because it's a correct statement. The next one is heavy water is used to study reaction mechanism. Yes, it is. The classic example of this that I remember is from, again, organic chemistry, where you have a reaction between carboxylic acid and alcohol. Let's suppose this is O18. So this is a reaction that we generally talk about, where if you write the product, the ester will have that oxygen which was present in alcohol. So oxygen 18, which is an isotope of oxygen, is used in studying the reaction mechanism. Similarly, if you have like a reaction where instead of hydrogen in the water, if you take D2O, because of the difference in the mass number of these two elements, I can exactly identify where this deuterium has gone, and that is how I'll know the mechanism. So let's just go back to the question. So heavy water, which is D2O, is used in this study, is used to study reaction mechanism. That is true, because I can identify clearly which one is hydrogen and which one is deuterium. So this is also a correct statement, so not the answer, because the question has not correct statements. So let's move on to the third option, which is hydrogen is used to make saturated. What is saturated fatty acid? It is a molecule where every pi bond is broken and hydrogens have been added to it. So if you see this statement, this is also true, because if you have to have a saturated molecule, you just need to break the pi bonds and add hydrogens. So that is also a true statement, so cannot be the answer. By eliminating these three, you can obviously tell which one is the answer. Now, in the options, just guess which one is the answer. See, not correct is asked. So these three are correct statements, so not the answer. So you have to tell me in the chat which one is the correct option. So there is only one option which doesn't have A, B, C, because there is A, B, C here, not the answer. B is there, not the answer. B is there. C is also there, not the answer. So there is only option 4 which is left, but still we'll see the statements that is given to me. The hydrogen-hydrogen bond dissociation enthalpy, that means bond energy, is lowest as compared to a single bond between two atoms of any element. That statement is not true. For hydrogen, it is the most stable. So it will have the highest dissociation energy for any sigma bond. So this statement is incorrect. So that is one of your answers. So option D is one of the answer. So that is there in these three options. Then go for the last statement here, which is hydrogen reduces oxides of metal that are more active than iron. This statement is not fully true, because it also reduces every element which is like higher in the electrochemical series than hydrogen, not iron. So that is the reason this statement is not fully true. So this is also your answer. So it is D and E, and yes, you are correct. You are telling in the options, whatever option you are telling is correct. It is option 4 which is the answer. Let's move on to the next question, which is homolyptic complex. First of all, what is homolyptic? Homo means same, lyptic means ligand. That complex which has same type of ligand is called homolyptic complex. The first thing that I'll do is explain this. A complex that has only one type of ligand. So how will you understand if a complex has only one type of ligand? All you have to do is check the name. In the name, if two different ligands are mentioned, that is not a homolyptic complex. So homolyptic complex from the following complexes is. So it's very simple. Triamine is the first ligand that you have, which is NH3. There is 3 times NH3. You don't have to count the number. So just write NH3, then you have triaqua. So there is water also, 3 water and 3 NH3. Because I can see 2 different ligands here, so this is not the answer. Let's go to the next question, which is potassium. It's useless. It's a cation. It's not a ligand, so just useless. So trioxalato. Oxalate is one of the ligand that I have, which is C2O4 2-. Aluminate is also not the ligand. So there's only one ligand there. So it should be the answer. But still what I'll do is, I'll look at the other options so that it is very clear to me. So diamine, amine is nothing but ammonia. So it is NH3. And then you have chlorido, which is Cl-. And you also have nitrito N. So which is NO2, where nitrogen is the one that is directly connected to the metal. So you have 3 different ligands, so cannot be the answer. So this is also not the answer. So next one is penta. What is written here? Pentaamine should be there. Pentaamine, carbonato. Amine is NH3. Carbonato is CO3 2-. So you have 2 different ligands. So again, this cannot be the answer, because in the question they have asked which one among this is a homolyptic complex, which means the ligands, the number of different types of ligands should be exactly 1. So the option is 2. So I hope that is clear. So let's move to the next question, which is the element expected to form largest ion to achieve the nearest noble gas. So first thing that I observe here is everything will form a configuration like argon. So it is argon 10, which is the nearest noble gas for all these. So it will gain or lose electrons to form the electronic configuration of argon. So all you have to do is, in the first one, just remove an electron, so minus electron, which makes this Na+. So I'll just write it here, Na+. The next one is oxygen. Now for oxygen, you'll have to add 2 electrons. So when you add 2 electrons to it, it will become O2 minus. Then you have fluorine, which is F minus. And then you have nitrogen with 3 minus charge, N3 minus. Now, how will you decide which one is the largest? So I use the technique here, and the technique is radius is inversely proportional to. Now what I do is, see, if I have -1 charge and +1 charge, the amount of charge in these two ions are exactly same. The sign is different, but the amount is same. But what I do is, I'll include this sign. So this is -1 and that is +1, so this will be lower. So radius is inversely proportional to charge with sign. Charge with sign. And this is only true for isoelectronic species. Isoelectronic species. Now what is isoelectronic species? That ions or atoms which have same number of electrons. So these have same number of electrons, and this has the least amount of charge because it's -3, because I have to include the sign. So this is -3, so this will have the largest size. So the answer is 4. So option 4 is the answer. It is including sign. So sodium is +1. So it has the highest charge. So size is the least. And in the question, they have asked largest ion. Sodium plus will be the smallest one, not the largest one. So let's move on to the next question. Now in this question, you have intermolecular. That means bond between two different molecules. Intermolecular forces are forces of attraction and repulsion between interacting particles that will include. So it is intermolecular, the bond between two different molecules. Dipole-dipole movement can be between two different atoms. Dipole-induced dipole, this is part of van der Waals force. So these two, and this dispersion force is also part of van der Waals force. So you have three things which are part of van der Waals force, which can be between two different molecules. Hydrogen bond can be of two types. Which the first one is intermolecular and the second one is intramolecular. So you have intermolecular hydrogen bond possible. But covalent bond, if there is a covalent bond between two different atoms, it will always be in the same molecule. So because it is in the same molecule, this bond cannot be considered as intermolecular force. So except D, everything else is the answer. So it is A, B, C and E. So it is this option which is the correct one. It's option 4. So I hope you are understanding things. So let's just move on to the next one. So the first thing is coke. Coke is a very good reducing agent. Coke is a very good reducing agent used in blast furnace. So this one is used in blast furnace. It's a reducing agent. So A is connected to 3. So it's either option 1 or 2. So you have already eliminated 2. Even if you have taken a chance and marked one of the options, you would have got the answer. But let's see the other options as well. Then you have the second one, which is diamond. Diamond is a molecule where you have 4 different bonds connected to a single carbon atom. So it is sp3 hybridized. So carbon atoms are sp3 hybridized. It is correct for diamond. So it is B is connected to 1. This is B4. So again, option 1 is eliminated. B1. So I've already eliminated 3 of the options. So one of them is left and that is the answer. But let's just complete the question fully. You have fullerene, which is like a football. So it is like a football. So it's like a cage-like molecule. So it is C connected to 4, and graphite is basically dry lubricant. So D to 2. So it is C4 and D2. So it is option 4, which is your correct answer. So let's just move on to the next question. I hope this is clear. If you have any doubt, you can ask me in the chat. Let's move on to the next question. Among the given options, which of the following molecules or ions act as a Lewis acid? Now, what is the condition for a molecule to act as a Lewis acid? It should not have excess of electrons. That means it will have some kind of positive charge or vacant orbital. So there should be positive charge or vacant orbital. Vacant orbital. So this is the condition. So there is no positive charge here. So you cannot get the answer by just looking at the positive charge. Now look at the vacant orbital. There's no vacant orbital here on oxygen because oxygen has one bond with hydrogen and it has 3 lone pairs here with a negative charge. So generally that which has a negative charge will never be a Lewis acid. So you have OH minus, not a Lewis acid because there's no vacant orbital. Now the next one is ammonia, which is a very good example of Lewis base. So it cannot be Lewis acid. The next one is water. Water also doesn't have any vacant orbital. It has 2 lone pairs here, so no vacant orbital. So these 3 are already eliminated. Now just go for option 4 as the answer. But let's just see the exact structure, which is BF3. And if you look at this boron over here, boron only has 3 bonds and no lone pair. So what is the number of vacant orbitals on this? It is 1. There's 1 vacant orbital on this. And because of this vacant orbital, this molecule behaves as a Lewis acid. So this one is a Lewis acid. So option 4 is your answer. So I hope this part is clear. Let's move on to the next question. Which one of the following statement is correct? So this is from your inorganic chemistry. So magnesium plays a role in neuromuscular function and intraneuronal transmission. It is calcium actually which plays a role. It's calcium, so not the answer. The daily requirement of magnesium and calcium in the human body is estimated to be 0.5 to 0.3 gram. That is true. So let's just mark this as true, but look at the other options as well. All enzymes that utilize ATP in phosphate transfer require magnesium as the cofactor. It is magnesium, that is not cobalt. So option 3 is also not the answer. So let's move on to the option 4 that we have. The bone in human body is an inert and unchanging substance. That is not true. Even if you have some fractures in your bones, like I had multiple fractures, so that fracture is actually healed. So that healing will take place because there is some changes. So again, option 4 is eliminated. So you have only one option, which is option 2, which is the answer. So I hope that is clear. Now in the chat, someone has asked what is a vacant orbital. See, this is the thing that is written. So I'll tell you what is a vacant orbital. A orbital where there is no electron. Orbital where electrons should have been there, but there is no electron there is called a vacant orbital. So let's move on to the next question. Given below are 2 statements. One is labeled as assertion A and the other is labeled as reason R. Okay, that's nice. Assertion A, metallic sodium dissolves in liquid ammonia giving deep blue solution which is paramagnetic. That is true. On leaving this paramagnetic solution for very long, it becomes diamagnetic and bronze in color. So this statement, this assertion is fully true. The reason given here is the deep blue solution is due to formation of amide. There is formation of amide, but the exact reason for this is the basic electrons that are present. So this statement is true, but this is not the correct explanation of this. So what I'll say is the paramagnetic nature is not the explanation because there are no free electrons there. So what I can say is both A and R are true, but R is not the correct explanation because the color is basically due to the paramagnetic nature is due to the electrons present there. So it's option 3, which is the answer. So let's start the next question. Here you have a lot of molecules. You'll have to know how to name these molecules, write the structure, only then you can get the answer. So let's just start writing the structures first. The first one is peroxo disulfuric, it should be peroxy disulfuric acid. Now, first of all, I'll write the structure of peroxy disulfuric acid. It looks something like this. Double bond O, OH. There will be oxygens. There will be one more sulfur with 2 double bond O's and 1 OH. So that is the molecule. That is the exact molecule that you have. Now just look at the options. So it should have 2 oxygens connected together. So there is only one option where it is there. So it is A with 3. So let's just eliminate the options. So option 1 and 3 can be correct, but 2 and 4 cannot be correct. Let's move on to the next question, which is sulfuric acid, which is also very famous. So I'll just write the structure of sulfuric acid, which looks something like this. This is sulfuric acid. This is your sulfuric acid. So as soon as you write the structure of sulfuric acid, so what you do is you look at the number of double bonded O. So there are 2 double bonded O here and 2 OH here. So look for that. There are 2 double bond O and 2 OH. So second, B is connected to 4. Now again look at the options and try to eliminate them. Option B is 4 is true for both of them. So again you cannot actually eliminate the options. So move to third one, which is pyrosulfuric acid. There is a very special name for this. Can you guess the special name of this? It is used in inorganic chemistry, organic chemistry, and it is used basically for sulfonation. And the molecule is called oleum. So I'll just write the structure of oleum as well. So it will be sulfur, oxygen, sulfur, double bond O, double bond O. There are 2 more double bonds here and OH. So it has 2 OH group, 4 double bond O and 1 sulfur oxygen sulfur bond. Sulfur oxygen sulfur, 4 OH and 4 double bond O and 2 OH. So pyrosulfuric acid is connected to 1. So C is 1. So option 3 is the answer. You can clearly eliminate the options and get the answer. So just mark option 3 as the answer. So we'll move on to the next question. Which complex compound is most stable? Now all you have to do is, if you look at the ligands here, one of the ligand here is a chelating ligand. So if you see this one, there is cobalt. What is the charge here? It is 3+, and it has En, which is ethylene diamine. And if you write the structure of ethylene diamine, it is basically NH2, CH2, CH2, NH2, and then there's a bond here. There's a 5-membered ring here, and because of this 5-membered ring, there is extra stability that is provided to this complex ion. So this complex is very stable because of chelation, and that is your answer. So it is option 4, which is your answer. So now we'll move to the next question. So next question is, which of the following statements are incorrect? Again, they have asked which one is incorrect. So what you have to do is, you have to look for the correct statements and just eliminate them. So let's start with the first statement. First statement is, all the transition metals except scandium form metal oxides which are ionic. That is a true statement, so not the answer. Incorrect, so not the answer. So I can directly eliminate option 2. So option 2 cannot be the answer. The next one is, the highest oxidation number corresponding to the group number in transition metal oxide is attained in Sc2O3 to Mn2O7. That is true. That is true. From group number 3 to group number 7, it is true. So this statement is true. So again, not the answer. So B cannot be the answer. So option 1 is eliminated. Option 4 is also eliminated because B cannot be the answer. So there's only one option left in front of you. So even without reading all these statements, by reading these 2 statements, you can get the answer directly. And that is what is this. So the last option, which is C and D, is the answer, because everywhere else you either have A or B, which cannot be the answer. So it is a very good technique to use in your exam, because you don't have to read the complete question. Without doing that, you're actually getting the answer. So that is what I've done here. So this is the statement where you can see the group number is attained in SCO3 and MnO2. So this is a correct statement. So you had to actually eliminate it. So let's just move to the next question. So there's a question here. The reaction that does not take place in a blast furnace between 900 Kelvin to 1500 Kelvin temperature range during extraction of iron. During extraction of iron, that means it is blast furnace. So if you are using a blast furnace, which of these reaction will not take place? So we have that NCERT exactly in front of us. So if you see 900 to 1500 Kelvin, you have FeO turning into Fe, C turning into CO, CaO reacting with SiO2 to form Thomas slag. This is CaSiO2, very important, slag. So this formation of slag is actually present, does not take place. So cannot be the answer because that takes place. FeO is converted into Fe using CO. That is also there, so not the answer. And the last one is carbon plus CO2. That is also there, so not the answer. So just eliminate the option to get the answer. So final answer is option 2. So now let's just move on to the next question. So now we have the questions of physical chemistry. Now you can use a lot of formula to get the answer directly. So let's just start with the first question. The first question is, which one is an example of heterogeneous catalyst? Now what is a heterogeneous catalyst? A heterogeneous catalyst can be very easily identified if the state of the reactants and products is different from the state of the catalyst. It is called as heterogeneous catalyst. So all you have to do is decide if the compound that is given to you is solid, liquid, gas or aqueous. If you can do this, you can directly get the answer. The first one is combination between dinitrogen, which is N2. What is the state? It is gaseous state. And dinitrogen, which is H2. Now what is the state here? It is again gaseous state to form ammonia. Now what is the state of ammonia? This is Haber's process. So the state will be gaseous state in presence of finely divided iron. Iron is a solid particle. So you can see this is in gaseous state. The reactants and products are in gaseous state. The catalyst is in solid state. So it is an example of heterogeneous catalysis, and directly you can mark this. So that is how you can mark this. And then we'll move on to the next question, which is given below are two statements. Again, you have assertion reasoning. Let's just look at the question. Labeled as assertion A and other labeled as reason R. So let's just read the assertion and the reasoning. So assertion A, in equation, there is a equation given to you. First of all, you have to check if the equation is correct. So delta G equal to minus NF E cell, that is true from electrochemistry. Value of delta G depends on N, that is also true. You can see there is a relation here. There is an equation given here. So if you change the value of N, delta G will also change. So the first statement is completely true. So true statement. The next statement is reason R. E cell is an intensive property, that is true. So if you use different cells, different types of cell having the same half cell, E cell will be exactly same. So E cell is an intensive property and delta G is an extensive property. This statement is also true. Both the statements are true. Now the question is it a correct explanation or not? It is a correct explanation. It is a correct explanation because this is the exact thing why I use this equation, because I cannot compare E values directly. I use delta G. So that is the reason this is the correct explanation for that. So I can write both A and R are true and R is the correct explanation of A. So option 2 is correct. Let's move to the next question, which is the relation between NM. So they have given some NM, which is NM equal to the number of permissible values of magnetic quantum number M for a given value of azimuthal quantum number. How do you get the permissible values? So there is a very good equation. So I'll just write it. Number of permissible values equal to 2L plus 1, which I write number of different values of M is 2L plus 1. From here, I'll just do some manipulation. Take the 1 to the other side. You'll get NM minus 1 equal to 2L. Now just divide it by 2. You get NM minus 1 divided by 2 equal to L. So L equal to NM minus 1 by 2. So it is option 2, which is the correct option. So all you had to do was write this equation and get the answer. Now the next question, which is for a certain reaction, the rate is given. This is rate law expression. This is called rate law expression. This is used to determine how fast the reaction is and how will the rate of reaction change if you change the concentration of one of the components. When the initial concentration of A is tripled, keeping concentration of B constant, the initial rate would... So I'll just write that. So what you do here is take the ratio of rate of reaction, and from there you can solve it. So let's suppose the initial rate of reaction was Ri. So Ri will be equal to rate constant, will not change if you change the concentration. So just write K into concentration initial. I'll just write it as AI. So AI or A1, whatever you want to write, power 2, and concentration of B, let's just keep it as B1 power 1. So let's just remove that 1. So I have to take the ratio of rate. Final rate is, let's suppose it is RF. Again, divide this. Now again, rate constant will not change with the concentration. So it is K. Concentration of A has now tripled. A is tripled. So instead of writing A1, I have to write 3 times A1 and power 2. And concentration of B is constant. Concentration of B is constant. So just write B1 there. Now just cancel things off. B1 will get fully cancelled. A1 will get cancelled. K will also get cancelled. So if you write this equation, it will become R1 by RF equal to, nothing is there in the numerator now. So it is 1. In the denominator, it is 3 square into 1. So just write 3 square, which is 9. So from here, if you take 9 to the left side and RF to the right side, what you have is 9 times R1 is RF. So the final rate of reaction is 9 times the initial. All you have to do is check the option now. Increases by, because it is 9 times, increases by a factor of 9, because I can see the number 9 here. So it is option 4, which is the answer. So all you had to do was take the ratio of rate of reaction. From there, you could have easily got the answer. So now we'll move on to the next question, which is a compound is formed by 2 elements A and B. Now here, there is a very important thing. You can use some kind of shortcut trick, which I generally like to use, because you can see number 3 here. So what I'll do is I have this freedom of assuming any number of B present in the cubic closed packing. So if I use 3 as the number, I'll get the answer in very few steps. But let's just not start with that as the method, but I'll just show you how you have to find using that method as well. So I'll use my first method. So it is method 1. So in method 1, what we do is B forms cubic closed packed structure. So I know it is cubic closed packed structure, and the effective number of atoms in a cubic closed packed structure is 4. So B will be equal to 4. And A occupy 1/3 of tetrahedral voids. Now how do you get the number of tetrahedral voids? Number of tetrahedral void is 2 times the effective number of atoms. So effective number of atoms is 4. 2 times effective number of atoms is 2 into 4. But only 1/3 of it is occupied. So just multiply with 1 by 3. So if you multiply all this, it is 8 by 3. So this becomes 8 by 3. So if you write this formula, it will look something like this. A 8 by 3, B 4. Now what you have to do first is multiply by 3 on both the sides. You get A 8, B 4 into 3 is 12. Now again you can see 4 common in both the cases. So divided by 4. So it becomes A2, because 8 by 4 is 2, and B3. So it is 3 plus 2, which is option 2, which is 5. So 3 plus 2 is 5. That is the answer. Now what I'll do is I'll show you the shortcut. Shortcut would have been B is 3. How did I decide that number 3? Because I could see 1 by 3 here. So that my calculation is simplified. I'll take this number as the effective number of atoms. I'll get the answer very easily. A is equal to 1 by 3, because 1/3 of tetrahedral voids is occupied. So 1 by 3 into how many tetrahedral void per atom? It is 2 times, twice. So it is 2 times 3, which is 6. 6 into this is 2. So I can directly get A2B3. I could have got the answer very easily, but still I've showed you the other method as well. So option 2 is your answer. So let's move on to the next question, which is given below are 2 statements. Again, it's an assertion reasoning question. One is labeled as assertion A, the other is labeled as reason R. The question is assertion A. Helium is used to dilute oxygen in diving apparatus. That is true. So this statement over here is true. The next statement is reason R. Helium is highly soluble in O2. It is actually insoluble in the blood, so it is false statement. So the first one is true, the second one is false. So it is option 4, which is the correct answer. Now let's move to the next question. Again, the question is select the correct statement. Now we have to select the correct statement. Till now we had many questions where we had to select the incorrect one. This is where you have to select the correct one. The question is, atoms of all elements are composed of 2 fundamental particles. This is incorrect statement, so not the answer. So again, I can eliminate option 2 and option 4. So now again, I have 50% probability. Even if you like mark one of them, you have 50% chances of getting it correct. So let's move on to the next statement, which is the mass of electron is 1.90939 into 10 power minus 31 kilo. This is a true statement. So B is a true statement. So this B is true. So just by looking at these two options, I can tell it is option 1. But let's just see other options as well. C is all the isotopes of a given element show same chemical property. That is also true. Protons and electrons are collectively known as nucleons. No, it is not true. Proton and neutron is considered as nucleon. So D is incorrect. E is Dalton's atomic theory regarded the atom as the ultimate particle of the matter. That is also a very true statement. So again, I've already marked option 1 because I could eliminate the options. So let's just move on to the next question, which is again, amongst the given option, which of the following molecule act as Lewis acid? So again, we have seen this question, which is option 4. So let's not discuss it again. So the next question is the right option for the mass of CO2 produced by heating 20 gram of 20% pure. As soon as you see percentage purity, there is a formula. Percentage purity is equal to weight of substance actually present in the mixture divided by weight of sample into 100. So I have been given this percentage purity, so use that number, right? 20 equal to weight of the compound actually present. What is the compound? It is CaCO3. So weight of CaCO3 divided by weight of sample is the total weight, which is 20 into 100. Now you'll have to do a little bit of calculations here. 25 times is 100, 4 times. So I can get the weight of CaCO3, which is 4 gram. Now, as soon as I get the weight, I can easily find the number of moles. How do you get the number of moles? Given mass by molar mass. Thankfully, the molar mass of CaCO3 is just 100 gram. So I can get the number of moles of CaCO3. CaCO3 is just 4 by 100. And because the coefficients are same for every molecule of CaCO3 that has reacted, you'll get 1 molecule of CO2. So I can say that this number is also equal to the number of moles of CO2 that is formed. So as soon as you know this, you can get the weight of CO2, weight equal to number of moles, which is 4 by 100 into molar mass of CO2 is 44. So 44 into 4 is 176 divided by 100. This number is 1.76 gram. So look at the option. There is option 3. So option 3 is the answer. So it is that simple. If you know the formulas correctly, just apply them, you'll get the answer. The next one is, among the following options is correct graphical representation of Boyle's law. Now what exactly is Boyle's law? It is PV equal to constant. So if you want to draw the graph between pressure and volume, it will be a rectangular hyperbola, which looks like this. So this feels as if it is true, but there is a condition for temperature given. And the condition here is, the real condition is, if you go away from the origin, this temperature should increase. So the real order of temperature should have been T1 greater than T2 greater than T3. This statement is not correct, so not the answer. So P versus V, there is P versus T. T is anyways not there in Boyle's law, so just ignore it. So now you have 2 options left. So in both the option, you can see 1 by V. So I'll just manipulate this equation. Let me write the equation again. So I'll make this as PV equal to, let's change the constant as some K. So we'll write PV equal to some value K. So I can write P equal to K by V, or I can write it as P equal to K into 1 by V. Now 1 by V is your x-axis, P is your y-axis, so it is in the form of y equal to mx. So it should be a straight line passing through the origin. Straight line passing through the origin, that is not a straight line, so not the answer. So out of 4 options I've eliminated, and this is also correct because it is the equation of straight line. So option 3 is the correct answer. Yes, you are right, option 3 is the correct answer. Then let's just move on to the next question, which is conductivity of, so they have given conductivity, which is kappa value, conductivity of centimolar, which is 0.01 molar solution of KCl at 25 degrees Celsius is, they have given this value. So kappa is 0.021, whatever is the unit. And the resistance of the cell containing the solution at 25 degrees Celsius, resistance is given as 60 ohms. So why am I writing the unit? Let's just remove the units because I know units are anyways matched properly. So what is the equation that you have to use? Equation is kappa equal to cell constant divided by R. So they are asking you the value of cell constant. So this is what you want to find. So all you have to do is take kappa, take R, multiply them, you'll get the answer. So G star, which is your cell constant, equal to kappa, which is 0.021 into 60, which is the resistance. So multiply with 10, so it becomes 0.21 into 6. So if you multiply with 6, it becomes 6, 6 to 12, so 1.26 centimeter inverse. So it is option 4, which is the correct answer. All you had to do was simple multiplication of the kappa value and the resistance to get the answer. I hope that is clear. Let's just move on to the next question. So next question is, given below are 2 statements. Again, it's an assertion reasoning question. We'll start from the assertion. A reaction can have 0 activation energy. This statement is true for free radical mechanism. This is a true statement. This is a true statement for free radical mechanism. Now go for the reason part. So reasoning is the minimum extra amount of energy absorbed by reactant molecules so that their energy becomes equal to the threshold energy is called the activation energy. This is also a true statement, but that is not the explanation of the first assertion. So it is both A and R are true, but R is not the correct explanation. So it is option 3 which is correct. So I hope this is clear. Now we have again a numerical. So first of all, what is the question? Question is the equilibrium concentration of, they have given the equilibrium concentration of species in the reaction. So I know the equilibrium concentration of all 4 is given as 2, 3, 10 and 6 molar respectively. Temperature is 300 Kelvin and delta G of the reaction is, they are asking me delta G and they have also given the value of R. So all you have to do is write delta G naught equal to minus RT ln K. So what is the value of R? R is given here as 2, so it is minus 2 into T is 300. Now because it is ln, I don't like using ln, I'll convert it into log. So how do you convert it? Just multiply 2.303 into log of, now what is the equilibrium constant? Concentration of products, which is concentration of C and D, concentration of C and D, which is nothing but 10 and 6 divided by concentration of reactants, which is A and B. Just write the concentrations, which is 2 and 3. Now I can clearly see that 2 into 3 is 6 and this 6 and the 6 in the numerator will get cancelled. So this is anyways cancelled. So now you have log 10. Thankfully, I know the value of log 10, which is 1. So I know that this whole thing is 1. Now I have to do this multiplication. So what I do is I'll just take this 100 from here, and multiply it with 2.303. So 3 into 2 is 6, so it is minus 6 into 100 into this number is 230.3. Now the calculation is relatively very simple. All I have to do is multiply with 6, which is relatively easier. So it is 6 3s 18. I have a decimal after it. 6 into 0 is 0 plus 1 is 1. 6 into 3 is again 18. And 6 into 2 is 12 plus 1 is 13 with a minus sign because minus sign was there in calories. So it is 1381.8. 1381.8. So it is option 4 which is the answer. So I hope that is clear. Now let's just move on to the next question. Now this is a question where you have to balance it. Now balance the chemical reaction can be done using n-factor method. So let's just start using n-factor method. Cr2O7 2 minus plus SO3 2 minus gives you Cr3 plus SO4 2 minus. The first thing that I'll do is find the oxidation state. Chromium here is in dichromate, so it is plus 6. Chromium there is plus 3. Sulfur here, because it is in sulfide, it is plus 4, and that is sulfate, it is plus 6. Sulfite is plus 4, sulfate is plus 6. So what is the n factor here? The change is only 3, but the number of atoms is 2. So 3 into 2 is 6. So n factor of this is 6. Now n factor of sulfur in SO3 and SO4, it is from plus 4 to plus 6. So there's a change of only 2. And what is the number of atoms of sulfur? It is 1. So it is 2 into 1, which is 2. Now what you can see is, you can see 2 common here. So just divide it by 2 in both the cases. So if you divide it by 2, which is the LCM of both the n factors, it is 3 and 1. Now these are the coefficient of the other molecule. So coefficient of SO3 is 3. Coefficient of this one is 1. From here you can see there are 2 chromiums here. So there are 2 chromiums in the product as well. There are 3 sulfurs here. So there should be 3 sulfurs in the product as well. So you have the coefficient. You know the value of A, which is 1. B is 3. So now with using 1 and 3, you know that only option 2 is the correct answer, but still will balance option, the last one as well. So you have H plus on the left side. So let me use a different color. You have H plus on the left side. You have water on the right side. Now if you see the number of oxygen atoms, everything will be solved. There are 7 atoms here. Plus 3 is 9. So 9 plus 7 is 16 atoms of oxygen on the left side. Here on chromium, there is no oxygen atoms. In sulfate, you have 4. 4 into 3 is 12. So you need 4 more to completely balance oxygen. So what you have to do is, you have to add 4 water. So as soon as I see that number, I know that it is C by 2. If I know that C by 2 should be equal to 4, so C should be equal to 8. And that is how I get the third option, third part, which is 138. And option 2 is the correct answer. That is how you have to solve it. So I hope this is clear. Now the next one is very simple. You have pumice stone, which is an example of, pumice stone is a stone which has a lot of holes. It is like a foam made up of solid. So it is solid sol. So option 4 is the answer. And the next one, what fraction of one edge center octahedral void lies in one unit cell? Now one edge center is shared by how many atoms? There are 4 atoms or 4 unit cell. 4 unit cell share this. Because 4 unit cells share this equally, all of them will have equal portion of the octahedral void. So how much will one of the unit cell have? It will be 1 by 4. Because 1 is divided in 4, which is nothing but 1 by 4. So option 4 is your answer. That is how you can mark very easily. Now let's move to the next question, which is again from thermodynamics. Which among the following option is the correct relation between enthalpy change, which is delta H, and change in internal energy, which is delta U. So what is the relationship between them? There is one relation which says delta H equal to delta U plus PV. Or if you don't like this, because there is no option like that, I can write PV equal to delta NGRT, or I have to actually write delta here. Delta H equal to delta U plus delta PV. If you don't like delta PV, because that is not easily like given in the question, so you can also write this delta PV as delta NG, which is the number of gaseous product minus gaseous reactant into RT. And everything else remains same. You have delta U, you have delta H. So you have delta H equal to delta U plus delta NGRT. Now just look at the option. It is delta H equal to delta U plus delta NGRT. So it is option 3, which is the answer. So that's it I had for this paper discussion. We'll meet in the next class. So bye-bye and take care. I hope you have done very well in this exam. Bye-bye and take care.